Let's start.
Oh, okay, sir. Heartiest greetings to all the delegates and participants. I, Manali Das, Senior Research Fellow in the School of Energy Science and Engineering, am extremely delighted to extend a warm welcome to the Indo-US workshop on recent advances in metabolic engineering for microbial cell factories, hosted by Indian Institute of Technology, Kharagpur, in association with the
Start. Now start.
Heartiest greetings to all the delegates and participants. I, Manali Das, Senior Research Fellow in the School of Energy Science and Engineering. I am Manali Das, Senior Research Fellow in the School of Energy Science and Engineering. I'm extremely delighted to extend a warm welcome to the Indo-US workshop on recent advances in metabolic engineering for microbial cell factories, hosted by the Indian Institute of Technology, Kharagpur, in association with the Washington University in St. Louis, USA, Arizona State University, USA, International Center for Genetic Engineering and Biotechnology, ICGEB, New Delhi, and CSIR National Chemical Laboratory, Pune. This workshop is organized under the Scheme for Promotion of Academic and Research Collaboration, or SPARC program, of the Ministry of Human Resources and Development, Government of India. This program aims to cover state-of-the-art research challenges and breakthrough technologies behind the current trends in the field of bioengineering technology. The objective of this workshop is to bring together national and international researchers to explore the myriad applications in the field of metabolic engineering for designing microbial cell factories. It will enlighten upon the cutting-edge tools, their applications, and associated limitations in systems and synthetic biology with customized tailoring of microbial cells for enhanced production of desired metabolites. We are very fortunate to have pioneer scientists from premier institutes of both India and the US who have contributed enormously in the flourishing of research in the field of systems and synthetic biology. And we anticipate that this workshop will create immense possibilities as well as generate opportunities in developing a global research platform towards biotechnological engineering. I would request the head of School of Energy Science and Engineering, Professor Siddharth Mukhopadhyay, who has been an integral part of the hosting of this event, to kindly inaugurate the workshop by delivering his note. Professor Mukhopadhyay has been associated with the prestigious institute since the last 40 years, and he is also currently the head of the Department of Electrical Engineering at IIT Kharagpur. In addition, he is the coordinator for the Indo-German Collaborative Research Center on Intelligent Transport Systems. Sir, please.
Namaskar. A very good morning to the Indian participants. A good evening to the US experts and speakers, participants, and a very warm welcome on behalf of the Institute and the School of Energy Science and Engineering. I am truly delighted that this workshop is happening for a good number of reasons. I was not, frankly, I was not so familiar with the area, but after Dr. Ghosh introduced me, I briefly looked at things, and it seems a very exciting and upcoming area, not only for research, but for a country like India. I remember, I recollect now that when I was a very young faculty, it'll be about 30 years back, there was those were very early days of patenting from academic institutions. And we had probably the first patent from the Department of Electrical Engineering on what was called at that time a seesaw bioreactor. It was a novel reactor which as far as I recall now, was trying to enhance the production of ethyl alcohol from yeast. And the group that worked on it, of course, worked in the context of control systems for that reactor. And I could see, and I vividly recall that it involved very, very sophisticated instrumentation and control for optimizing the production of those fledgling cells in the reactor. So it turns out that at some level, at the initial level, while it will probably involve a lot of science, not probably, surely involve a lot of science in the interface areas of biology, chemistry, genomics, maybe areas like systems biology, computational genomics, etc. There's a lot of engineering to come to it when you try to build it at scale and try to. So this workshop talks about a cell factory. That's interesting. The moment you talk about a factory and you take it up the scale, there are lots of other aspects of engineering. Like for example, my major area of research is actually control systems, and I felt excited to see that there's going to be a lot of control systems in this area, controls and instrumentation and AI and signal processing. So being an electrical engineer, it seems very exciting. It seems a very exciting opening vista to me. And this factory of cells is certainly a very exciting aspects of the future. We in engineering talk a lot about a term which is called Industry 4.0, which has been christened as the fourth industrial revolution, which is upon us, which is based on information, and the thought occurred to me that this cell factory could be called probably Industry 5.0. And there are so many applications. Energy can be an application. Energy will certainly be an application, but there's going to be a whole lot of other applications, including healthcare, including other aspects of the, let's say transportation. It's going to really impact a country like India, which is so rich in biological resources. So I'm sure that this is going to be a very, very attractive area of research, and I really congratulate all the institutions which have come together to conceive and finally deliver this workshop. I welcome all the participants. At the same time, I feel a little regret cannot be avoided that you could not come to see our beautiful campus right at this moment. Typically for such workshops, we invite people to our beautiful institute, and that's not possible today. But on the other hand, I'm sure that has increased the level of participation to a much higher level. So that's very heartening. So overall, I will not take much of your time. I welcome you once again to this workshop. I thank all the experts and speakers for contributing their knowledge. And I surely tell the participants that you are going to have an exciting day ahead. With this, let me inaugurate the workshop formally and wish it great success. Thank you very much.
Thank you very much, sir, for your delightful and motivational words. I now request the Dean of Postgraduates and Research, IIT Kharagpur, Professor P.K. Das, to kindly say a few words on this occasion. Professor Das is a professor at the Department of Mechanical Engineering at IIT Kharagpur and has been associated with the institute since 1987. Sir, please.
Hello everyone, am I visible and audible? There was some problem, so
yes, sir.
I have unmuted the microphone. Somebody has muted it. So, am I audible?
Yes, sir.
Am I visible?
Yes, sir.
Okay, good. So, at the outset, on behalf of this institute and on my personal behalf, I like to extend a hearty welcome to all the participants. As Professor Siddhartha Mukhopadhyay, the head of the school, told that this is a very important topic. So I can see this is a very exciting topic, this workshop, topic of this workshop. It is on metabolic engineering for microbial cell factories. And then going through a little bit deeper, I could see that it encompasses many kind of cutting edge aspects of biotechnology and bioengineering or bioscience in a broader perspective, which include metabolism and metabolic engineering, which include bioenergy, which includes enzyme. And I also can see that the participation is from different corners of our country. Even some participation is there from other countries and probably a few from industries, what I could find. See, I am not an expert in this field, but again, going through the booklet of abstract which you have kindly provided, I could see that there are many topics, and it is very heartening to see that in our country, so many diverse topics related to this broad theme is being worked very enthusiastically by different researchers in IITs, in different NITs and universities. Now, biotechnology, actually, if we see, there are, of course, there could be, it is not beyond debate, but people have tried to see the development of industry through different kind of industrial revolution. The first industrial revolution everybody knows, but then there are so many other things, and digital technology, now data science have come, and people also anticipate that the next industrial revolution is going to come in the form of biotechnology and bioengineering. Whether that is absolutely true or not, let us not go into that debate, but definitely the combination of machine learning, artificial intelligence, data science, and this biotechnology, that will be showing, or that will be leading in the field of industry for coming years. There is no doubt in it. The main challenges what the human civilization is facing at this moment, many of these challenges, at least scientists believe, can be met with the advancement of biotechnology, be it food, be it energy, be it affordable healthcare. So that's why this has got a very, this topic has got a very far-reaching effect. And as far as the activities of this institute is concerned, in this institute, in different department and center, the works related to this, as far as my knowledge goes, that is being carried out over the years. We know that there is a Department of Biotechnology, then there is Bioscience, and even in School of Medical Science and Technology related works are being done. There is a specific center for bioenergy, and we have also done on topics like microbial fuel cell and enzyme. So it is very heartening to see that IIT Kharagpur has organized such a workshop under the umbrella of Spark, and organizers are taken from different countries. So I wish all the success to this workshop. I wish the participants, where there will be a number of researchers who are trying to do invent new things. So they will be benefited through the lectures, by some deliberations during the lecture and beyond the lecture, and some sort of network will be established which will stay for long. Obviously, it would have been good that if we could have met face to face, but this kind of meeting also has got its own advantage, that many people can join without without much of expenditure. So that is a good part of it. And we have to get used to this kind of new norms, new new modes of interaction in the scientific and other forums. So, and we will learn also. This is one of the few online workshop, international workshop. So we are also learning from these kind of workshops, so that in future we can do it in a better way. So finally, I like to congratulate the organizers. I like to congratulate the participants, and also I want to extend my thanks to all the speakers and wish a grand success to this workshop. Thank you.
Thank you, sir, for your kind words. To quickly conclude the inaugurating session and start with the lecture series, I humbly request the coordinator, Dr. Amit Ghosh, to deliver the vote of thanks. Sir, please. Sir, the mic is unmuted.
Sure. Is it audible now?
Yes, sir.
So I'd like to invite all participants who have joined already online for their interest in metabolic engineering. I would like to thank our respected Dean Sir, Professor P.K. Das. In spite of his busy schedule, he graced this occasion and inaugurated our workshop in a grand way. And I'd like to thank for his valuable words, which will motivate the student to progress further in their life. I also would like to thank our head, Professor Siddhartha Mukhopadhyay, who has supported me throughout this workshop from beginning. Without his help, I would not been able to actually do this workshop in a proper way. I also would like to thank Professor Ghagrekar from head PK Bioenergy Center for his valuable support and encouragement. I also want to extend my heartfelt regard to the co-convener, Professor Ridhiman Dhar, who has supported me from the beginning, how to actually go about the workshop. It is also my pleasure to take this moment to express my gratitude to all the foreign speakers and the delegates, especially Professor Yinjie Tang from Washington University in St. Louis for his valuable time, who will be speaking the first lecture. He will be starting and then we have Professor Arzul Varman from Arizona State University. Then we have Dr. Sirish Srivastav from ICGB, and then we have Dr. Anu Ranganathan from NCL Pune. And then our respected Professor Pinaki Sir from Department of Biotechnology will also give valuable lecture in this workshop. So last but not the least, I would like to thank our CP team who is actually coordinating all these events like recording and then live this workshop into YouTube. And finally, I sincerely hope Kharagpur for arranging the virtual setup to conduct this workshop. I sincerely hope that all enthusiastic participants will benefit a great deal from the lectures to be delivered by leading scientists and their students. I wish all of you the best and a very good day. Thank you.
Thank you, sir. Since the first session will start in another 15 minutes, because the speaker is an international dignitary, I would once again like to give you a brief overview what this workshop is about. So this workshop is organized under the Scheme for Promotion of Academic and Research Collaboration, or the SPARK program of the Ministry of Human Resource and Development by the Government of India. This program aims to cover state-of-art research challenges and breakthrough technologies behind the current trends in the field of bioengineering technology. The objective of this workshop is to bring together national and international researchers to explore the myriad applications in the field of metabolic engineering for designing microbial cell factories. It will enlighten upon the cutting edge tools, their applications, and associated limitations in systems and synthetic biology with customized tailoring of microbial cells for enhanced production of desired metabolites. We are very fortunate to have pioneer research scientists from premier institutes of both India and the US who have contributed enormously in the flourishing of research in the field of systems and synthetic biology. The professors that have agreed to conduct these sessions are Professor Yinjie Tang from uh, University of Washington, St. Louis, Professor Arul Varman from Arizona State University, Dr. Shiri Srivastava from ICGEB New Delhi, and Dr. Anu Ranganathan from NCL Pune. As we anticipate that this workshop will create immense possibilities as well as generate opportunities in developing a global research platform towards the biotechnological engineering. We kindly hope that you will wait till the first session starts. Thank you.
Yeah, so Dr. Ghosh, maybe you can call this part of the program has been concluded, and so that people can. You are muted. You are muted. Amit, you are muted. Amit, you have to unmute yourself.
Thank you, respected sir, Professor Mukhopadhyay, for inaugurating the workshop. I'd like to thank Professor P.K. Das for gracing this occasion. And we are ending the inauguration session, and we are stepping forward for the first lecture, which will be given by Professor Yinjie Tang from Washington University, St. Louis. So he will be joining in a few minutes. We'll wait for that. Thank you all. Thank you for all the invited guests.
Thank you. You have a great day. Bye-bye.
Thank you.
Hello, Amit. Hello, Amit. Can you hear me?
Sir, you're muted. You have to unmute yourself.
Oh, I mute myself.
Good morning, Professor Tang.
Hey, good morning. Actually, it's a good evening. I don't know.
Sorry. Good evening. Good evening. Have a nice, great evening. So I'll
Okay.
just introduce and then I'll start.
Mhm.
Thank you, Professor Tang, for joining. We'll officially begin the lecture from the first talk, which Professor Tang will be giving. So we are delighted to introduce our first dignitary, Professor Yinjie Tang, from University of Washington, St. Louis. He is a professor in the School of Engineering in Energy, Environment, and Chemical Engineering at Washington University in St. Louis. He earned his PhD from he earned his PhD degree from the University of Washington in 2004. He then worked as a postdoctoral researcher at Lawrence Berkeley National Lab with Professor Jay Kisling as his advisor. He has served as the reviewer of over 30 journals in biotechnology, microbiology, and has also been in a review panel for NSF, and other funding agency. He received NSF Career Award and then in 2010 an earned Department Chair Award for Outstanding Teaching in 2013. In 2010, he earned a National Science Foundation Career Award for developing 13C metabolic flux analysis tool for cyanobacteria. His research involved two fields, including the bioremediation of toxic compound, such as heavy metals, aromatic compound, and analysis of cellular metabolic network using mass spectroscopic based tools. His work have provided important knowledge of cellular metabolic network for rational genetic engineering of microbes for biofuel production. Today, his topic is metabolic analysis to identify the bottlenecks in microbial production. He has been working with experiment and also with mathematical tools. He is expert in both areas, which is very rare in this world in metabolic engineering. So I would request Professor Tang to kindly begin this talk. Thank you, Professor Tang, for joining the meeting.
Thank you very much, Amit. So I'm very happy to give you your institute a presentation about my lab's research. And so good morning, everyone. And can you hear me well?
Yes,
sir.
Yes, sir. You are audible.
Yes, sir.
Right, great. So let me just start. So my today's talk is actually have the two parts. So one is about the non-model microbial platform. Then the second part is that what are bottlenecks.
Excuse me, Professor Tang. Excuse me. Can please switch on your camera so that we can see your face also.
You want me? Okay.
Sure. Thank you.
Sometimes actually I'm at home, so once camera on, then there will be slow down a little bit about the. Okay, so I start my camera. Can you hear me well, or this voice will be okay, right?
Yeah, yeah.
Yes,
Voice
sir.
is okay.
Okay, okay, yeah. All right. So so my talk has two parts. One is about the Microbial platform. The second is about the bottleneck for synthetic biology. So for the synthetic biology, we know that in the past 20 years, this technology achieved huge success. We can make a lot of chemicals and such as biofuel, biogas, and natural products such as terpene. And they also can make the drugs such as antibiotics, right? And they also can make the protein. So in general, this technology actually has big success in the lab. But when we try to move the synthetic biology strain to the industry, and we often face many problems, right? So the cell, one is that the productivity is very low once you scale up, right? So it is difficult to increase the production yield, and meanwhile also increase the production title and the rate. And this rate yield title, a lot of cases are trade-off, and you cannot achieve the optimization for all three variables. And during the scale up problem, there's also a lot of stress in the bioreactor. And so that's actually gives the cell a lot of metabolic response or metabolic shifting under this stress condition. And then, of course, the purification cost is another big issue. So if your production title is very low, and your cost for purification will be very high, right? And another issue is that for synthetic biology products, we always face the low profit margin issue. So a lot of cases that your production cost will be higher than your revenue, right? So that make the process difficult to be commercialized. And so if we look at the cell and we want to engineer it as a cell factory, so you have to deal with all kinds of problems such as toxicity or the metabolic burden, poor enzyme activity, low expression level, cofactor imbalance, genetic instability that is related to the scale up process. And you also have to knock out these competitive pathways, and you have to resolve some feedback inhibitions from the product. And so also a lot of cases the cell don't have the sufficient supply of the precursors. So when you design a cell factory, you have to consider all these issues, all these challenges, and you also have to balance them, right? A lot of cases that you solve one problem, you solve one bottleneck, then there will be another bottleneck appear, right? So that is the most challenging in this field. Now, if we look at the industrial success products, and a lot of industrial products actually are successful, they are using actually native species, right? They're not using synthetic biology strains, they're using the native species that already has the power to make the product, and then you train the strain, you engineer the strain, so you improve its productivity, right? So the famous hosts including Corynebacteria, Aspergillus, right? So penicillin. Pseudomonas is another very hot field. In my lab, we are recently working on the yellow wire, Lepiletica. And so basically, you can think about there's a lot of species we actually can use and as a platform for the bioproduction rather than the E. coli or Saccharomyces, right? So this non-model species actually can give you the advantages to overcome all these challenges or difficulties we mentioned in last slides. So now, if we explore new platform hosts, we have several directions, right? So we want to let the microbes to utilize the cheap feedstock, such as lignocellulose, right? So basically, that feedstock is not from the crop, right? So it's not from food. So they will be very cheap and they can be used as the carbon source for the bioproduction. Another new platform we are interested in is that they can actually use sunlight and the CO2 to make the product, right? The CO2 as a carbon substrate while the sunlight as an energy source. And this is for the feedstock, right? So on the other hand, a lot of hosts, they have the native pathways. The native pathway, they can make some product or they can make some precursors of natural product. So this type of the host we are also very interested in and hope they can be engineered and they use their native pathway or native capability to utilize cheap feedstock to make the product, right? So the product you want. So if we study the non-model organisms, we usually use the functional genomics tools such as genome sequencing and the metabolic characterization, that is system biology, right? Then you can do the genetic engineering and you make the pathway or you make the synthetic component so that the cell can actually make some product, right? You make a pathway. Then once you build this synthetic path, your next step, you have to think about the optimization, right? Pathway engineering and the optimization. So this is actually related to the protein engineering and how you make the protein more efficient and how you actually can balance the pathway, these cofactor use or precursors use. So in this case, we can see that if we want to study the non-model organisms, we have to use both system biology and synthetic biology tools, and also the traditional such as traditional tools such as enzyme engineering or the fermentation optimization are also necessary, right, to improve the host production. Now, if we look at the system biology first, so the system biology is that we can be used to understand the host, right? The host. So my research mostly focuses on this field. So in the system biology, basically, you can study the host from the genomics. So you study the host the genome, then you study the RNA, right? So transcription. Then you study the protein, enzyme. Then enzyme goes to metabolites, right? So metabolites. So at the end, we wanted to know the enzyme reaction rates. So that is called the fluxomics. And so flux is actually the in vivo enzyme reaction rates, right? So my major research are focusing on characterize the flux or enzyme reaction rates in microorganisms. So basically, we want to understand how metabolic network in a species that they can utilize the substrate and convert the substrate to the downstream products. So if we think about a cell is like a factory, right? So basically the flux analysis is a tool that understands the mass flow in the factory. So we try to understand how much flow goes in and which pathway they use and what type of product they are making, right? So this is the overall flux analysis goal. So in order to resolve the complex flux network, and we have to assume that the metabolic is under the steady state. So in the steady state, all the mass is balanced, so there is no accumulation at each metabolite. There's no dynamic change of the metabolite pool size. And so you can write down all the equation using a stoichiometric matrix, right? So basically all the equations are linear mass balance equation. And then the flux analysis actually can help you to identify the novel enzyme reactions, and also can identify the bottleneck pathway and the rate limiting steps. For example, like which pathway has the lower flux, that this flux can be a rate limiting step for producing the product you want, right? So doing the flux analysis, you can actually understand the metabolic network. Then you can actually try to design your strain, try to design your engineering approach to improve the strain performance. Now, if we look at the system biology, and they actually have to work with strain engineering or synthetic biology closely in order to improve the strain production. So this is famous DBTL cycle. This cycle is often used by engineer to improve the host bioproduction capability. So the D means the design, right? So you can use a genome scale model, you can use a flux model to design an optimal flux network for the strain to make the product. Then you can start to build your network. You can use pathway engineering, enzyme engineering, and to redirect the flux towards the biosynthesis, right? Then after you engineer your strain, then you need to do the test. And this is more like the system biology type of the test. You have to understand their production, understand the enzyme expression, understand the transcription level. So based on this test, you will learn what is the bottleneck, right? What is the next step you can improve your production? So during this process, or one process, the flux analysis actually give you the most direct readout or most direct information about your metabolic pathway, and that can help you to understand that what you should do that for the next round of the design approach, and how you can actually improve the production based on the bottleneck you identify from the test and the run, right? So this type of the approach and the design, build, test, run cycle had to be run iteratively and many rounds in order to achieve the final success to make industrial viable strain, and that can finally have the impact on the human society, right? So this process is painful, and it takes a lot of time. Now, let's look at the non-model species. We, in my lab, I have actually, I worked on this species before. So compared to E. coli, so when I first joined Jay Kissing's lab, and we work on the E. coli a lot, and so most the bioproduction, they're focusing on actually use E. coli. And however, we know that E. coli have a lot of issues. And so then when I start my career 12 years ago at Washington University, so I work with Yinjie Pakwasi, and so he's an expert on the cyanobacteria. So my interest actually shift to the cyanobacteria. So the cyanobacteria is a very good species. They can use sunlight, and they can use CO2 and to make the product, right? So then fortunately, a few years ago, I got some funding to study the lignocellulose degradation. And so how to degrade the lignocellulose, there's a bacteria called the Rhodococcus, right? So they can be useful to actually degrade some lignin byproduct. And then this Rhodococcus can be used as a platform to convert the lignin to the useful product. Also, very recently, I had funding from USDA. So the Department of the Agriculture, that actually supported me to study the yellow wire yeast. And this yeast actually can be used to make the terpenoid product. So finally, the colibacteria, actually, we had some collaboration at the JBay, Joint Bioenergy Institute. My collaborator, Angela, so she's actually working on the colibacteria. Actually, we wrote several proposals, and we actually initiating some new collaboration to study the colibacteria. And this species is actually a very good host for amino acid synthesis. And the amino acids can be actually a building block for many other bioproduction, right? So now, actually, if we use these species, and you can think about that, these species actually, they have the native pathway. And so basically, their native pathway to make product or utilize the cheap feedstock. So you don't need to engineer this pathway like extensively, because they already have the native pathway. And so what you need to do is that you need to understand the pathway, then you add the established pathway, and so you add several enzymes, and so hope that they can actually make the a product and in like the cheap and high efficient way, right? So the first species I worked on, non-model species I worked on at WashU is cyanobacteria. This species actually, there's several cyanobacteria species. So the most famous is cyanobacteria Synechocystis, the PCC 6803. And this species actually was discovered like 40 years ago, and they actually can be engineered quite easily, and they can make the all kind of the products. The problem is that this species grow very slow, so it's a green line you can see. So later on, the researchers in United States, they actually focus on other cyanobacteria, including the Synechococcus, the strain name 7942, and then 7002. And then later on, I work with Himachal Pradesh on newly discovered species called the UTEX 2973. And this species is Synechococcus UTEX 2973, has a doubling time of 2 hours. So if you grow them in a very optimized conditions that you provide enough light and CO2, this species can grow as fast as a yeast, right? Doubling time of the 2 hours. And so this species, actually, if you look at the TM picture, they have a very thick, like a photosystem protein. So you can see the outside of the cell or wall, they actually have the covered with the photos, the system 1 and the photosystem 2 protein, and they also had the antenna system. So they can harvest the light very efficiently. And compared to the slow growing Synechocystis 7942, you can see that the major difference is that the fast growing cell have more protein and more enzyme built inside of a cell to harvest the light. So they can grow very fast. Now, all these species actually can be engineered, and we put a pathway and make them to make the product. So actually, my former student, Arzul Varman, and he made some success for engineering cyanobacteria to make lactate, right? D-lactate, so you put some lactate dehydrogenase enzyme, and so basically the cyanobacteria can convert the CO2 to the lactate, and which is a very important polymer precursor, right? And also you can make the isobutanol, right? Isobutanol is the a biofuel, right, from the cyanobacteria. And so basically this type of the example showed that the cyanobacteria is an excellent platform that can convert the light energy and the CO2 to the final product, right? So the product like you want to make, such as a biofuel or polymer or other chemicals. Okay, so the second species I want to discuss is Rhodococcus, right? So the Rhodococcus is a species that that can degrade the glucose, cellulose, or phenolic compounds. So this species actually is very unique, and so you can see they have a lot of tag. So there are a lot of lipid inside of the cell once they grow in the nitrogen limited condition. And so they can actually tolerant to the solvents, right? So they can accumulate like 70 to 80% of the tag. And so they can convert the lignin byproducts to the biodiesel. In our study, right? So the lignin, we get the lignin, then you can use the thermochemical method to convert the lignin to the bio-oil, right? So this bio-oil is most like the phenolic compounds. And these phenolic compounds can feed into the cell. The cell actually eat these phenolic compounds and make the biodiesel. And this species actually is very interesting. And so basically they use a very unique pathway called the ED pathway, right? So basically for this species, because they use the ED pathway, so you can see that they actually can avoid the catabolic repression. So what is the ED pathway? So the ED pathway is a shortcut pathway. They can directly convert the G6P to pyruvate and GAP, right? So through the ED pathway, they don't use the EMP pathway. And this pathway can generate both NADH and NADPH. So because of using this pathway, the cell actually avoid the use of the pentose phosphate pathway, which is the CO2 generation pathway, right? So in this case, the cell have the higher carbon yield for making the product. And also the ED pathway can avoid the catabolic repression. So this species have a very unique capability, metabolic features. So they can use phenol, right? The phenol can come from the lignin, right? So lignin, you treat them, use the thermochemical method, you will get a phenol. And so if you feed the cell with the phenol will be degraded and utilized, and those phenol will goes into the acetyl-CoA and the succinate-CoA pool. Then they will go digest by the TCA cycle, right? Then so the carbon from the phenol can first go up to the PEP, and they actually can goes to the G6P through the reverse EMP pathway, or we call the glucogenesis. When the phenol and the glucose both present, right? So we know that for E. coli, if you have glucose and then you have, say, xylose, the E. coli will eat one substrate first, right? So say they eat the glucose first, then they eat the xylose. While for the rhodococcus, they don't have the catabolic repression. So even you have the glucose as a favorable substrate, they still use a phenol. And so basically the pathway for phenol utilization, they are actually using the TC cycle, and the radius goes to the top so they can synthesize the sugar phosphate, while meanwhile the glucose can also be consumed via the ED pathway. So basically these two pathways became decoupled, right? So they use glucose goes to ED pathway, then this carbon goes to the other product. So because of this, and we found that the glucose and the phenol can be co-utilized as the favorable substrate to promote the lipid production or TAC production. So this is the rhodococcus, right? So the third species in my lab we work on is Yersinia lipolytica. And for this species, we actually work with a company, and also we work with a university in Hawaii. And so basically this species are very good hosts to make the natural product. So they can convert the glucose to the mammalian pathway. So this species have very strong mammalian pathway. And this mammalian pathway can be engineered by introducing some enzymes. So this enzyme is from the plant enzyme, such as P450. And then that can be produce lycopene or other terpene product. So if we look at this pathway, basically in our lab and our collaborators, we engineer this species through a process called the power, push, block, and pull. So the power means that we need the cell to have more power, right? So we can get the cell like oxygen, more oxygen, we can feed the formate so the cell can actually generate more NADH. We can overexpress the VHB protein. The VHB protein actually can improve the cell respiration. Then we also try to push the pathway, mammalian pathway, to make more precursors for the biosynthesis. So basically you can overexpress the acetyl-CoA synthase, or you can overexpress the ACL enzymes, so acetyl lysis, and also the ALD enzyme. So basically you push the acetyl-CoA pool, right, towards the GGPP. And so we also block the competing pathways such as scoring pathway. We knock down this pathway, and so that more fluxes goes to the final product. And in the last step is called the pull. The pull means that we improve final enzyme activities such as a lycopene synthesis enzyme, right? So and the P450 enzyme. So that is we target on the last one or two enzymes. So basically, this is the enzyme from plants, and so this enzyme can be optimized and engineered so that they can make more like the product. So the overall engineering idea is that we have to give the cell enough the energy molecules, then we make this pathway too strong, and we balance the pathway, and we make the product. And so this is one example we achieve the production of the alpha inon, right? So, and if we see the fermentation process, we're making this product is actually can be used as the perfumes. They smell very good. So they usually come from the flower, right? So, but we can actually use yeast to make this type of the perfume product. And so during this fermentation process, actually we can see that the isoCoA can goes to the malonate, malonate can goes to the final product, the alpha inol. So the engineering step, actually, if you look at the red step, so that is we put the enzyme to enhance this pathway. So you can see that they have the long step, the enzyme overexpression, and then we finally get the alpha inol. So the overall engineering is quite a challenge because the pathway is very long, right? So, and we can achieve like about 400 milligram per liter production. And so for the yeast, and this yeast, the Yawa yeast, is very interesting. So during the fermentation, we also see that at the beginning, right, when the cell grow, so they actually they form like some normal large shape like cells look like this type of shape. But when the cells go to the late phase, and they will form the filamentous cell, right? The cell will become very, very long. And so like this cell, so like this cell. So we're still not fully understand like why that the cell actually started to have the morphology change during the fermentation. Very likely that during the late fermentation process, maybe the cell have a lot of stress, then they trigger the cell to have the morphology change, right? So that is the third example about the host we have worked on before, and that is Yalowaya yeast for natural product synthesis. Actually, a couple of weeks ago, and Amit wants me to discuss some like machine learning topic. So that is a very hot topic now in United States. And so I did some machine learning work, but it is very superficial. So I have a project about 6-7 years ago. I worked with two professors from computer science. So this is an NSF project. And so we try to use machine learning to guide our bioproduction. So the idea is like this. So every year, there's a lot of papers published, right? So you can get to the PubMed. There's tons of the paper, and you can actually try to read this paper, and you extract the information from the paper, and which called data mining, right? So then you have this paper, and you can organize this paper and organize what they did. And so the data organization, and you can categorize this data, say that what type of enzyme they are expressed, what type of flux it will be. So you get a lot of information. And this information you can be used as a database for the machine learning. So we tried this approach. So basically we collected the synthetic biology papers, and then we converted them into the structured data, say that the structured data include like the genetics, species name, reactor type, nutrients conditioning. And so we will manually assign the value to each parameter, right? So because different papers have the different condition, so we have to use a categorical number to describe each different condition. So finally, we will build a big Excel sheets and including all the information. Now then, we will search the features, like what is the input. We need to find what is the input and what is the output. So the input will be some like the features such as the environmental condition, genetic factors, and the output will be the fluxes and or the production yield or the fermentation performance. So after we just define the input and output, so we can actually try to build a model such as a neural network. And so we can use the input and then to predict what is output will be, right? So that is some machine learning work we did before, and we still have some type of the work going on for using the machine learning to predict the bioproduction or predict the strategies for bioproduction. So yeah, so if we had more resource, and now the funding is relatively hard, if we have more resource, I'm very interested in continuing this type of research, right? Okay, so the last part I want to focus on what are load blocks to synthetic biology. So this is what my own opinion, right? So what I found in the past research. So I work with several research groups, and so we try to work on some pathways. We try to commercialize some bioproduction, but we always have some problems. So that is also the common case for the biotech or synthetic biology company, right? So if you look at their stock, like Gevo, they are making the biofuel, Emirates making the natural product, and also biofuel. And so all these synthetic biology companies, actually they are suffering about the making the profit or making the a success to move the laboratory strain to the industry application. So how to solve this problem? Why there's the load blocks, right? So I want to discuss like two load blocks that we have noticed, right? So one thing is that this is like energy or ADP balance calculation slide. So that is from my bioengineering class that J.K. saying when he teaches this bioengineering, he uses this slide to show students how to calculate the ATP balance. This is a very old slide, right? This is probably 20 years ago, 30 years ago. And so, and the people in a long time ago, they noticed that if you have sugar and you want to make the cell to survive, so the cell need ATP, right? So ATP is keeping the cell alive, and the cell need ATP to synthesize and to survive under stress. So this ATP production is very costly. So you can think about like the ATP is from the NADH, and some is from the FADH. Then the NADH have to be oxidized and to make the ATP. So basically, if you do an energy balance, right, so you will immediately see that if the glucose have the energy, which is about minus 686 kilocal per mole, this is glucose energy, or 1 mole, right? So once they oxidize, they get the ATP. This ATP that the cell can use. So we assume that 38 ATP, right? We can get one glucose, we get 38 ATP. And then what we will actually lost about 60% of our energy. So 60% of energy are lost as a heat, right? So during the metabolism, the overall thermodynamics are not efficient. So you will actually lose 60% of the energy as a heat. You only can have 40% you can be used as the biological energy, right? So that is already reached the theoretical maximum. So if you, based on this slide, you will immediately see that if you want to use a biological way to make some energy intense city high energy molecule, that would be very difficult because your consumption of the ATP and your efficiency will not be high, right? So that is for sure that, but because the nature of the reaction, metabolic reaction, so you will lose majority of the energy as a heat, not your product. So and then this problem will lead to another issue is called the metabolic burden, right? Metabolic burden. So what is the metabolic burden? So when we look at a pathway, and we look at the host. You want to engineer this host and to make the product. And then, so basically your general idea is we say that we should pull the pathway for the product, and we knock out the competing pathway, and then we push the flux from the substrate to the product, right? So that is pretty obvious genetic target that you want to work on to improve the production. However, a lot of cases, the effectiveness of your strategy are limited by a hidden force, which is energy metabolism, because every pathway or every pathway expression, right? So, or you knock out the pathway, or you want the pathway function of bioproduction, they are constrained by the energy metabolism. That is mean that how much ATP, right? So ATP or NADPH you have to power the energy metabolism. Now, we know that ATP production is very costly. So that gives you the challenge that if the whole pathway require a lot of ATP, for example, you need ATP to overexpress the enzyme, right? So then the cell may face like the shortage of the ATP, right? So just like this type of the system, so we always think about synthetic biology is like electronic device, and we hope like the cell can carry all kind of the synthetic parts to do your work. However, for the electronic device, the battery technology in the past 50 years or past 100 years, they improved so much, right? The battery getting smaller and the battery getting even more higher energy density, right? For the microbial system, the energy is coming from the ATP synthesis, and this one never can be improved, right? The ATP synthesis is actually the energy producing enzyme that is evolved through years, and you never can be upgraded like the battery system. So that is actually explain that why the synthetic biology meets so much trouble to be successful, right, in industry, while the electronic device or like the cell phone, you can actually be very successful, and you can be every year you will see some like new product come out, right? So in this case, you can think about like the cell factory, and the major limitation is more like so, your power plant, right? So you don't have enough power plant, and the power energy limitation will limit the manufacturer capability. So if we look at the cell, and a lot of cases the cell, you during the cell growth, and so the ADP consumption is high. They needed the synthesis of the plasmid and enzyme, cell repair, stress tolerance, energy sparing, and the filter cycle or chemical gradient preservation, proofreading, or cell motility. They all needed the ATP, right? So on the other hand is that during the respiration, right? So a lot of cases, the NADH conversion to ATP is not fully efficient. So a lot of cases, the proton leakage will cause the ATP production less than the theoretical value. If you oxidize one more NADH, you often get less than 3 ATP rather than theoretical value is 3, right? So actually, a lot of cases people found that you only get 2 ATP rather than 3. So in this case, you can think about the cell have to consume more NADH, the cell have to lose a lot of ATP to maintain the survive, right? So therefore, if you think about the cell is going to make the production, and so this figures to give you some kind of general simulation, say that I simulate a yield, fatty acid yield, right? The fatty acid is a product yield as a function of the ATP consumption, right? ATP consumption for partial reconstruction and the oxygen flux. So we can see that the ATP consumption is more like a burden, right? So metabolic burden. So if the ATP consumption increase, then what happens is the cell at the beginning, the cell have some energy, redundant energy. So you will not affect the production, right? So, but if you engineer pathway and you add more component and you make the cell to use more ATP, so what happens is that the yield will be dropped quickly, right? So at this cliff, you will see that the cell will become not very stable. So even tiny amount of the oxygen change here, so the production loss can be high, right? So this simulation is based on the flux balance analysis. So this message tells you that if you want to make some product like the fatty acids, so that requires a lot of ATP. And if your cell, meanwhile, you also consume a lot of ATP for partial reconstruction and maintenance. And while if the oxygen supply is limited, like in the bioreactor system, then you will face a problem that your productivity is on the metabolic cliff. So this area, right? This area means that your production is not stable. So your repeatability, your results are highly sensitive. Your performance, your strain performance, are highly sensitive to the oxygen condition, and also the highly sensitive to the ATP consumption of the cell ATP loss, right, during this maintenance process. For example, if the cell had more stress, they consume more ATP, then the production will have the quick loss. So in order to overcome this type of problem, and we did this study before. So for example, the E. coli, we engineer a species to make the fatty acid, and we found that actually energy metabolism is limited. So to overcome this problem, so we actually want the cell to make more ATP rather than actually making more carbon for the production. So in this case, we actually engineer an enzyme called the VHB. So VHB enzyme is actually try to promote the cell respiration. So they make the more ATP for cell, and then you can make the higher production, improve the fatty acid production yield, right? So this strategy is that if you think about, if you want the cell oxidize more carbon, you probably think about that it's not good, right? Because more carbon will be lost, and so you will have the less yield to the fatty acid. But if the energy is the rate limiting step, and we found actually you make the cell lose more carbon, actually can improve the production, because that when the ATP is limited, the cell cannot achieve the higher yield, right? So the ATP block the cell the production. So if we let the more glucose be oxidized and overcome the energy bottleneck, then the production will be increased. So then the second load block we had is related to the enzyme channeling. So the substrate channeling is phenomena is that a lot of cases enzyme can be naturally linked together, and this enzyme interaction and so allows the intermediates between enzymes. Actually, they can have the reaction without release to the book solution, and then they can increase reaction efficiency, right? So the substrate channeling, usually you can through the membrane or covalent bond or within the large enzyme complex. So basically, the enzyme can form some like a tunnel or scaffold, and so then they can actually direct the flux. And however, we found actually, also other people found that the proximate channeling via weak force through long step cascade enzymes in central pathway is also be possible. And so basically, in native pathway, the enzyme may actually link them together, like they will naturally recognize each other and to stick each other to direct the flux. So my colleague a long time ago, he found that for EMP pathway, so if you have a pathway that they can degrade the glucose, right? So basically, he found that if you put the F6P as a second substrate, and these two pathways will goes to the two different routes. So this one is channel. This one is unchanneled, right? So basically, they found that actually EMP pathway have the two routes of the flux direction. So if we think about this enzyme system, right? So in say the in E. coli, and so basically they may not be randomly distributed. They actually can form some kind of subtle metabolome, and this metabolome can actually direct the flux toward the final product. And this type of metabolome may be widely distributed in the cell and direct the flux, right? So, however, if you engineer your pathway and you may not realize that this channel is there, and it's difficult to replicate the native pathway, how to actually coordinate cascade enzymes, long step cascade enzymes in a very well-controlled metabolome structure, right? So if this channeling is true, then we will see that we will rethink about our kinetics, right? The concentration, local concentration will be changed, and we will have the Gibbs free energy, we will have to be revisit, and we have to revisit the model. Flux labeling assays will be changed. The catabolic repression mechanism will be changed. And also they will affect our synthetic biology design, right? So the long story short, since I don't have enough time, I just want to give you some engineering approach. We worked on two species. So one is on E. coli, one is on the cyanobacteria. So here, so the message is that if we knock out the channel, the first example, if we knock out the channel is that we found that the heterologous ED pathway will become more effective here. So if we keep the channel, so we don't knock out the channels, that is a PTS. And so basically your engineering of the heterologous enzyme will be a little difficult because the pathway is very tightly controlled by the channel, right? So the second example is for cyanobacteria study. So our recent study with Jeff Cameron, we found that if we knock out carbosome, and the carbosome we believe that is that form the Calvin cycle pathway channel. And so by doing this one, we found that the flux do not have much change, but the interestingly is that the precursor metabolites such as pyruvate and UDPG, they have the significant change. So in this case, that the cell is more easily to leak the metabolites from the Calvin cycle to downstream product for the production, right? So that Brian Feiger, he used this species actually to produce lactate more effectively, and because once the channel is broken and your heterologous pathway is more easier to hijack the flux from its native pathway, right? Okay, so today I just stop here. So basically, the summary is that the Mother Nature actually makes us in cell biology difficult to be applied. Understanding and the employment of non-model microbial metabolism are a promising direction. A lot of cases in my study, we use metabolic flags a lot, and they give us a lot of guidelines for synthetic biology applications. And so, yeah, a new field is knowledge engineering, and machine learning can play an important role in synthetic biology study. Okay, so this is all my talk. And do you have any questions?
Thank you, Professor Tang. Such a wonderful talk. I really enjoyed the talk. Professor Tang has covered metabolic engineering in cyanobacteria, and then E. coli, and then yeast, and then especially the rhodococcus species, which he considered for co-utilization of lignin and glucose, which is very wonderful, and we really enjoy his talk. Thank you, Professor Tang, for giving such a interesting talk, which will motivate more students to work on metabolic engineering. So I will we have few questions to ask Professor Tang. The first question is, when you do the push and block strategy for Yara Lipolytica, which include format, is it audible?
Let's see. So the question is about the format utilization.
Yeah, format glucose as a substrate. The audience want to know how to remove the carbon catabolic repression when two substrates are added. So, is there any repression, or can utilize together?
Okay, so yeah, this is a very good question. So the question is that if glucose and the format are co-feeding, co-utilization, then whether that actually the cell will just use glucose and they don't use format. So the format is a kind of idea that recently got very interested by DOE ARPA-E program. So the format can come from the cheap source, from the catalytic process, so you can get the CO2 and the sunlight to make the format directly using electricity, right? And so then the format can be used. Actually, the people found that if you feed the format into the yeast or some certain type of yeast, and the format will be oxidized. Yeast thinks that this is toxic chemicals, so they will not uptake the format. They just oxidize it. And meanwhile, during the oxidization, they will generate NADH, and that is a reducing power for the cell. So actually, recently there's a lot of study on the format, how to actually extract this NADH from format to promote the fermentation, which called the CO2 zero emission fermentation. So basically, you don't need the cell to run the TCA cycle, but the format will be the power source. So that is one direction. Currently, a lot of research here are interested in.
Okay, thank you. So in this, yeah, Arul.
Hi, Professor Tang. How are you?
Good. Hey, Arul.
I got a question while we are on this slide. Okay, so you have 3 pathways for the conversion of, or for the production of acetyl-CoA here, right? You are engineering 3 different routes. So which of these routes you found to be very critical to that led to acetyl-CoA accommodation?
Yeah, ACS, acetyl-CoA synthetase. This one is most factor. ACL overexpression not helpful. Actually, the Peng Xu from the Greg Stepanowicz lab, they have a paper and they studied which pathway, which enzyme improve the acetyl-CoA pool size. So the acetate, that's a CoA synthetase. So ACS is actually the most key enzyme. So ACL, the native enzyme, is already very strong. So basically, you overexpress this one, it not help too much.
Okay, Thank you.
Professor Tang, I have one question. Like in Rhodococcus species, you have used two carbon sources like sugar and that is glucose and phenol, right? So, is there any catabolic repression in this organism as well, or how do
Oh
you?
no, there's no catabolic repression. Yeah, so that is a nice thing for this species. So if you use glucose and the phenol, they can use simultaneously. So because they're using two different pathways, right? The phenol mainly are digested by the TC cycle, while the glucose can still working on the glycolysis, and also because the glucose utilization is using ED pathway, while the phenol utilization that can go to the glucogenesis. So basically, our labeling we found that the phenylcarbon goes to sugar phosphate pathway. So while the glucose carbon also can go to the TCA cycle, so they can be actually used simultaneously. Yeah.
Very nice. So one more question we have from the audience. So, is there any advantage of using variability for terpenoid synthesis over Saccharomyces cerevisiae, or how do you compare?
Oh yes, this is an excellent question. So I think there's the number one is that the yellow wire don't make the ethanol, right? So basically, for the bigger yeast, there are a lot of carbon lost as ethanol. So if you knock out the ethanol pathway, the cell is not happy. The second thing is the most important for yellow wire is that they have very strong native membrane pathway. So this pathway is very strong. They have the ACL enzyme, so the citrate lysis enzyme is very strong. So basically, they can make a lot of acetyl-CoA. So this is why the yellow wire can actually accumulate a lot of lipid inside of a cell. So because their acetyl-CoA synthesis is very powerful. So then once the cell are getting fat, so they form the lipid body, right? So the lipid body. The lipid body is like, because the cell is very fat, so lipid body can actually dissolve some natural products such as the, so say if you are making the lycopene or beta carotene, so the product can be inside of the lipid body. So in this case, that the toxicity issue can be resolved because once the cell uses lipid to dissolve this natural product inside of the body, and they will not be toxic to the cell. So basically, they have a lot of advantages over the baker yeast.
Yeah. One more question from the audience. Although you have explained already, but you can say a little bit about the critical factor for pushing the flux toward a compound of interest for different organisms you have tried for metabolic engineering. Overall,
I cannot hear your voice well. Sound is like not disconnected. Can you repeat your question? Yeah.
I repeat the question. So, what are the critical point for pushing the flux toward desired compound? Like you have explained pull and push mechanism, but what are the steps one should follow? Suppose you want to somebody is starting metabolic engineering. What are the steps he should follow to get the maximum pull and push mechanism happening in the microorganism, any microorganism?
I still don't catch your question. Can you repeat again? I don't catch.
Can I try to repeat?
Yes.
Okay. Okay. So Professor Tang, they want to know what is the main bottleneck one needs to address to increase flux towards a product?
One bottleneck to increase flux to the product.
Yeah. So if you want to produce a product, what are the steps you want to take to increase the flux towards a product?
So in this figure, we show that you have 4 methods, right? So the push is that you overexpress the pathway to increase the precursors. Block, you actually kill this competing pathway. And the pull, you engineer the last enzymes, right, for the product. And then you also have to balance the energy, right? The power, right? It's very important, NADH and NADPH. And these four things you have to work simultaneously. And in a lot of cases, there can be a trade-off. So you also have to balance this. So that is a very difficult process, right? So you have to do the design, build, test, learn cycle to understand the effectiveness of each strategy, how to improve the production.
Thank you, Professor.
Hi.
Okay.
If there's time, can I ask a question, Amit?
Yes, yes.
Hi, so I'm Anu Raghunathan. I have a question related to your machine learning models. So I was wondering, there's a lot of incomplete data, right, that exists in literature about the fermentation, yields, titers, etc. So how do you actually overcome this limitation how do you curate the data? How do you overcome that limitation when you develop your models? Because that would be very critical to the success of the learning curve and the training set, etc.
This is an excellent question. So currently we get the paper, we manually read the paper. Actually, Alu published a paper using the machine learning method how many years ago? 9 years ago, 8 years ago, right? So on the Microbial cell factory, what he did is it's on yeast, right? So he read like 100 paper maybe, and each paper you get a fermentation product and what type of genetics they use, species, and you make the category. And then once you have this data, then it's doing the machine learning is easy. So if we say that we manually extract data, say probably take the 99% of time, and only 1% time you build a model, because the model you can very easily to apply, right? So how you build this database, you need a lot of human curation of the data. So I agree that sometimes the paper is difficult to understand, right? So sometimes the paper don't have a high quality. That is, I think, the major challenge for machine learning. So if we can have more standardized database, that will make the machine learning more powerful and more like precise. Right? Did I answer your question? Alu, you want to add more?
No, I think you did. But what I'm really concerned about is that, there are very robust methods to curate metabolic networks to make sure that the stoichiometry is correct. There are no inconsistencies. But when you look at a model to make predictions of yields and titers and then fluxes through that pathway, then there's a lot of incomplete information in papers. A lot of times they do not provide information about what were the conditions of say the fermentation, what were the substrates, what were the extra nutrients, etc. And those are things you cannot extrapolate, as opposed to a network, which is more, it's static in some sense. It's defined by the genome architecture. So I'm just curious, how does one go about it? We can take this offline as well, but I was just curious, how did you manage to get around all that incomplete information, missing information in papers?
Exactly. So you answered a very, very good question. Yes, we haven't resolved this problem well. We agree. We hope like when we have more and more data, more and more paper, adding to this data, even the data is not accurate, right? As long as the data getting big enough, you still will have the decent prediction power. Now, here's the thing. What we talk about machine learning is a little different probably from the machine learning you learned from other system biology or metabolic modeling machine learning. Our thinking is like here, right? So if you have the flux balance model, you can predict the optimal condition, right? And you can predict the yield. But for the titer, it's difficult to predict because first is that you can use different media, right? So a lot of cases of fermentation using a rich media, they add a lot of yeast extract and they add all kind of other things, right? And also the racked operation, the fed batch versus the batch culture, they will have a very different title, right? And also you may have the genetic part, right? So say that how you overexpress the enzyme and what type of level you add in. So this may also affect the yield. So we want to actually understand like all these kind of features or variables, how whether we can use these variables to predict the title. And our model actually, our recent paper, our model is on the top of the regular metabolic flux variance analysis. So we have a metabolic flux analysis model, and on top of this model, we use machine learning to fine tune or to tune the model prediction, right? So FBA model often predict optimal condition. Now we will see that if the condition change, what is a real prediction, right? So we're still working on it. Yeah, as you mentioned, there's a lot of challenge, especially for the data quality. We are struggling on this part. Yeah.
Yeah, especially for less studied organisms, I think it would be really difficult. Anyway, thank you so much
Yeah.
for
Thank you for your question. Yeah.
Professor Tang, I have one question.
Yes, you can continue.
Yeah, I'm actually from the Department of Electrical Engineering, but I have just specific interest about this. What type of machine learning techniques do you use?
Actually, we tried several times. The neural network we use a lot, and the decision tree, and there's a support vector machine. So that's the three common methods we use.
Okay. Now, this quadratic programming you have mentioned, where does it come into the picture?
Okay, so that we don't have it. So basically, say this part we will add the at the end. So this is not in the picture. So basically, sometimes the machine learning we did a prediction. They don't make sense, right? So the result actually is out of like story geometry, and the values can be so different. So that we have to use some, say, the flux balance analysis to give the constraint for the machine learning model prediction. Otherwise, sometimes they just give very ridiculous predictions. So we have to use some mechanics-based model to change the solution space.
Okay, thank you.
Thank you.
Thank you, Professor Tang. Such a wonderful talk. We really enjoy your talk. It is already midnight there. It's 11:15 p.m. Thank you for taking the time out and sharing your research work. We really appreciate your time and work, and we'll catch you later. Thank you.
Yeah, and thank you very much. And I think my student Alicia will give a talk tomorrow morning, right?
Today evening in Indian time.
Yeah. I lost contact for her. Actually, I have a little worry about it. Actually, she got the COVID recently, so actually, I don't know what's going on. I tried to contact her about her presentation, and she don't reply to me. So I emailed her several times, and I want to see her slides, but I don't get her back. So actually, I don't know whether she will show up tomorrow, but if she don't show up tomorrow, then please just be prepared. Right. So I got some the information from the schools. She actually diagnosed positive COVID recently. So that's very informative. Yeah.
Okay. Thank you, Professor Tang. We'll give.
Thank you very much, and I hope, can see you in the future in India or in US.
Yeah, we will have the in-person workshop next summer. So hopefully things will be improving. Then we can meet in person. Thank you so much.
Yeah, bye.
Good night. Good night.
Good night. Bye.
we can move on to the next talk, right, Amit?
Yeah, you can carry on.
Oh, okay, all right. Good morning, everyone. My name is Arzul Varman. So I collaborate with Amit on this Spark project. I'm from Arizona State University. And I hope you all enjoyed Professor Tang's talk. This next talk, it is going to be an introductory talk, introduction to flux balance analysis and labeling approaches in metabolic engineering. Okay? And as the title says, this is in fact an introductory talk. So when Amit approached me and we were discussing about the various talks, we thought it would be good to have one talk that gives a theoretical background for beginners or even for experimentalists, so on flux balance analysis and modeling approaches, people can learn and collaborate better. In fact, as this is an introductory talk, it would have been appropriate that it is at the beginning of the workshop. On the other hand, not many of you would be aware that Professor Tang, he mentioned, but he's actually my PhD advisor as well. So as the Indian culture, the student follows the guru, right? So in that manner, I think this is a good way we have scheduled it. And in fact, it will be my student who would be actually giving this talk. So it will be more in type of lecture mode, lecture type of mode. So students, if you have any questions, you could interfere and ask him. I think we lost some bit of time, but Apur, probably you could skip one of the examples, we catch back on the time. And Apur, he's my second year or third year PhD student. He's been doing very good in his research as well. So in my talk tomorrow, as per US time, but as per the Indian time today night, when I talk about my presentation, I would also be talking some of Purv's research work. So he has been doing excellent on his research aspect. Plus, we thought it will be a very wonderful opportunity for him to hone his teaching skills. So I have given this platform for Apurv to present on introduction to flux analysis. Apurv, do you want to share your slides? The stage is yours.
Yeah, thank you, Dr. Gordon. All right, so am I audible?
Yes, very well.
Yes, yes.
Okay. So very good morning to everyone. I'm Apurv from Arizona State University. And today I will give you introduction to some of those interesting techniques which Professor Tang talked about, mainly metabolic flux analysis and flux balance analysis. So to start with, cell as a microreactor. If we consider a bacterial cell where numerous reactions happen inside it, looking at this figure, it's a very complicated map which keep on happening inside a bacterial cell. If we just take a small part of it, and if we are able to play with these pathways, we can be able to make some useful biochemicals or biofuels out of it. So there are different techniques which can be used, as Professor Tang talked about, like power, push, block mechanisms, knockouts, etc. So to do something like this, you need to know metabolic pathway in a better way. You need to know the directions. Metabolic pathways can be confusing, just like this moving stairs in Harry Potter. They can be changed to different directions for your own interest. So how do you do it? So there are different techniques you can use for doing it, including genomics, transcriptomics, understanding proteins, metabolites. One of them is fluxomics. So understanding metabolic flux inside a pathway is extremely helpful to know what's going on and to also use it to your advantage. So flux modeling had started with elemental balance modeling and stoichiometry model, and they were some initial steps in developing models to understand flux in a pathway. And they were stepping stones to these new techniques which include flux balance analysis and metabolic flux analysis. So today I will mainly talk about flux balance analysis and metabolic flux analysis. So this would be outline of my talk. So I'll begin with flux balance analysis. I'll give short introduction to what 13C fingerprinting is, and then we would use the knowledge of 13C fingerprinting to improve flux balance analysis and to use something called metabolic flux analysis. Okay, so flux balance analysis. So in simple stoichiometry models, there can be mainly three different systems: determined, overdetermined, and underdetermined. If the number of reactions is equal to number of metabolites, then it's a determined system. If you have more information, if you have less number of reactions and more number of metabolites, it's overdetermined system and it's easy to find unknowns. But if there is a system where you have more number of reactions and less information about metabolites, it's an underdetermined system. And the system which we study using flux balance analysis is an underdetermined system. So how to solve this issue where you have more number of unknowns to find? So the first step is you assume a steady state assumption, where you assume that the rate of change of concentration of metabolites with respect to time is 0. So this helps you in developing this equation, stoichiometry into flux equal to 0. So this is the first assumption we make in flux balance analysis. So yeah, so this is like, I threw a lot of words on you all, a lot of data here. So let's try to take an example and try to make it simple. So this is simple mixed acid fermentation pathway. And the first step we would do in flux balance analysis is we would assign numbers to reactions. So we would call it step 1, define metabolic network. So when we define metabolic network, we have given numbers from 1 to 8. And you would notice that glucose to PEP is one step, is given one number, but when PEP goes to succinate, there are multiple steps involved, but we have considered it as one number 2. So I would discuss about it further in my presentation. But at the moment now, we have given numbers to these different reactions. In step 2, we build model equations. So for example, how did we develop these model equations? Let's consider relative rate of phosphoenol pyruvate. So in phosphoenol pyruvate, glucose is going in, and pyruvate and oxaloacetate are two reactions going out of PEP. So the reaction which goes into PEP would be positive, and those leaving it would be negative. So V1, flux 1, which is V1, would be positive, and V3 and V2 would be negative. So V1 minus V2 minus V3. So this is the way we have made equations for all of the rest of them. So then we would make a stoichiometry matrix, which you could see here. I hope you guys can see the pointer, right?
Yes, we can see the pointer.
Thank you. So in this stoichiometry matrix, we have information about V1, V2, V3 in the first row. So hence we assign variables V1, V2, V3 in the first 3 columns, and rest of the columns are left 0. So this is the way we make a stoichiometry matrix. The next step is now over here, if you see, we have 5 equations, 1, 2, 3, 4, 5, but we have 8 unknowns, which are V1 to V8. So we cannot solve this, especially when we have more number of unknowns and less equations. So the next step is we would measure the extracellular flux. So we would try to use information which we get with the help of wet lab experiments. First assumption would be, let's assume 100 moles of glucose is consumed. And then when we study the products formed in that pathway using techniques like HPLC, mainly acetate, lactate, we could distribute them based on 100 moles of glucose being added. So now we have more information. And in this way, we make our system determined. So when initially, when I said we have 5 equations and 8 unknowns, it was not a determined system because we had more unknowns, but now using the information, we are able to develop a determined system. And now we could solve these equations for finding V1, V2, V3, V4 till V8. And hence we have flux of these different variables in that simple metabolic pathway. So this is the simplest example where we could use a pen and paper and solve and find fluxes of this simple metabolic pathway I talked initially. So we had one more example, which I would skip. So, okay, but what if we are trying to study a more complex system, like a genome of a bacteria where there are numerous reactions happening at the same time? So the first thing, as we did previously, we tried to assign numbers to the network, right? If you look at this complex pathway, things get more difficult. So you normally would start with the databases available online, like KEGG, to get more information about the active pathways in that organism. If it's E. coli, we would have more specific databases available. And then you could select the pathways of your interest. And that's the way you would start. Now, how would you select the pathways of your interest? The most important thing here would be, if you select incorrect pathways, your solution would be wrong. So the first important thing, which is multiple reactions in a linear pathway can be considered single reactions. So this is what we saw over here initially in this pathway, where I said the 2 which is assigned to this linear reaction, which I said I would tell later. So if a reaction is linear, the flux does not change because the metabolite is not branched into any other direction. And hence you can consider it as a single variable. And that kind of helps a lot. So you can use such things to make your pathways simpler, especially when you are dealing with complex pathways. So the second thing is, now it's still an undetermined pathway. So the next inputs you develop for this is consumption of growth substrates as major input to pathways. So when I say consumption of growth substrates, it would be, for example, glucose, which is being consumed. So rate of consumption of glucose. Or then second would be biomass composition, CHONS, the elemental analysis of biomass. So you know that these are the number of C's which are present in biomass. So you can relatively try to connect the dots and understand and have a more understanding of that pathway. So let's try to take a more simple example. If I have 2 variables in this equation, 2x plus y is equal to 4, I cannot solve the x and y straight away because it's just one equation. So now if we try to maximize the objective function, for example, 2x plus 3y, so I have just assumed an objective function. If you look at this graph, 2x plus y would be this red line in this graph. And if you maximize 2x plus 3y with all possible numbers, 2x plus 3y is equal to 6, is equal to 12, etc., you are kind of covering the area in which 2x plus y is equal to 4 lies. So this is a simplest example of linear programming, which is extrapolated in flux balance analysis. So in flux balance analysis, this is the objective function. So Z is equal to CIVI, where CI is the weights of biomass. So the objective function can vary based on what pathway you are trying to study. And there can be different objective functions other than just biomass. Maximize biomass is the most common objective function considered in flux balance analysis. But other than that, these are all the other examples which can be used for flux balance analysis. Okay. So let's consider this FBA example. So the arrows in green are building block flux. The arrows in purple are transport flux. So when I say transport flux, these are metabolites which are, or substrate or sugars which are either consumed by bacteria or the products which are formed and exported by the bacterial cells. So they are denoted by purple. And the ones which are intracellular flux denotes those metabolites which are present inside a cell. So over here we have total 16 variables. So as we discussed previously, we can develop these equations, these equations for these variables. So now we already have information of V1 and V6 as glucose, because you can measure these things using wet lab. So we have V1 and V6. And the building block fluxes can be assumed from biomass composition. So as we discussed initially, that maximization of biomass components can help us to get more equations, which can help us to solve these flux balance analysis problems. So when you feed all this, you get this complex matrix. So it's very difficult to solve something like this, and it might get more complicated. And it's very difficult to solve something like this with MATLAB. And that's the reason we try to use computational models or models developed by research labs like Professor Tang's lab for solving these equations so that we could get a flux map of a pathway. So now I gave you introduction of flux balance analysis, and now I'll give you introduction to 13C fingerprinting. So 13C fingerprinting, as name suggests, so we know carbon has atomic weight of 12. And when we say 13C, it's an isotopic form of carbon. So we call it labeled carbon. For example, glucose, in this simple example, let's consider that glucose goes to G6P, and there can be two pathways which can be active at this one time. So it can be either PP pathway or a glycolysis pathway. So if glucose goes to PP pathway, this first carbon is lost, as C5P has 5 carbons in its structure. And hence, when you study C5P, you won't find labeling in it. On the other hand, if glucose goes to glycolysis pathway, the metabolites like pyruvate and acetyl-CoA would have this one carbon, which would be labeled. It would have atomic weight 13 instead of 12. And if you find labeling in your metabolites, you would be able to predict that, which of the two pathways are active. So that is rough background of 13C fingerprinting. And that's the reason it's done, to understand in which direction carbon is flowing. And each metabolite fixes and forms different amino acids, which I would talk later why we study them. So this is one more interesting example of 13C fingerprinting, where, for example, if we have non-labeled carbon dioxide and labeled glucose being added in a pathway at the same time, if you see high enrichment of carbon, 13C, it would mean that there's higher uptake of glucose or acetate because they were labeled. So an organism is consuming glucose and acetate more easily if you see more labeling. if you see less labeling, it would mean that there is low 13C enrichment and higher uptake of CO2. So which substrate is being consumed by an organism can be more better understood using these labeling studies. So this is basics of labeling studies. If you label something, you would know which pathway is active, etc. But this can be taken at next level. So simply labeling studies, we use the simple formula of yield is equal to labeled carbon divided by total carbons. And then you can compare percent labeling in charts like these to understand which metabolite has comparatively more labeling than other metabolites. So we talked about labeling, but this labeling data can be used in a better way, and it can be used as one of the constraints in flux balance analysis. So first I talked about flux balance, now labeling, and now we'll try to combine this information together to get metabolic flux information about a pathway. So let's talk about 13C metabolic flux analysis. So metabolic flux analysis mainly can be used for labeling substrate in specific positions, and it can help elucidate fluxes. It can give us more information about about the flux map and in which direction the rate of the flux is higher using the labeling information we get from 13C fingerprinting. For example, in isotope labeling, in this case, the A goes to C, 2D, and F, where you see the first carbon being labeled in all the cases. The degree of carbon enrichment of first position of C will be directly proportional to rate of reaction and relative to total consumption of A. So this isotopic labeling experiment is used in metabolic flux analysis. So let's talk about how analysis is done. So for example, in this pathway, different amino acids mentioned here, mainly histidine, phenyl, alanine, serine, etc. So when your cell consumes sugars, it takes this energy and it's biosynthesized in form of amino acids, which are building blocks of proteins, right? Cells are made of proteins. And we try to use this bacterial biomass and hydrolyze and derivatize it, break it down, and try to study these amino acids, because these amino acids give us fingerprint of the carbon which is fixed into a bacterial cell. For example, something like oxaloacetate leads to formation of aspartic acid mainly. So if you study aspartic acid, the carbon labeling of aspartic acid, you could understand how oxaloacetate was labeled. So these different amino acids can give us information of different steps in the metabolic pathway. And that is analyzed mainly using machines like GCMS and LCMS. So let's talk about first method. So metabolic flux analysis in steady state labeling. As we talked about, we consider steady state. In this study, we normally take samples in the logarithmic phase when the cells are growing. And then we derivatize the cells and we do this GCMS. GCMS is used for development of MDV values, which is mass distribution vector values. And these values are fed into a software, which is mainly WooFlux in this case. There are different softwares like WooFlux. WooFlux was developed by Tang's lab. And this software gives you information about internal fluxes. So over here, you're labeling your bacterial cells. You are studying them in GCMS. You are processing the data in WooFlux, and then you are getting internal flux information about that. So I'll try to give you more detailed information about it. So once we harvest the cells, the bacterial cells are hydrolyzed and they're treated with TBD-MS. So TBD-MS is a chemical used in derivatization, and it mainly stabilizes the amino acids which are hydrolyzed initially from bacterial cells, and they help us in analysis in GCMS. Once we do GCMS, we get different peaks for different amino acids. For example, I have given here. So each amino acid would give you different peaks, and each peak would have multiple mass spectra or M by Z values, as you see in this small chart. So these M by Z values would change if your sample is labeled. For example, alanine has 158 as M by Z. If I add non-labeled alanine, which we find in nature, it would give you 158 as M by Z. But if I add glucose as labeled glucose and feed that to my E. coli cells, and then I study the biomass of that E. coli culture, my alanine value would increase by 158 plus 6 because glucose is 6 carbon sugar, right? So this M by Z information is raw information, and it is further processed to get MDV values. And this MDV values are used in softwares like WFlux to get internal flux analysis. So this is MFA via steady state labeling. This is one method. Second method is, for example, if you are trying to study labeling in something like cyanobacteria, and its carbon source is carbon dioxide. So as cyanobacteria consumes carbon dioxide, which is a sole carbon source, all the amino acids in cyano would be labeled completely. But you would like to study them when they are not completely labeled. So for that, we do 13C non-stationary MFA. So what you do that is when the cells are growing, you do not add labeled carbon dioxide initially. But when they reach steady state, when they're in log phase, you add labeled carbon dioxide at that point, and then you study the increase in the labeling. You take samples at different time points and you see how the labeling increase in that biomass. And then you process the biomass, you extract it using solvents like chloroform and methanol. You use GCMS and LCMS to study the M by Z values. And then you try to compare relative abundance. And this would give you more dynamic pattern of data. So in this case, as in the previous case, in example one, we used WFlux as a software. INCA is more commonly used, which was developed by Tang's lab as well, for 13C non-stationary MFA analysis. So yeah, this is metabolic flux analysis. And these are the two main methods I would like to talk about. So yeah, thank you. I'd like to especially thank Professor Tang for sharing his lectures to us. We are Varman's lab. And thank you for giving me this opportunity to present to all of you.
Thank you, Apurv, for such a wonderful talk. Very interesting. You have explained all the theory very clearly. Appreciate your time and sharing all the detail. So we have few questions from the audience. And then, so in mammalian cells, MFA faces various problems like unbalanced growth behavior, and achieving a true steady state is quite a challenge. So how we can translate this for such a complex system? Basically, how you can extend from microbe to mammalian cell, which is very complex.
Well, this is more very common in bacterial cells, but I have very less knowledge about how, because mammalian cells are comparatively so tedious to grow and they are adherent cell lines, but I haven't done it personally. I don't know if Dr. Varman would be able to have any information about how this can be done in mammalian cells, but this is mainly in cells like E. coli or non-model organisms like cyno and bacillus.
So yeah, Amit, this is your expertise as well, so you can add into my answer. One major challenge with mammalian cells is going to be compartmentalization, right? So then what we will do is I will develop a set of reactions for different compartments, and we will include exchange fluxes. And based on that, with those exchange fluxes, it would connect the metabolites moving across compartments. And like Apoorva presented, we will still go with development of models and estimate fluxes. Amit, if you want to jump in more on this, please.
Yeah, in the mammalian cell, rightly, Professor Arul told correct. We have compartment just like we do yeast. So yeast has very similar to mammalian cell. But yeast we can go to steady state very easily. So you can go to steady state where you can measure the fluxes. But in mammalian cell, you cannot go to a steady state. For that, you have to do another type of Inca you told, right? You told about Inca, right? So the Inca will be the right method where you cannot go to steady state. That time you have to solve the equation, differential equation. So you cannot use algebraic equation. So that is the difference. So most of the 13CFA, if you see, which is actually using steady state approximation, where you solve the equation numerically. But for a non-steady 13CFA, like the Inca method, which is published by Jamie Young. So you can
Apoorva, you
read
can
that
go to the
paper.
previous slide.
Professor Jamie Young from Vanderbilt University. You can he is actually working on mammalian cell. So you can read his paper. Right now, none of us are actually working on mammalian cell. But it is the method is same, so there is not much difference. The only thing you have to keep in mind that it is a non-stationary 13CMFA, where we are using you have to solve the differential equation using the software. The only software which is available is known as Inca. So using Inca, you can actually measure the fluxes. Yeah. So the next question is about the batch culture. When you consider the batch culture, the media composition is changing with time, and how the cellular metabolism has to adapt the changing environment. So you can shed some light how the cell physiology changes in terms of media condition.
So in this study, we mainly do it in batch culture itself. And although the cellular metabolism changes in batch culture and it's not completely steady state, but when the cells are in their logarithmic growth phase, in that case, the growth rate is constant with respect to time for some point. And that is where we consider a steady state condition. And that's where we harvest our cells, and we try to harvest them in the log phase itself, just to make sure that the amount of metabolites consumed and formed by the cells are in sync. So in batch cultures, we try to take samples in the log phase itself.
Okay, thank you. And one more question regarding if you know about the elementary flux vector or elementary flux modes in metabolic modeling, how you can use that, and what is the difference between these two techniques?
Elementary flux vectors and elementary flux modes.
In fact, my laboratory does not have experience with working with such models on it. So if you have some experience, you can add or not. Yeah, I'm sorry if we don't have answer for this question.
We are also not using basically elementary modes. You generally do it for a small set of pathways to see how it is influencing the entire metabolism. So it doesn't require any level experiment. So you can do on any model very easily. So it doesn't require any requirement like 13C. The next question is there any method to reduce the economics of the entire analysis process to make it more convenient for industries? So it is very, very interesting question because the 13C is very costly. So, and suppose you have to repeat the experiment, so you have to buy the level glucose, which is very expensive. So the industry will think twice to actually do this kind of experiment. Even for the academics also, sometimes it is very difficult to buy the ingredient or the raw materials to run the experiment, and then you feed the model. So is there any way to reduce the cost in terms of running 13C?
I think in 13C analysis mainly, you don't need to, so for example, in industry, if you just scale up the whole process and you are increasing the volumes, etc. But 13C analysis doesn't need a lot of sample. So you don't need to grow your samples in extremely high scale where you have substrate cost issues. You can study your samples in a small reactor and you can still extrapolate it with large scale reactors.
Apurva, that was an excellent answer. To add to what Apurva provided, so when we think about steady state, most often people think about using a bioreactor with a continuous flow in and a continuous flow out, and keep the cell density and the other factors at a steady state, right? Of course, if you do such experiment, it is going to be very expensive, and some really rich labs do it, but in our lab or in Dr. Tang's lab, we always do it in a shake flask or culture tubes, which wherein the volume ranges from like 5 ml to 10 ml. And like Apoor mentioned in the previous slides, we just take or we collect samples in the exponential phase, and we have never had issues. Okay, so all we have to collect is 5 ml of samples for one time point. Okay, so that is not very expensive. So only when you think about using a bioreactor, then it becomes expensive, but you can bypass that route by using small scale cultures and using and taking samples in the exponential phase. That is good enough. Next question, Amit.
Next question is by Sarath. He is asking, how can we choose the important reaction toward our objective function from a pool of more reaction? Is there any tool for rationalize the selection of reaction?
So it depends on it. It would change based on what we are trying to study. So if my work would include specific pathways, which, for example, in this case, we talked about objective functions. So it would depend on what we are trying to study. For example, if my products are mainly metabolites, which I mentioned initially, like pyruvate, etc. So maximizing biomass function is the most straightforward way. And selection of reactions would depend on what we are trying to study in that particular E. coli, particular organism. So when I say selection of reactions, you could try to reduce the number of reactions if they are linear reactions, but you need to be sure that you make sure you include the parts of the pathway which are part of your study. For example, as Dr. Tang said, he was trying to make products like ethanol in his pathway. So you need to make sure that the pathways where you have made changes are included in your reactions. If that helps to answer the question.
Yes, Apoor, you are right on target. Yeah. So yeah, it depends upon the experiment that you are performing. Based on that, you have to rationally choose the objective you want to use. Okay, and the important thing for you to keep in mind is the output of the flux balance analysis is an in silico flux. All right. So it's up to you to how to use it wisely to design your experiments or to perform analysis out of it. It's not actually an real flux. It's an in silico flux that approaches reality. Right? So with that, you have to digest that information, and it's up to you to how to wisely use it. Yes, Amit, if you want to add any, or we can jump to next question.
The next question is about 13C MFA. So, Manali is asking about 13C leveling experiment. How can you uh, uh, is there any bias in the distribution of 13C, the 13C leveling experiment on the process? How do you overcome the bias in the 13C leveling experiment
Okay.
in terms of distribution of the leveling?
So there is 13C labeling experiments are pretty accurate, and especially when I do them in duplicates, you would not see a lot of difference in the MDV vectors which we get when I study the amino acids. So there is very less bias. Also, if she feels that adding labeled substrate would affect the cellular organisms by any chance, we haven't seen any issues with the growth rates, etc., with the labeled substrate. If that is the side of the question, if she wants to ask.
So I asked this question, and this was the slide based on which I asked it. So when explaining, you said that the glucose is 6 carbon and one of them is labeled, but when it goes to the PPP, there are 5 carbons and the labeled carbon is not recorded here. Something along that line.
Yeah.
And in glycolysis, you get the labeled carbon. So my question was, why the labeled carbon is omitted from the, exclusively the labeled carbon is omitted from the PPP? And like, why is it not like the labeled carbon stays and one of the unlabeled carbon goes away? And if there is this sort of bias, then won't it be that this PPP information is lost from the system? Like we don't get what happens.
Okay, so this PPP pathway, I have just shown it as just one line. But if we closely look at the structure, so it depends on which carbon I label. So for example, I have labeled my first carbon in this case. You normally, if you look at a chemical reaction, you would know that if A goes to B, and if, for example, CO2 is going out, you would specifically know which carbon of glucose contributes to that CO2. So, for example, if I labeled third carbon, I would get labeling in PP pathway. So the way chemical reaction happens does not change when we label them. So that's the reason that that first carbon is lost, and it is not random. So every time I label them in any fashion, only the first carbon is the one which is going to get lost in that chemical reaction. And you can make variations in your labeling pattern in your glucose, and you can confirm your hypothesis using that. But it's very accurate. Carbon transition is very accurate in metabolic pathways. And that is the beauty of this, because of which you can accurately tell where a carbon is going and understand how these pathways are working.
Okay, thank you.
So basically, Manali, so if the molecule if the molecule glucose or the entire carbon goes through the PP pathway, the first carbon gets lost as carbon dioxide, and that's the reason you don't see the labeled carbon in the PP pathway metabolite.
Yes, sir. Yes, sir.
So it's
thank you very much.
So this is a very good question. So like Apurv answered, okay, so here this is a classic example where we try to show that by using first position labeled glucose, we can differentiate whether a PP pathway was highly active or was glycolysis highly active. But depending upon the pathway you would want to investigate, you can use different labeled carbon substrates, right? So probably, when I present my lecture, there could be 1 or 2 examples on fingerprinting as well. where we use different labeled carbon substrates and which pathways do we investigate there. So you can take a look at it. That should answer it much further. Yes, Amit.
The last question is.
Go ahead.
Continue.
No, I mean, like, if we have questions, yeah, we can go ahead. It's about 10 o'clock. So
the last question is there any methodology by which one can account for higher order reaction?
Higher order reactions.
I'm sorry, I don't get the question.
Is there a methodology by which we can account for higher order reactions?
I'm hearing it, but I don't, understand what he's trying to ask. Do you get it, Amit?
We are including, right? We are including higher order reaction in 13CI.
What do you mean by higher order reactions?
He want to tell that there are like first order reaction, second order reaction, like that.
Okay. Okay. So in a way to put, we are not. So to solve for fluxes, we are not writing down rate equations here, right? So we are going with the assumption of a steady state, where we assume that each of the intracellular metabolite is at steady state, and we do a balance of fluxes around it. Okay, so there is no rate involved. So it's just a balance. Okay, so if we want, you can go back to that example, Apur, where you have where you did a balance around PEP, right? That could explain to him again how these equations are written down. Yeah, even here, yeah, even there. Yeah, so you can explain one of the node. Okay, so we don't actually write down rate equations when you use computational codes. You won't see the model, but at the very basic or in the foundation, we don't write down any rate equations. In a way, something I think about is, that's one of the limitations. So as youngsters, you guys are listening. Okay, so this is the approximation we made, or the field has made, for which flux balance analysis have been developed. But as you are thinking, okay, so you could for different reactions, you can propose different rate equations, whether all of them can be inbuilt into the model. That's something you could think about. Okay, so this could lay a future direction for you. Apur, do you want to explain, how we write down equation for one metabolite?
Yeah, so as we saw here, that the rate of change of concentration with respect to time, we consider steady state. So stoichiometry into flux is 0. So for example, let's consider PEP. So 1 goes in and 3 and 2 are leaving PEP. So when you develop an equation, the flux V1 would be positive and V2 and V3 would be negative. So your equation would be V1 minus V2 minus V3 is equal to 0. So your steady state assumption dx by dt is 0. So this first equation would be equal to 0. So I wonder, I don't think we have like second order rate equation, etc. We just consider steady state as 0. Right? This is one way of just making an equation for PEP. The same way you can make equations for ATP or pyruvate.
Yeah. Or in other way to answer your question, within when you look at the genome scale, so you have all those reactions. So we include all the reactions irrespective of what order it is to build the model. Okay, so as long as we have the network, we can build this model as shown here. So we don't think about whether this falls under first order, second order, or third order. Okay, so that is not involved in the decision making. But it's a good question, something you can think about. We always ponder about the accuracy of the result from these, but there is always room for improvement. You can see how you can improve this model. Okay, so that is something you can explore. Good question.
Okay, we are going out of time, so the next speaker is waiting. So thank you, Apur, for a wonderful talk. You gained so nice insight from this flux analysis so early. Thank you for your effort and sharing your research and teaching to us. Thank you, Professor Arul, for sharing your insight and appreciate. I will get back to you today in the evening. I will listen to you today evening about your research work. So I invite the next speaker right now. Thank you, Professor Arul, and thank you, Professor.
Okay, so everyone enjoy the talk. See you tomorrow morning for me and tonight for you.
Yeah, thank you. Thank you so much. So our next speaker is Professor Pinaki Sir, who is from IIT Kharagpur, Department of Biotechnology. He is my colleague. We teach together metabolic engineering course for the undergrad. And he is a senior professor who got his PhD from Banaras Hindu University. And then he is a research, Dr. K.S. Krishan, Research Associate at Bhabha Atomic Research Center, Mumbai. And as well as he is a Bioscast fellow at Rice University, Houston, USA. His major research area in the field of microbial ecology and environmental biotechnology using both culture-independent metagenomics as well as culture-based approaches. This includes understanding the role of indigenous microbial community in mobilization of arsenic in groundwater of West Bengal Delta Plain, in addition to the structural and functional analysis of microbial community in mine sites and potential for bioremediation. So his talk for today is about the enhancing microbial appetite for toxic pollutant. That is advances in metabolic engineering and systems biology for bioremediation. So I welcome Professor Pinaki Sir to give his lecture and continue.
Good morning, everyone, and thank you, Dr. Ghosh, and all other participants. Dr. Ghosh is deserving a special thanks for inviting me and also for organizing this workshop. And I will just take maybe a minute or so to share my screen with you. Okay, please bear with me. Hello, Amit. Hello.
Hello.
Amit, there is some issue in sharing the PowerPoint slides. Can you please help me in that?
Yeah.
So I'm just clicking.
should I go for open system preference and
open system.
Okay. Are you able to see my presentation?
No.
Amit, are you able to see this presentation now?
No.
So how to do it? Can you please
select desktop. And then you
select desktop and then share.
Then you can use any other option.
No, this share is taking me to open system preference.
If it is not working, you can use the other version. It should open. Like if you share the desktop, then it will open. Okay, maybe you can try. Maybe I can start the talk. Is that okay? In between, you can.
It's
Okay, correct.
Amit, are you able to hear me?
Yes, sir, it's audible.
Okay, thank you. And I'm really sorry for this inconvenience. So anyway, so good morning, everyone. And I'm going to talk today on the topic uh, enhancing the microbial appetite for toxic pollutants. And okay, so enhancing the microbial appetite for toxic pollutant, advances in metabolic engineering and systems biology for bioremediation. So increasing pollution of air, soil, water, surface water, groundwater everywhere is a major threat to public health, and that all of us are aware of, both in the developing countries as well as the industrial countries, including the European Union states, the US, the India, China, everywhere. So this is the NASA generated map on particulate matter in our air, and this is an European data suggesting how our soils are contaminated or degraded by different anthropogenic activities. And the major pollutants of concerns are the heavy metals and organic compounds, particularly the hydrocarbons of different aromatic rings, alkanes, halogenated compounds. Interestingly, many of these organic polluting compounds are xenobiotics uh, of anthropogenic origin, and they are used in agricultural, industrial, or government services like military, etc. And among these we do have the emerging contaminants like the pharmaceuticals, the wastewater products, personal care products, and different type of nanomaterials. Now, despite these recalcitrant nature of some of these pollutant compounds, including the pesticides, herbicides, plasticizers, pharmaceutical, etc., many of these compounds are more or less susceptible to biodegradation. Now, biodegradation is basically referred to the term where biologically catalyzed reduction in complexity of the organic compounds are manifested. And mostly it is carried out by indigenous microorganisms present in soil, water, and any of the natural environments. And these microorganisms, they use their metabolic abilities to obtain energy and other required precursor molecules from the pollutant organic compound using different pathways of biodegradation. And these processes of natural microorganisms often proceed without any human interventions. And during this bioremediation and the metabolic engineering uh, aspect, we actually need to work on these natural microorganisms and try to harness their metabolic abilities to achieve a better degradation abilities. Now, microorganisms, particularly bacteria, are the major entities of transformation of the pollutants, both organic as well as the inorganic compounds. Now, the major entity that causes large-scale transformation in the biosphere are the microorganism because of their incredible number, incredible abundance, and versatile metabolic pathways. So, as you can see here, this very simple schematic diagram I have used to represent different type of organic compounds which are used by microorganisms as their source of carbon and energy, just like a glucose molecule or yeast extract molecule, and eventually converted to carbon dioxide or intermediate metabolites. So the degradation of the organic compounds could lead to either the complete mineralization of the compound, like in this case CO2, or it could be producing intermediate metabolites, or it, it might be a mechanism where direct metabolic involvement of the organism is restricted to what we refer as co-metabolic activities, and it can proceed in both aerobic and as well as anaerobic conditions. Now, why do we select microorganisms for these biodegradation and bioremediation studies? Because they are the first life form to evolve in this planet. They are the most ubiquitous, abundant, metabolically most versatile, with highest genomic diversity, and they represent about 2/3 of the entire domain of life. Now, specifically, when we think of engineering these organisms for their better performance, their small genome size, relative simplicity of the cell functioning, cell physiology, the short replication times, evolution and adaptation to the new environmental conditions are quite rapid. All these factors are considered. Now, many of these microorganisms are extremophilic, so they can grow up to a temperature of 121 degrees centigrade, or they can survive up to 130 degrees centigrade. They can withstand gamma radiation up to 5 megarad, which is more than 10,000 times what is lethal for a human being. They are also resistant to high hydrostatic pressure, high osmotic pressure, extreme pH conditions, and many more extremities. It is not only their physiological abilities to withstand the extremities, but also their metabolic robustness. So many of the organisms, as we have seen, maybe we will discuss gradually in this lecture, that the picky eaters like Pseudomonas, which can actually consume or degrade more than 100 substrates or even more than 150 substrates. We do have oligotrophic organisms which are very fastidious, and they thrive only on very selected nutritional substrates. Now, one of the major factor that we have learned over these last 2 decades, that the natural microorganisms, when they are isolated, they are studied, and they are to some extent engineered, they can metabolize a large number of compounds, metabolize up to the extent that compounds are converted to benign compounds like carbon dioxide, as you can see here. So this is about the metabolic capacity of Pseudomonas putida strain KT2440, which is able to convert a large number of diverse organic compounds, substituted organic compound, halogenated compounds, and funnel the carbon towards the central metabolism, producing finally the carbon dioxide and certain other desirable metabolic products. Now, microorganisms, particularly the bacteria, are the most favorable candidates for bioremediation technologies. When we say bioremediation technology, it is basically a pollution treatment technology that uses microbial metabolic process to reduce, eliminate, or contain or transform various contaminants. And this can be applied both in situ as well as ex situ processes. So bioremediation is basically allowing us to achieve the circular economy and the fourth industrial revolution, where the removal of pollutants as well as valorization, that is converting the trash into wealth, is being achieved successfully. So it is not only a waste remediation technology in today's time, it is also recovering the valuable molecules and resources, including energy, from the waste molecules. Now, in order to understand the scope and the application of metabolic engineering in bioremediation, we need to study the progress in bioremediation, that how it is actually practiced over the last maybe 4 decades or so. So although we know that there are directed bioremediation like microorganisms were in use for treatment of wastewater, etc., from 19th century, it is only about half a century that we started isolating the bacterial organism, bacterial strains from different contaminated sites, and started studying their degradative pathway. So it is more precisely like 45 years or maybe 48 years from now that we see published literatures available on use of bacterial strains for degradation studies. And the first report on in situ remediation of soil contaminated with petroleum-derived hydrocarbon was published in 1975. Now, following this initial isolation and application of natural microorganisms in remediation of contamination, organic contamination particularly, natural microbial degraders, microbial strains, bacterial strains were applied with success worldwide, like large-scale wastewater denitrification, uranium removal from wastewater, from groundwater, degradation of dichloroethane from groundwater, organophosphate, phosphorus pesticide removal, all through the use of selected cultivable pure culture species and just relying on their intrinsic degradative abilities without much of engineering or say metabolic engineering or recombinant DNA technology based modification of their metabolic pathways. So this is generally termed as bioremediation 1, because the advent of technologies for pollutant removal using naturally emerging microorganisms. It was solely based on the natural abilities without much of engineering done on their genome or their biological pathways. However, what we observed over these isolation-based methods, that these strains are often capable of degrading many of the organic compounds, but they are not capable of degrading mostly the anthropogenically produced compounds like the persistent organic pollutants, different type of polychlorinated biphenyls, dioxin, and many other pesticides, including the plasticizer and other compounds, which continued to be resistant to natural degradation due to lack of efficient microbial catabolic traits. Now, when we say that lack of efficient catabolic trait, it means basically lack of appropriate enzyme or efficient enzyme who can or which can convert these toxic compounds, the persistent, mostly anthropogenic compound, into substances which are easily metabolized by bacteria or other organisms, if not up to the carbon dioxide. Now, with the rise in recombinant DNA technologies, transformation of bioremediation, transformation of different type of bioremediation relevant reactions from empirical practice into an exercise of genetic engineering. So we started using recombinant DNA technology to genetically engineer many of the naturally obtained isolated organisms, which are naturally very efficient organisms, but we wanted to increase their efficacy, increase their substrate range, including the anthropogenic and highly recalcitrant organic compounds. So in this regard, the discoveries in molecular biology and progress in biological engineering helped us towards the rational interventions in the metabolic networks of selected hosts. Because with the onset of the discoveries and knowledge on metabolic networks, metabolic flux analysis, so the microbiologists started working on looking into the metabolic pathways which are important, which are critical for degradation or oxidation of the recalcitrant and anthropogenically produced compounds, which eventually led to engineering the whole microbes. So again, these are engineering the microbes which were naturally obtained, naturally isolated, and engineering with respect to their biodegradation pathways, particularly with the enzymes that increase their substrate specificity, increase their the product range, and make them more suitable for a diverse in situ applications like on-site applications. So that is referred as bioremediation 2. So from the bioremediation 1, which was purely utilizing the native culturable bacteria, the bioremediation 2 allowed the use of recombinant DNA technology and genetic engineering to engineer the microbes with respect to individual pathways or reactions, and thereby making them more efficient towards in situ pollutant degradation. This particularly, if you want to refer specifically to this happened around maybe 40 years from now, at the generation of superbugs, what we generally referred as. So these superbugs, which are much superior than their counterparts, these bacteria are expected to provide an economically feasible, environmental friendly alternative to the costly conventional technologies for pollutant removal available at that time. So these organisms are mostly generated in the laboratory, which is a kind of a very efficient technique of molecular breeding. So one of the first report came in 1980s, which is referred as the golden era of biodegradation, because the scientists started generating recombinant Pseudomonas strains, Pseudomonas putida strains by plasmid-assisted molecular breeding. And in this regard, Professor Ananda Mohan Chakrabarty is referred as the superbug superhero. The Indian worked in the University of Illinois for a very long period of time and obtained the first patent on the genetically modified microorganism through the molecular breeding. So his work was well debated, well cited, well exploited, and well discussed, and possibly paved the way for metabolic engineering towards getting the best out of the class microorganism for biodegradation and bioremediation. Soon after this, the molecular breeding was found to be successful in the plasmid-assisted mechanism. Patchwork assembly was adopted, wherein the recruitment of complementary enzyme sequences by conjugative gene transfer and bringing all these efficient genes from different organisms to a single host organism provided a very efficient way of degrading the contaminants because that provided perhaps the first round of functional synthetic groups. Because in the patchwork assembly, the several key enzymes or efficient enzymes were brought within the plasmids, and the plasmids were transferred into a suitable host, and then the suitable host was supposed to be or generated to function more efficiently, and was called as a synthetic base for the degradation of compounds. So one of the first report that we see for this kind of patchwork assembly is in 1987 for different kind of chloro and methyl aromatics. So entire the orthocleavage pathway for the degradation of these organic compounds, you can see here from multiple strains like from critical enzymes from 5 different catabolic pathways of 3 distinct soil bacteria have been combined through a plasmid in a patchwork fashion into a functional orthocleavage pathway and then deployed through the host organism. Now, unfortunately, what has been observed that although these recombinant organisms, engineered organisms with the patchwork style were successful to degrade some of the compounds, but were unable to degrade many of the compounds. So except for one enzymatic, the pathway was fully regulated, but it was not successful for other type of compounds which are available along with this particular type of compounds. So despite some success with the patchwork strategy, which are found to be very efficient with a selected 1 or 2 organic compound, but not for the other compounds of the same class like the halogenated organic compound or the benzene group of compound, including this, the engineering of superbug with extended substrates. The initial and rather the native approach led to the many disappointment because as a research point of view, it was very successful, very interesting. All this patchwork strategy and doing the molecular breeding. However, from a real in situ application of such superbugs or such engineered organisms were not often successful. Like the engineered Pseudomonas strains did not grow on 2-chlorotoluene as the sole carbon source, and 2-chlorotoluene is one of the major pollutant in many of the organically polluted sites. So we need to improve this biodegradation pathway, biodegradation mechanism beyond this patchwork and the development of these superbugs through molecular breeding. So metabolic engineering of bacteria for environmental applications, perhaps the same study which discussed very specifically around 2 decades ago the drawbacks and the flaws which are associated with this. So now reasons for these failures were nicely explained in this particular study by Di Lorenzo. We realized that the lack of insight into the important factors like the thermodynamic feasibility of assembled catabolic network. So it is not just a kind of engineered artwork that we bring the important genes only in a particular plasmid and then transfer the plasmid into a suitable host, but we need to look into the thermodynamics feasibility that we actually regularly do in metabolic engineering. Kinetic characteristics of enzymes and physiochemical properties of the metabolites, expression level of the pathway modules, crosstalks between the endogenous and exogenous metabolic routes, and stress responses and changes in overall host physiology after introduction of the new metabolic modules and exposure to toxic substances and metabolites. Mind that these are all discussed and highlighted by people who are purely working on bioremediation and bioremediation related strain improvement. They were not directly working on metabolic engineering. But when we look into all these aspects, the thermodynamic feasibility, expression level studies, crosstalks, stress responses, and overall physiological maintenance of the host cell, these are all very important criteria of successful metabolic engineering of organisms. So this understanding that we are lacking insight on these important factors about the bioremediation relevant microorganisms led to the onset of the new era, wherein systemic biology is incorporated, which merges the different approaches of systems biology, metabolic engineering, and synthetic biology for the sake of understanding and reprogramming the biological system. So system biology has the potential to remove the unknown and the bottlenecks encountered in the past trials and pave the way towards the era of bioremediation 3. The molecular breeding, which has led to the discovery of the superbug, the patchwork assembly, they were all done very intelligently, but they failed to perform in the real in situ application front, possibly because they were suffered from unknown and some of the possibly known limitations which were not looked into because people were not able to understand the importance of these the broad, the holistic metabolic function of the organism. Now, the joint power of the systemic biology disciplines, the metabolic engineering as well as the system biology, can ensure that biodegradation and bioremediation using genetically modified microorganism will remain a vital concept, deserving of the full attention of the new generation of bioengineers. Now, the approaches and tools of systems biology and metabolic engineering for tailoring the biodegradation pathway. Now, metabolic engineering is defined as the directed improvement of product formation or cellular properties through the modification of specific biochemical reaction or the introduction of new ones. It was defined originally by Bailey in 1991, and later it is understood as the practice for optimizing genetic and regulatory process within the cell to improve the yield and productivity of native products, including the metabolites which could be produced by the degradation or oxidation of the pollutants. Establish the synthesis of products new to the host cell. Often if we are exposing our cells to purely anthropogenic or the xenobiotic compounds, it is expected that within the cell some new metabolites will be produced. The host cell were unable to understand that what type of products are these. And the 2 points especially relevant for biodegradation because the yield productivity as well as the production of the metabolites which are new to the host cells, as well as the extension of the substrate range and their uptake. Because in the natural environment, when we intend to release our organism and we intend that these organisms are going to function very well, it is hard to believe that the contaminated site will have only one type of compounds. It may have a class of compound, but within that class of compound, there might be a range of substrates present. So the organisms must be able to utilize a range of substrates and utilize in the sense, utilize with respect to the degradation, activation, and their metabolism, as well as their uptake inside the cell. And overall cell robustness. The cell has to be robust enough to withstand all these kind of genetic engineering as well as the toxicity and the stress expected because of the production of these metabolites or the other toxic compounds which are likely to be present in the cell in the real environment. So these are basically amended along with the original definition of metabolic engineering in 2014. Now, all these goals, whatever is mentioned over here, can be achieved by engineering the natural metabolic pathways in the host cell, or it could be utilizing the synthetic route. So we have isolated organism, as I mentioned before, for the last maybe 1975 or before that, we have been isolating organisms which are suitable for different biodegradation and bioremediation processes. But engineering them in a metabolic engineering perspective would be required, not engineering them as just for a recombinant DNA technology approach. It has to be looked into a more holistic way, like the metabolic engineers generally do that, or incorporation of the synthetic routes, which are generated using the different type of synthetic biology concepts. Now, the general metabolic engineering strategies, which are generally adopted for the improvement of product titers, can all be utilized for improvement of the biodegradation abilities of the cell, like including the increasing supply of the precursor cofactors, overexpression of the regulators, removing the bottlenecks, improving the enzyme specificity, and blocking the competing pathways. So all the necessary steps for the routine metabolic engineering were found to be fully applicable to for the metabolic engineering of the remediation relevant organisms. Now, with respect to systems biology, which is an integration of the data from all level of complexity, including the genomics, proteomics, metabolomics, and other molecular mechanism using advanced computational method to study how networks of interacting biological components determine the properties and activities of living systems. Now, unless we look into the networks of interacting biological components, the success of any biological transformation will remain incomplete. So with respect to this system biology concept, its intervention into the bioremediation studies, one key objective is to gain the comprehensive quantitative understanding of the cells by combining the high throughput techniques that we generally do routinely in metabolic engineering for the product development of the biofuel or maybe the drug production or other, but the bioremediation relevant organisms were adopted for the same set of studies little lately. Now, these concepts of the system biology and metabolic engineering, they coincide fully with the objective of each other. And system biology and metabolic engineering are now inseparable and complementary disciplines, so that all the bioremediation people have understood that it has to be looked into like in that way only. And that the amalgamation of the system biology and metabolic engineering leads to the development of the systems metabolic engineering for the bioremediation of organisms. So metabolic engineering engineers use systems biology, computational tools and omics technique to gain a deeper insight into the genetic and physiological background of the target organism. So the message is very clear that we may have efficient degradative organism, but we might be lacking the insight into the genetic and physiological background of the organism. Merely sequencing the whole genome or sequencing one or few genes or the operons or their regulators are not enough. We need to understand the function of the whole cell in its entirety, and then model the enzymatic reaction and find out what are the constraints for the efficient biocatalysis. That means the bio transformation of the pollutants. Now, in order to overcome the constraints, the computational tools are generally applied together with the established experimental protocol. So both application of computational tools, which are mostly provided by the system biology people, and then the experimental biologists, they use the experimental setup with the improved idea, improved design of the experiment, and then the overall improvement of the organisms evolve through a fine tuning of the laboratory evolution experiments. Now, this is particularly referred as design, build, test, analysis cycle, DBTA cycle. So we design based on the information available with us, then we build the organism with the suitable gene and regulators and other things. Then we test it for the degradation or other performance. We analyze the performance both in vitro and in silico, and then we go for a second round of improvement on that, and it goes on. So this DBTA cycle is a kind of repetitive cycle until the performance of the engineered cell or cell factory is optimized. So that concept has been brought into the bioremediation study. So it has found that this is the only thing which will help us for intellectual workflow to engineer the biodegradation path. So what are these then the major steps of metabolic engineering and systems biology or systems metabolic engineering? So the first is know the contaminant and find a suitable catabolic pathway. The second would be the select and get to know a suitable host and build the pathway and optimize its performance in the context of the host metabolism. That is going to be the last option. So step one is the know the pollutant and find out the pathway. So know the contaminant and find a suitable catabolic pathway. Now the know the compound to be degraded is very important. Okay. There is nothing called ad hoc selection. So in that type of site, those type of pollutants are there. Prior knowledge may be useful, may be important, but we need to know before we deploy or before we start working on developing strains through metabolic engineering for remediation purpose. So identifying the compounds, its physicochemical properties, heterogeneous physical niches, because environmental setups are not always exactly like a kind of a bioreactor where everything is under control. So environmental samples setups like the open ecosystems of soil or water or other setups are highly heterogeneous with micro variation in the physical and chemical parameters, as well as the concentration range. The variation is also there with respect to the concentrations of the pollutant, the particular pollutant, and associated pollutants. And this may vary in several orders of magnitude from nanogram per liter to milligram per liter. Then comes the most suitable pathway for degradation. Ideally, the mineralization. Mineralization means the complete conversion of the compound from the parent compound degradation up to the mostly the carbon dioxide and water. So for this, the finding out the suitable pathway can be achieved by using the computational tools as if it is done by the other metabolic engineers, where the physicochemical properties of the target compounds or the basic building blocks of the catabolic pathways like the enzymes and metabolites can be used to find out that what could be the best pathway for degradation of this type of compounds. Use of different databases and prediction systems. So there are development, as I'm going to discuss about that briefly, that which will provide us the information on biodegradative or biosynthetic routes of different organisms which are already available to us. So one of the first kind of its kind database for bioremediation or biodegradation studies was started with University of Minnesota, a biocatalysis biodegradation database and pathway prediction system, very popular and used widely. It is later taken up by the Swiss government and updated further, and it is now available as EAWAG BBD. This remains the first choice search engine for finding out the organisms and their pathways, the enzymes, and perhaps their thermodynamic abilities, etc. Now this allows the search through 543, or maybe now it is slightly increased to 550 organisms or so, including both bacteria and archaea and fungi. 219 metabolic routes. Routes means the complete pathways. 1,500 plus reactions, several chemicals, and almost close to 1,000 enzymes are well described, and we can have a search based on these parameters. This also allows the prediction of novel biotransformation reactions, leading from target xenobiotic substrate to the molecule that can be metabolized by central catabolic pathway like acetyl-CoA or pyruvic acid. So this is the web page of this NVPath, which provide us the designing of biochemical routes, including the prediction of new catabolic pathways. So we can upload the genome sequences, or we can use the genome sequences available over there and the organismic details available there to find out how particular type of organism can metabolize, or up to what extent the organism can metabolize a particular compound. There are also databases like MetaSeq and BioSeq, where substantial number of experimentally elucidated pathways are included. And the MetaSeq is one of the largest repositories in metabolism data, and it serves primarily as an online encyclopedia for prediction of metabolic routes from available sequence genome. It can be browsed very easily, and it includes the Gibbs free energy value. So the thermodynamic feasibility of the individual reactions can be calculated based on that. BioSeq is having 9,000 plus organism specific pathway or genome databases within that, and each encompasses sequence genome and predicted metabolic network of single organism. It can provide us the predicted operants, metabolites, enzymes, their reactions. So the first task, that the identifying or knowing the contaminants and identifying the potential pathways are for their degradation, are substantially worked out by using these databases. Now this MetaSeq and BioSeq are linked to numerous other databases For example, Uniport, which provides the missing information on function, sequence, taxonomy of proteins, etc., or RCSB protein data bank, or Brenda, more specific protein database, completing the picture with structural or kinetic data. So the extent of information available on the required enzymes or the predicted enzymes could be possible through utilizing these databases. The next is the pathway prediction systems and toxicity prediction algorithms. So significant development has happened with this pathway prediction system and toxicity prediction, because many of these xenobiotic compounds, when they are metabolized or degraded, the products might be toxic. So I don't know whether my product or metabolic product will be toxic or not. So for numerous anthropogenic chemicals released into the environment, the catabolic pathways are either completely unknown or poorly understood. And the development of reliable pathway prediction system is of major importance. So it is not only that knowing the known genomes or analyzing the known genomes, but also development of the reliable pathway prediction system. Now these tools, which are based on different new algorithms, consider many factors like substrate specificity, binding site, reaction mechanisms of enzyme, structural changes of substrate product pair, etc., etc. Now, pathway prediction system in UR BBD PPS. So in the BBD database, we have the facility now to have a pathway prediction. It can highlight initiating reaction types likely to occur in aerobic environment, or it can function like in extended other environment also. So it can actually provide us a very important clue that, for example, in this case, the emerging contaminant 1,2,3 trichloropropen. So this is the predicted biodegradation pathway for this emerging contaminant, which was never present in our environment. So naturally, microorganisms often do not have any biodegradative reaction pathway for degradation of such compounds. But using this BBD pathway, where the predictive models are available, the aerobic degradation, the likely or the neutral situation can be obtained. And now, based on these, the scientists can work further to identify how to find out the suitable enzyme for such reactions. Now, detection and quantification of the pathway building blocks, the enzymes and the metabolites. Now, once the suitable pathway is selected, like using the BBD pathway, we can identify the predicted pathway and validate those pathways and find out the one of the best pathway which could be feasible. Now, the detection and the quantification of the building blocks. One of the most common method of detecting these enzymes and proteins are the chromatographic separation, followed by mass spectrometric analysis. In this case, the SDS page and Western blot analysis, followed by determination of enzyme activity. And the selected reaction monitoring mass spectrometry protocols are also developed, which provides information on rapid simultaneous quantification of many proteins in the cell. So this enables us to find out the metabolites, including the enzymes, which are present or which are produced inside a particular cell when they are exposed to the target compounds. Next, once the pathway is predicted, the enzymes are identified and they are quantified. The next step is the selection and knowing the suitable microbial host. Now, selection of a suitable host is a crucial initial step. It is not advisable that any organism of your choice can be used as a suitable host. So several microbial hosts have been considered for application for in situ development of the bioremediation strategy, but not a single naturally isolated bacterial strain possesses all the desired characteristic of the optimal degrader. So soil bacteria generally found to satisfy many of the requirements for a suitable host. Why soil bacteria? Because soil bacteria thrives in an environment where conditions vary significantly within their niches. They are naturally exposed to different environmental contaminants, including the heavy metals and other organic pollutants. And there is always a competition between the different type of species for the nutrition and for the predator species. Generally, it has been found that the soil bacteria, they possess very versatile metabolic lifestyle, which allow them to adapt to changing conditions, sometimes adverse conditions like oxidative stress, temperature challenge, osmotic perturbations, etc., which to a very practical sense are highly desirable for a bioremediation relevant organism, ability to withstand the multiple stress. So one of the most successful case is the isolation and characterization of Pseudomonas putida KT240, which is the one of the best characterized organism isolated from soil, and then characterized. The genome has been sequenced long time ago. Nearly 18 years ago, the genome was sequenced. And it was found that these kind of Pseudomonas type of organisms are naturally occurring. Naturally, they are able to degrade several aromatic compounds, and they are mostly plasmid bound, rapid growth, low nutrient demand, extremely versatile metabolism, high capacity to supply redox power, and endurance to oxidative stress. So most of these desirable activities make such kind of natural soil bacteria an ideal candidate for bioremediation related metabolic engineering. Now, most of these properties arise from the robust central metabolism. What has been found with respect to these Pseudomonas type of organisms, that they have a very important and significant amalgamation of both ED as well as EMP and pentose phosphate pathway, which are all well supported by peripheral reaction. So overall, their central metabolism is very robust. So when we go for the suitable host organism, one of the criteria must be, along with all the stress resistance and metabolic versatility, is the robustness of the central metabolism. So it is able to handle the flux very efficiently. Now, there have been very systematic studies over the past years to identify organism and then develop what we call bacterial chassis for metabolic engineering. So how we can take an wild type bacterium like the Pseudomonas putida, then deploy genetic tools or supported by genome sequencing, physiological characterization. Then the metabolic models are included into that to find out the possible scope for improvement. Mutants are collected, mutants are developed, and then the functions are streamlined. So maybe I will just come in a few minutes onto that how this is being done. Now the next aspect is the omics techniques to study the bacterial degraders. System biology, particularly the omic technique within system biology, the use of omic techniques contributed markedly for the rational selection of suitable host. So one aspect is the available genome data enabled new insight into the evolution of microbial metabolism towards biodegradation, which are available in the different databases. 16S rRNA phylogenetic microarray, functional gene microarray are also being used regularly to monitor the microbial community dynamics within the contaminated site or within the laboratory-based microcosm or mesocosm study to understand how the degradation is carried out by different organism, or how the organismic preferences vary when they are exposed to different phases of this contaminant exposure. Next is to build the pathway and optimize its performance in the context of the host metabolism, wherein the computational tools are used for pathway and strain improvement. So both mathematical model to identify, to select and analyze the flux distribution and identify the reactions which are considered to be maybe the nodes or the limiting points, and then successfully they can be modified through different kind of in silico and then in vitro experimentation. Genome scale metabolic models are also developed by comparative genomics, are built by compiling the data on genes related to biochemical reactions from databases such as KEGG and BioSeq. And finally, the flux balance analysis and metabolic flux analysis both are used to develop the metabolic models to predict the intercellular flux for the xenobiotic or other pollutant compounds. Now, experimental tools. When the distribution of metabolic fluxes and the rate limiting reactions or the bottlenecks are identified through this modeling process, the experimental techniques are used. A large number of experimental variation can be created in the laboratory, and engineers are traditionally conducting these experiments at the level of gene expression, like after exposure to different experimental conditions. One of the best way to look into the gene expression, either using microarray or maybe the transcriptome sequencing. So the genes in native or synthetic pathway are usually overexpressed by introducing the plasmid vector. So the overexpression of desirable gene, knocking out of the undesirable gene, and studying the overall expression of the genes when they are exposed to different compounds are all being attempted. Now the synthetic biology approaches and tools for biodegradation pathway analysis or engineering. Now the principal underlying thought in synthetic biology is that any living system can be considered a set of separate usable components. It's a set of usable components that can be combined by means of biological engineering in new arrangement, which is to some extent similar to the concept that is used by electronic engineer or computer scientist to produce predictable and robust system. Okay, so we have separate usable components which can be combined. The systems could be the genetic control system, metabolic pathways, the chromosome, whole cell at different level. We can construct the robust system. Now, the development of robust microbial chassis that I was referring to earlier was one of the major area for developing or for allowing these genetically combined pathways to house inside the host cells. So genetic instability and negatively affected fitness of organisms are frequently encountered for natural organisms. Many of the natural organisms are unable to bear the plasmids that are added, or they are negatively impacted because of the plasmid flow, the metabolites accumulated, or they are unable to survive in the stress which are experienced in the natural environment. So synthetic biology offers possible solution for this problem by implementing DNA synthesis and genome editing strategies. So in this case, the microbial chassis is developed where the cells with reduced genomes are developed. So the deleted, many parts of the genomes are deleted very systematically, and these deleted parts of their genome that encode redundant, cryptic, or even deleterious functions, thereby reducing the genomic load of the organism and creating the scope for housing these extra genes within them. Now, discarding the non-essential cell functions show considerable potential not only for biosynthetic purpose, but also for the designer biodegradation and bioremediation. So unnecessary genes are often discarded to prepare that. Now, a recent report is on the systematic deletion of 11 non-adjacent genomic region of a P. putida KT2440, which is an unique example, where we can see a large part of the genome is almost removed in order to make it a more streamlined and more stable and capable of functioning more efficiently with respect to multiple xenobiotic compounds. Now, lastly, the development of synthetic microbial consortia for enhanced biodegradation and bioremediation of pollutants. So long we discussed about the development and the improvements done through metabolic engineering, concept of metabolic engineering and system biology for individual strains. But at the same time, development of synthetic consortia or natural consortia with engineered systems are used for enhanced biodegradation and remediation of pollutants. So this is an alternative strategy. It allows for the emergence of more robust microbe-based bioprocess. It is opposite to the systemic development of bacterial chassis. So in this case, we are not emphasizing on developing a chassis by reducing the genome and making more space for the desirable genes to be expressed within that, but it is kind of developing the community function of many species within that. So using the natural microbial consortia. So for example, so one of the best case studies was this, that where we saw that the engineering of bacterial consortia with adhesin. So in the left side, you can see there are 5 species, each of which are capable of degrading to some extent a particular compound, but not to the full extent. They are possibly complementary to each other. That means if we put all of them together, they might function towards the mineralization of the compound, because all of them possess a very important step encoding enzymes, which is different from each other. However, these organisms often do not want to live together or stay together. So genetic engineering or metabolic engineering is done with respect to like incorporation of a particular adhesine protein and the respective release of antibodies by the other organism, so that the one organism harbors the docking site for the other organism, and all the four organisms which are represented in the community actually stay together with physically in contact with each other, and thereby facilitating the rapid and more efficient conversion of the organic pollutants to carbon dioxide. So with this, I would like to conclude my lecture today, that synthetic biology, which has brought a renaissance of bioremediation using anthropogenically enhanced microbial degrader. But it has also paved the entry for a new era of bioremediation, which is called bioremediation 3. So it is not only building up new strains or new pathways for remediation, but also a new way of understanding and looking into the application of bioremediation. The bottlenecks which are described at the beginning of this, like there are many limitations which were identified, are gradually released or removed with the help of the new computational and experimental tools. Because once we know the data using the different computational tools or different databases, and then validating them through experimental protocols or experiments, we are more close towards removing each of those bottlenecks. However, despite a clear progress over the last decade for biochemical and biological engineering, the complexity of the living cell remains vastly unexplored and unknown to us, and it is still a major hurdle for any attempt for full rational pathway design, particularly for the bioremediation relevant organisms, because these organisms may not belong to always organisms like E. coli or Bacillus subtilis, for whom the complexity is still to a large extent is resolved. Now, one approach to overcome the complexity of living system towards bioremediation is to fine-tune the performance of the selected organism following their isolation from natural environment by the design, build, test, analyze, evolve repetitive process, coupled with the adaptive laboratory evolution, where we allow the cells to utilize certain substrates and give them a kind of a situation where they are they are compelled or forced towards kind of an evolution. And that is identified could be a high caliber tool for this approach, for understanding the complexity of the system. Another approach is the technological revolution. Technological revolution with respect to the analytics, the sequencing tools, and the informatic tools. And lastly, the remediation phenomenon must not be considered only as a removal of the pollutant from the environment, but must be considered to be a microbe-driven trash to treasure methodology. So that will give the economic viability and sustainability of the entire process. Because most of the cases, the bioremediation research is constrained by the industrial interest, because the revenue generation is one of the major constraints, particularly in countries like India, where we need to look into the cost effectiveness of the process. So converting the natural remediation ability of the organism towards addition of value to the corporate or the company or to the state is to be one of the major phenomena. So with this, I end my presentation today, and I thank you all for your kind patience.
Amit sir, I think your mic is off.
So thank you, Professor Pinaki sir, for such a wonderful talk. Really interesting. We really enjoy listening your talk, and many people have got what Professor sir is telling about the metabolic engineering, how you can use systems biology, metabolic engineering, and metagenomics to actually do bioremediation, which is very important topic in today, because you have to do bioremediation process to save our environment. So, Professor Sir, we have some quick question from the student. So, one Alok Samal is asking that is there any established microbial degradation pathway for arsenic removal in commercial level?
Okay. So first of all, arsenic cannot be removed completely, because arsenic is not an organic substance which you can oxidize and break down completely to carbon dioxide, something like this. So arsenic can be converted from one oxidation state to other, like either arsenic 3 to arsenic 5 or arsenic 5 to arsenic 3. So oxidation or reduction reactions. So there have been numerous experimental endeavor to achieve a very successful remediation strategies, but considering the application scenario, like arsenic is a problem with the groundwater mostly. So to come to your answer, this is still in the research level, that conversion or removal of arsenic from groundwater and from drinking water is still in the research level. We are yet to achieve such level where the water treated by microbiological processes for arsenic is available for drinking water, because there are other health related issues which must be addressed, because arsenic is not to be converted to any other form, but it will remain as arsenic only. So you can only limit its solubility, or you can limit or convert its oxidation state. But research is going on that, and I hope that within a decade or so we'll be seeing some breakthrough.
Okay, next question is how much is the microbial bioremediation helpful when it comes to rice straw treatment, as it emits carbon dioxide in larger amount when burnt?
Mm-hmm. So Professor Ghosh, can you repeat the question?
So how much is the microbial bioremediation helpful? So whether microbial bioremediation is helpful for rice straw treatment. So as it emits rice straw.
Okay, rice straw. Okay.
Because it produces carbon dioxide when it burnt.
Okay, thank you. So when you I have answered to the question in a different way, that why do you want rice straw to be converted to carbon dioxide? So rice straw can be metabolized, or the substances present in the rice straw can be utilized by microorganisms and converted to some valuable product, metabolites. Simple, your ethanol, methanol, butanol, or some kind of stuff like which is useful for bioenergy or for certain other purposes. So must be looked into instead of only carbon dioxide. So ideally, it is not desirable that the lignocellulosic biomass like the rice straw be converted to carbon dioxide. You must not, because that's a huge resource. You should conserve that resource and energy and convert into some kind of useful biomolecules, like the different metabolites through fermentation and other reactions.
Correct. So next question is Shikha Thakur. She is asking, can you comment on using microbial genome to sequester more and more recalcitrant pollutant and degrade and eventually get rid of it from the environment?
Yes. So that is become one of the very important and one of the, what do you call that, breakthrough. Because only when the genome sequences are available, the sequences are compared through different platforms, the bioinformatic platforms, to identify the genes and predict the pathways. So even for the unknown organism also, we can use the phylogenetic neighbors to identify the possible reaction pathway. And they are found to be very useful for finding out suitable organism and their metabolic pathways.
So the next question is about using bioremediation, using microbe. You are doing the bioremediation, and then what value-added product can be generated while removing pollutant like Vitex compound and other.
So one of the, there are a couple of compounds or maybe a list of compounds you can generate. You can generate lipid, you can generate methane, you can generate methanol. It has to be decided by you that what you want to produce. What you want to produce. If you want to produce methane, you need to set up your organismic selection to your experimental selection, set up everything. If you want to produce lipids, the experimental and the organism selection should be different. So you can select any one of these metabolites or more than one metabolites which are useful industrially.
Okay, there are no more questions. I would like to thank Professor Pinaki sir for such an interesting talk, which actually motivated a lot of people now. And we will really thank you for valuable talk and appreciate your time. Thank you,
Thank
sir.
you. Thank you. Thank you, Amit. Thank you. Thank you, everyone, for your patience hearing and the questions. Okay. Thank you.
Thank you. Bye.
Bye.
Hello. So our next lecture is by Dr. Amit Ghosh, who is an assistant professor at School of Energy Science and Engineering, IIT Kharagpur. He had earned his PhD degree from Indian Institute of Science, Bangalore, and has worked as postdoctoral research associate in the University of Illinois under the mentorship of Dr. Human Zhao and Professor Nathan Price, as well as at the Joint Bioenergy Institute, Lawrence Berkeley National Laboratory in California, USA, under the guidance of Professor J.D. Keesley. His research interest includes three major areas: metabolic engineering in yeast to create designer strains for the bioproduction of value-added chemicals, understanding the molecular mechanism of various biological processes in the domain of carbohydrate as well as protein chemistry using molecular dynamic simulation, and the development of models to understand the interactions of microbial communities from different natural consortia. His talk today is on CRISPR-Cas9 tool to engineer yeast for production of industrially important biochemicals or metabolites using biofuels. I request him to kindly start this session. Sir, please.
Thank you, Manali, for the introduction. Today we will discuss about the CRISPR tool. Is it audible?
Yes, sir.
So we'll be I'll discuss the CRISPR tool to engineer yeast for production of industrially important biochemical metabolite, including biofuels. So as you know, that most of our product today are derived from petroleum. That is the raw petroleum, which is derived from the underground, and then it is processed in petroleum refinery, and then what you get gasoline, which is used for driving car and vehicle, and also this raw petroleum used for producing a lot of chemicals, and then drugs and so on. Most of the chemical which are actually available today are derived from petroleum resources. The more you are dependent on petroleum resources, then more amount of carbons are actually added to the atmosphere, because the fuel you burn in the car is actually petroleum derived, and you are actually releasing the carbon which is stored underground and releasing in the atmosphere. So this is going to be a huge problem if you keep on dependent on the raw petroleum. So if you use a biofuel or any renewable product, any chemical which is produced in a renewable way, then what happened? If you use biofuel for running your car, then the carbon released from burning the biofuel will release the carbon dioxide, and this carbon dioxide will be absorbed by the plant. And again, you are using the plant to generate the biofuel. So there is a carbon neutrality where you see that the carbon dioxide are not actually added It is just recycling from one region to the other, and you are not having any extra carbon in the atmosphere. So by using biofuel is actually helping that you are not adding any extra carbon in the atmosphere. So these are the three types of biofuel which exist today, like first generation, second generation, and third generation biofuel. The first generation biofuel is basically mostly using food, like sugarcane or corn that is used for production of the bioethanol. Right now, world produces around 100 billion liters of bioethanol. Mostly it is produced in USA and Brazil. 90% of the bioethanol produced in USA and Brazil. USA produce more than 60% of bioethanol using the corn, sugar, corn, and it's a first generation biofuel. Then comes the second generation biofuel, where you use the non-food part of the plant and to generate bioethanol or any other product. And the third generation biofuel is basically deal with algae, which actually uses carbon dioxide and doesn't require much land, agricultural land. So our country is actually where we have very less land, so we can actually implement the algal biofuel or fuel from photosynthetic microorganisms. So here I come the route for production of bioethanol or for the production of bioproduct, where you can see the sunlight energy is absorbed in the plant, and this plant is deconstructed into monomer sugar, and then this deconstruction process is involving different enzyme, acid, and so on. You get the monomer sugar. The microbe actually consume this monomer sugar because this is a plant-derived sugar. So the microbes use this sugar to produce different kind of product. So this is the biorefinery we have, where the uh, main part of the biorefinery is the microorganism. You can see the microorganisms is actually involved in converting the monomer sugar into different compound. And then the strategy over here is to actually engineer this microbial cell to produce the desired product. The whole intention is to actually how you can actually engineer the microbe. So this is on the top. You can see the microbial cell, and then how you can use different tool to engineer the cell so that you can have a desired product. So here are the many professors have told about the design, build, test, learn technique. So for the design, you actually choose different metabolic pathway, and then you build the metabolic pathway in vivo inside the cell using different tool like CRISPR-Cas9, and then you assembly different part, and then you try to test the path. So how do you test the path is basically measuring the compound in a GC or any other high throughput technique, and then you try to see whether the production is you're reaching your target or not. So similarly, you can do metabolic flux analysis, what Professor Arzul told, how you can do the metabolic flux analysis to estimate the carbon going through different pathways. And if you think that you have to further modify, then again you go to design, and in this way you go in this loop until and unless you get a desired product at a desired level. So far the renewable product which are obtained using metabolic engineering is one of the product which came in the market is the artemisinin, which was made by J. Kisling with an estimated cost of $50 million, and the time for which the project ran was 10 years. So you can think that amount of effort and amount of time needed to actually synthesize one product ultimately going to the market. And this product was made the artemisinin, it was the Nobel Prize in 2016. Nobel Prize was given in artemisinin, where the artemisinin is one of the important drug for malaria, and this artemisinin is produced mostly in China from a plant, and this artemisinin was synthesized in yeast, and the genes which are actually present in the plant are actually inserted in the yeast microorganism, and this product was made. Now, because of the genomics, you don't need the plant. So genes are not actually extracted from the plant. You don't need the plant because the plant is in a different country. But what you do is collect the genome sequence from the PubMed and the different databases, and you synthesize that gene and you make the pathway and insert it in the chromosome so that it can synthesize that compound. In this way, you can actually make a lot of compound which are actually useful for our life. Similarly, one more compound, that is PDO13 propanidol, which was also made by DuPont, which with an estimated cost of $130 million. And it took almost 15 years to actually make this product on the commercial scale. There are many other products which are actually on process and under development, and and the metabolic engineering plays a huge role actually to make this compound available. So some of the tools we will explain how you can actually engineer using different tools. So one of the tool which emerges actually the CRISPR-Cas9 tool, which was developed in bacteria first, and then it slowly it is available in many organisms, almost all organisms. And the benefit of CRISPR-Cas9 is limitless. There's an enormous amount of application which you can utilize the CRISPR-Cas9 tool to engineer the cell to serve your purpose. So in the CRISPR-Cas9 tool, I will go through some basic idea, like many of you know already, but I'll just go through if in any case somebody don't know. What you have is basically in a CRISPR system, the two things are required. One is the CRISPR, the DNA sequence, and the prokaryotic DNA sequence, and that is derived from the viral DNA. So the CRISPR system was first detected in the bacteria, where they want to, where these bacteria are involved in defensing from different virus. So this is used to detect and destroy the DNA present in the viruses. And this method is actually copied from the bacterial system and is implemented in many organisms. Almost all the organisms right now we have the CRISPR system. So it has the CRISPR, the clustered regularly short sequences, and then we have the CRISPR associated protein, that is Cas protein. These two things are required for building the CRISPR system. So here you can see these are the three different methods: the adaptation, expression, and interference, where we get the DNA cleavage. Whenever the virus attack any organism like bacteria, then if it can cleave the DNA of the virus, then the cell will survive. Otherwise, the cell will die because of the virus. So if the indigenous endonuclease is there, which can actually kill the virus, then the part of the virus genome is integrated in the host genome. And whenever the virus attack again, then it generated the pcRNA. The pcRNA is generated based on the viral genome, and then the Cas9 protein is also generated. And these two act together and against the invading DNA, and then it cleaves the invading DNA. So these are the two things we have to remember, that we are whenever a virus attack happens, the Cas gene is transcribed, and also the CRISPR array is transcribed. The Cas gene produces Cas protein, and the CRISPR array generate the precursor CRISPR RNA, that is pcRNA. pcRNA molecule undergoes this maturation to yield the short cRNA and recruit endonuclease and direct them to target DNA. So these two things happen where you have the endonuclease and the guide RNA. Whenever the guide RNA binds, there is a conformation change in the Cas9 endonuclease. So as you can see, there are 5 domains which are present in the Cas system, Cas protein, and so the RECA domain is responsible for helicase activity, and then HNH domain is responsible for breakage of the bottom strand, and RUBC domain is responsible for the breakage of the top strand. And whenever the guide RNA comes in, and then there is a conformation change, and then the endonuclease is able to cut different DNA sequences. So in the CRISPR-Cas9 system in type 2 for yeast, what we have is basically the cRNA and tracer RNA. These two domains, when they bind together, because they have a complementary region. These two complementary regions, when they bind together, they form a duplex DNA, or in other way, they become a active guide RNA. So this guide RNA is very important because it detects the nucleotide sequence in the genome where you want to target. Suppose you want to target a particular region of the genome, like 20 nucleotide you design, and then you put inside the guide RNA. So the guide RNA, here you can see that the 20 nucleotide over here is actually detector of the genome. So if you design any 20 nucleotide which is unique to the chromosome, and then you target the genome, then you will see that it will bind to that particular region of the chromosome, and it will recruit the endonuclease, that is the Cas9 protein. The Cas9 protein will come and then it will clip the DNA. So this way the CRISPR system works for type 2 system. Now, once there is a breakage in the DNA, what will happen? The cell will die unless it is repaired. So the repair mechanism is very important. If there is no repair mechanism, the cell will die. And the repair mechanism happens in two ways. One is basically homology-based directed repair. So based on homology, it repairs. So on the right-hand side, you can see DSB. So in the DSB, you see that there is a breakage of the DNA, and then we have the donor DNA. So the donor DNA should have a overlap, sufficiently overlap, and then it joins the end and try to recover the damage. So if the damage is not recovered, then there is another way to cell to repair, that is known as a non-homologous end joining repair. So this is the non-homologous end joining where there is no donor DNA. Suppose you have running a transformation and you have the CRISPR plasmid and you have the donor DNA. Somehow the donor DNA is not available. Then what will happen that the cell will repair on its own. And how it repairs, basically it incorporates some mutation in the genome. And this is not desired in metabolic engineering. So what you on the left hand side, what you see, NHAG repair is not desired in metabolic engineering because cell is repairing on its own by incorporating mutation. And what you desire is basically homologous recombination, because here you can insert any gene or any deletion. You can go into detail further. So these are the CRISPR tools available in different yeast. Especially it has started in Saccharomyces cerevisiae in 2013, and then slowly it was developed in Pichia pastoris, Aurora lepiletica, K. marcinus, and we have CRISPR system for Candida albicans. So what you do, you basically design a Cas9 protein, which is actually can be expressed in that particular organism, and also you need the native promoter. So you should know the promoter. So these are the two things you take care when you design a new plasmid or the CRISPR-Cas system for a new organism. Even though it is available for Saccharomyces cerevisiae, it may not work in non-model yeast. So on the bottom, all the below Saccharomyces cerevisiae, these are all non-model yeast which people are using, like Professor Tang is using Aurora lepiletica. Many people are using Cliveri marcinus, where the CRISPR-Cas9 system is available. You can start using the Saccharomyces cerevisiae CRISPR system, but what you need is actually you have to remove the native promoter of Saccharomyces cerevisiae, because the promoters are very unique to the microorganism. It will not work. The Saccharomyces cerevisiae promoter will not work in any other yeast, even though they are on the same yeast, but it will not work simply because the promoter which are working for Saccharomyces cerevisiae, it may not work in other non-model yeast. So first thing, you have to replace the promoter, and you have to see whether the guide RNA Cas9 is working or not. Most of the time it works. Otherwise, you have to synthesize. You have to codon optimize for that particular organism so that you have a better expression. Still, if you can use Saccharomyces cerevisiae plasmid by replacing the promoter, you will see that the efficiency of the CRISPR plasmid may not be 100%. But to get 100% efficiency of the CRISPR system for non-model yeast, you have to actually replace the promoter. So these are the different approaches, CRISPR approaches available for genome editing. On the left-hand side, you can see genome engineering with Cas9 nuclease, where you can see you can clip the DNA based on the guide RNA. So the guide RNA will detect the region where you need to clip, and the Cas9 comes in, and it will recruit the Cas9 and it will clip the DNA. And based on the donor DNA, you can see that new DNA you can insert. So here you can see that the in cyan color, the new DNA comes in, and this is where you can insert the new pathway. Or you can incorporate a deletion. Suppose you just want to remove the gene, then you can remove the gene and you design the donor DNA in such a way that you get rid of the native gene. On the middle one, you can see that you can design the donor DNA in such a way that you can have a homology directed repair, and you can insert a new gene. And if you are not giving any donor DNA, then you will non-homologous end joining will be happening, where it will incorporate some mutation, which is not desired in metabolic engineering. And on the right hand side, in the block, you can see that we have dCas9. What is dCas9? dCas9 is basically dead Cas9. Basically, it will it will not cleave the DNA. It will just bind. So guide RNA will recruit this Cas9, and this is a dCas9. It will sit on the genome, and then it will not cleave. It will just sit and nicely sit there. So it will not cleave, but it will have an activator. So you can design the activator in your CRISPR-Cas system. It will bind the Cas9 and it try to regulate the particular gene. So you can design properly where you can upregulate or downregulate a gene based on a CRISPR-Cas system. Also, you can bind a GFP gene where you can actually see the visualization how the expressions are happening. So these are the available technique using CRISPR-Cas9 where you can delete a gene or insert a gene, or you can just upregulate or downregulate. So all these things you can do using a single CRISPR system. So this is another example where you can delete the gene using disruption, and then you can choose if you are providing the donor DNA with sufficient flanking region, then you will be able to remove the gene. And similarly, you can actually integrate a fragment of DNA using some homology at the end joining. So in this way, you can make assembly, DNA assembly also, and insert the entire assembly into the chromosome. So these are all actually, you can do it at the chromosome level. So industrially, this is what required in metabolic engineering. You do the all pathway construction inside the chromosome so that we don't have to construct again. So once you have done it, then it can be reused n number of times without any problem. So this is another example of multiplexing using CRISPR-Cas9 system where specifically in industry, they have less time, less manpower. What they do, they integrate many genes together in one transformation. So if you want to do integration of many gene in one transformation, then you have to do multiplexing. And in multiplexing, what you have is basically we have different guide RNA. So for one gene, we have one guide RNA. Suppose you want to insert 3 genes, then you have to have 3 guide RNA. And also you have to design 3 donor DNA as well. Because whenever there is a clip, you have to actually have a donor DNA to repair it. If it doesn't repair by donor DNA, then the mutation will happen, and that is not desirable in metabolic engineering. But you can only have one Cas9 enzyme in the cell. So this way you can transfer all this fragment and the gRNA into the cell. Then what will happen? All the 3 genes will be inserted at that particular locus, and you have the pathway ready in one transformation. Otherwise, you know that yeast for one transformation it takes 1 or 2 weeks to grow the cell and check it. So if you are using multiplexing, you can actually save a lot of time, which is very desirable, very much desirable in industry. So this is an example where meconic acid pathway, which consists of 11 genes over 6 DNA fragment, has been inserted into the Saccharomyces cerevisiae using CRISPR-Cas9 engineering. So here you can see using the multiplexing, they have inserted all the genes. That is 6 DNA fragments they have inserted. So out of 11 genes, you can have 2 genes per insertion. So when you have 2 genes per insertion, you make sure that the 2 promoters are different, because we have very limited promoter in yeast. That's why you have to integrate many fragments at a different locus so that there is no interaction between the promoter. So now we'll go into other tool which are available for targeting the genome for metabolic engineering. So for example, the genome scale 13C metabolic flux analysis. So Professor Tang, Professor Arzul and his student has already explained about the 13C MFA, but I will explain about the genome scale 13C MFA. Why genome scale 13C MFA? I will go into it. So this is the central dogma in biology, where you know the DNA, RNA, protein, and the reaction. Especially the DNA is transcribed into RNA, and then RNA is translated into protein, and then protein is catalyzing the reaction. So in the metabolic flux analysis, you operate in the reaction domain, where you make you understand how many reactions the genome have. Based on the reaction, you construct the network and try to estimate the fluxes. So these are the high throughput technique available like genomics, transcriptomics, proteomics, metabolomics, and fluxomics. But you already heard about genomics, transcriptomics, proteomics, and metabolomics, where some equipment is used to actually generate the data, and experimentally you may measure the DNA sequence and the mRNA level, protein level, and metabolites level. But fluxomics is another field which you cannot measure the flux directly from the equipment. Here you have to do some modeling, and then you can incorporate the experimental data and measure the fluxes. Because fluxes are measured in terms of number of molecule per unit time through a reaction, and the rate of a reaction that you can calculate, which is the most relevant phenotype in a cell, where you can, if you know the flux of a cell, of a reaction, then you will be able to estimate how much it is growing and how much the compound is produced. So this is a direct measurement that is the most relevant phenotype for measurement of any cell. So this is the flux profile which you can get from 13C MFA. So for doing 13C MFA, you need a metabolic network, and you need a genome scale. For genome scale, basically, if you want to do 13C MFA, you should know about the flux balance analysis. The flux balance analysis, you know, is where you construct the genome and the network, the biochemical network for all the reaction. From genome, you annotate the genome and then try to get all the reaction, and you tabulate all the reaction and form a stoichiometric matrix. And using this stoichiometric matrix, you actually optimize the biomass equation, that is the growth rate. So you maximize the growth rate, and then you get the fluxes by using the condition like the lower bound and upper bound of the fluxes. You apply the boundary condition so that you can measure the fluxes more accurately. So this is the strategy used for flux balance analysis, where you can measure the fluxes in silico. You don't need to do any experiment, but you need a metabolic model. But how can you use this metabolic model for actually 13C MFA? So this is the problem we address to how you can get the fluxes for all the reactions, because 13C MFA deal with only few hundred reactions. Not all the reactions are available where you can measure the fluxes. So these are the biochemical data where you can build the network using the available data. You can build the network, the reaction and the gene. You can collect from different database. And for example, the K-base is used for automatic reconstruction of metabolic network. Also, model seed is used for reconstruction of metabolic network, where you can start from the genome sequence and you can build the network very easily. So in the biochemical network, what you have is basically not only the reaction. I told about reaction, but what is more important in the biochemical network is the gene. Because if you are not able to connect the gene with the reaction, then you have a missing information. So any reaction in the model, you have to connect it with the gene. And the genes are actually can be in a Boolean logic. Like in the first case, you can see that there are four genes which are actually forming the four subunit in a protein, and this protein is catalyzing two reactions. And in third case, you can see that the three genes which are actually forming the three subunit, and this is catalyzing one reaction. This way, you can form a Boolean logic among the gene, and that is responsible for catalyzing some reaction. And this genotype to phenotype you can easily bring about by removing one gene, and you can measure the fluxes. That is the reaction, the flux going through a particular reaction. So this is the overall reconstruction procedure, where you start with the genome and then you annotate the genome and you find out how many reactions, you make mass and charge balance, and you construct the stoichiometric matrix. So these are the steps followed in metabolic reconstruction. So the metabolic reconstruction, where you have to identify how many reactions the model is having, and then you build the network. You build the network and make a stoichiometric matrix. So this is a very well-known method when you do the model reconstruction. So once you have the model ready, then what you do, you apply the flux balance analysis method, where you optimize the biomass equation. You maximize the biomass equation subject to your stoichiometry, that is steady state condition, S dot B equal to 0, and then you apply the boundary condition. That is, you give lower and upper bound for each and every reaction based on the data available or the thermodynamic data available. Suppose if the thermodynamic data says that particular reaction is irreversible, then you put lower bound as 0, like that. If there is no data available for a given thermodynamic data is not available, then you put the upper bound and lower bound as infinity, minus infinity to plus infinity. And then you apply the steady state condition. The steady state condition says that the concentration of the metabolite is not changing. So at a steady state, the concentration inside the cell is constant. So when you take a derivative of the concentration, it is zero. So that's why you become S dot B becomes zero. And then you solve this equation and you get the fluxes. So using this method, we recently reconstructed the genome scale metabolic network of industrial yeast like L. colubriae, which has a higher biomass and is a capillary negative, weakly capillary negative organism, which can be used for not only for bioethanol, but also it can be used for advanced biofuel or any other terpenoid based compound because it produces less ethanol. The model comprises of around 1235 reaction and then 1179 metabolite and 730 genes, and it has 8 components. So it uses, when we build the network, we actually used a already available well curated metabolic model of other yeast. Around 13 other yeast have been considered as a template for building the L. colubriae metabolic model. And we found that Saccharomyces cerevisiae actually contributed around 17% similarity in terms of number of reaction. So now 17% of the genes are actually coming from Saccharomyces cerevisiae, and the remaining you can see Candida tropically and then other viral politica, they contributed a lot in terms of the number of genes which are similar. Then we compare the gene lethality. Any model, if you build, the first thing you have to compare the gene lethality. In gene lethality, what you did is actually compare the growth and how many genes are comparing with in terms of lethality. You can compare with the experimental data. Now you see that L. colubriae, the number of experiments are very, very less. None of the genes have been mutated so far. So you build the model, but if you are not able to compare the data with the experiment, then the model is not validated. So for that purpose, what we did actually, we have done the homology search with Saccharomyces cerevisiae to see how many genes are similar. Then we found that around 700 genes which are similar to Saccharomyces cerevisiae, that we compared the knockout. Here you can see that around 719 genes are actually matching under 2 negative category, and then 24 genes are matching in no growth category. So they are compared, and we got a false positive rate around 19 genes which are actually false positive, and then 78 genes which are false negative. But as we go from rich media to minimal media, you can see that the false positive actually increase from 19 to 65. This is because that there are a lot of duplicate genes in Saccharomyces cerevisiae, which actually optimize the lethality when genes are removed one at a time. But in L. colubriae, what you see that there are very less number of duplicate genes. When you check the lethality, you find that some of the genes are actually lethal. So the lethality in terms of number of lethals are more when you compare with Saccharomyces cerevisiae. Similarly, we see the gene lethality in galactose media, ethanol media, and all of them compared very well. So then now I come to 13C steady state metabolic flux analysis, which has already been explained in previous lecture. I will not go into detail. I'll just focus on how you can actually extend the 13C metabolic flux analysis at a genome scale level. So in 13C metabolic flux analysis, you start the experiment with a level glucose, like you use the first carbon level glucose and then allow the cell to grow in a glucose. And when most of the metabolites are level, you take out the sample from the exponential phase and you measure the leveling in a GC-mass. And using GC-mass, you get the probability distribution or the distribution of the carbon which are 0 carbon level, first carbon level, 2 carbon level, 3 carbon level, like that you get a profile. And this you can feed into 13C MFA analysis to get the experimentally derived fluxes. So this is the algorithm, like we start with a initial flux distribution and you estimate the leveling theoretically, and then you compare it with the experimental data. And once you get a very good match with the experimental data, you stop the calculation. Otherwise, it is a recursive procedure. You keep on calculating the amino acid labeling theoretically, and then compared with the experimental data, means the GC-mass labeling you compare. And this way you can calculate the fluxes based on experiment. So this is the most suitable method for calculating the fluxes at the experimental level. Here you can see there is no biomass optimization. So you don't use any biomass optimization, you just have the error function where it minimizes the error function. This is the error function. You can see the experimentally derived metabolite labeling and the theoretically obtained metabolic labeling based on the flux distribution. So when you calculate the metabolite labeling theoretically, you actually use the flux profile. So given a flux profile, you calculate the metabolite labeling, and then you compare it with the experimental labeling. And when the error function is minimum, then you stop the calculation, and then you say that this is the most correct flux or the experimentally derived flux for a given network. So this we want to extend at the genome scale level, because 13CMFA only deal with only a few hundred reactions. So how do you can do the genome scale 13CMFA? This is known as 2-scale 13CMFA, which we published earlier, where it uses the genome scale stoichiometry. So in this 13CMFA, we feed the genome scale metabolic network. So generally in 13CMFA, you choose particular reaction, like you choose few hundred reaction which you are interested in, and those 100 reactions are only the central metabolic network present in this central metabolic network. And in 2-scale 13CMFA, you just feed the genome scale metabolic network directly. You don't have to modify the network. Directly you can feed. And then you feed the extracellular fluxes, which you measure experimentally, and the 13C level metabolite data. So these three things are required in 2-scale 13CMFA. In 2-scale 13CMFA, you feed the genome scale model so that you can get fluxes for all the reaction. Why you are interested in all the reaction, I'll come to it. So this is the method where you have the central metabolic network bounded in a blue, in a green rectangle. In the green rectangle, you see this central metabolic network, and outside you see all other network metabolic reaction which are not part of the central metabolic network. So in the central metabolic network, you measure the fluxes by tracking the carbon in the central metabolic network, but for remaining other reaction outside the central metabolic network, you are not actually tracking the carbon leveling. So those can be treated as a metabolite balances. So whole genome is actually, you run FBA, only the central metabolic network, you run the 13CMFA. So this way you can calculate the fluxes for all the reaction, because the central metabolic network is actually deciding how much fluxes it will go into remaining part of the network. So you don't worry about the carbon tracking measurement on the remaining part of the network. You are just interested in the central metabolic network, but you would be able to calculate the fluxes for all other reaction using by combining these two techniques, the FBA I have explained just now, FBA, and then 13CMFA, which I have already explained in the previous lecture, so I have not gone into detail. But if you combine these two things, then you can actually able to measure the fluxes for all reaction in one go. So using this technique, we actually have chosen the L-Klubery flux for flux measurement. Since we have built the metabolic network for L-Klubery, we measure the fluxes for L-Klubery, where you use the genome scale metabolic network for L-Klubery, and using the metabolic model, we have calculated the genome scale level flux for entire metabolic network. So once you have the metabolic fluxes for all the reaction, the benefit is that you can do a balancing kind of plot, which is shown at the bottom. You can see that how much acetyl coenzyme is used by different reaction. Here you can see that ACS is producing a maximum amount of acetyl coenzyme, and then it is used by so many reactions. So how much flux is going into different reactions, that you can calculate using this technique. And then you can do CRISPR-Cas9 or metabolic engineering, where you can modify the gene accordingly. So I will give one example where we did exactly the same, where we use the flux analysis data, which is at the genome scale level, and we found out that which are the reactions which are actually consuming most of the acetyl coenzyme, and we have targeted those reaction for modification. So I will come to the last part of my talk, where we use the genome engineering technique, that is CRISPR-Cas9, for production of biofuel. So I have taken the free fatty acid overproduction pathway, where you can see that acetyl coenzyme A is converted into fatty acyl coenzyme A using these three enzymes, that is ACC, FAS1, FAS2. So if you upregulate these three enzymes, then you will see that the fatty acyl coenzyme A increasing, and then you can get many products. Like free fatty acid you can get by upregulating TSA, and then you can get fatty alcohol by upregulate or inserting FAR gene, and then you can get FAE by inserting ATFA gene, another heterologous gene you can insert, and also you can increase TAG by upregulating DGAN. So this is the metabolic engineering strategy we have implemented while we applied the native. So you don't have to have heterologous gene, just you have to upregulate the native gene. So how you upregulate the native gene? You just replace the native promoter, because the native promoter for this FAS1, FAS2, SEC1 is not actually a constitutive promoter. So for constitutive promoter, you which is available already for Saccharomyces cerevisiae like F1, that you can integrate in the chromosome. So this need to be done at the chromosome level when you upregulate. So CRISPR-Cas9 technology can be implemented here, where we replace the native promoter with a constitutive promoter TF1, and we have upregulated FAS1, FAS2, SCC, and we got a significant improvement in free fatty acid production. So these are the two flux phase. You can see the glycolytic phase and the respiratory phase. So yeast uses glycolytic phase when glucose is available in the environment or in the media. So whenever the glucose is present, then it is using the glycolytic phase, that is glucose is metabolized, and then you see the ethanol production. So this is very well known pathway. And then if the glucose is over, then what will happen? It will consume all the ethanol present in the media. So it will consume ethanol. You can see the pathway for ethanol. It will go to the TCA cycle, and then enter the metabolism glycolysis pathway. So in these two phase, like glycolytic phase and respiratory phase, we have measured the fluxes and at two different time point, and also we have get the genome scale metabolic flux profile. So this is experimentally derived 13C NFA, which is done at the genome scale level, where we got the fluxes for all the reaction. And why all the reactions are important? Because you want to know where this acetyl coenzyme is consumed. And for that, we also have done the flux measurement at a respiratory phase, when it is consuming ethanol. You can see that in the yellow curve, the ethanol is consumed slowly. It is very slow. It takes almost 90 hours to actually consume all ethanol present in the media. And at a particular time point, we took the sample and measured the fluxes, and to know how the flux profile changes when you go from glycolytic phase to the respiratory phase. So here you can see the flux profile when it goes on ethanol. You can see at the bottom that ethanol is entering the cell, and then it is going into TCA cycle, and this is where, and also it is some part of the flux is going into glycolysis as well. So this way you can actually get the flux profile at when the cell is growing on ethanol. So now we have two flux profile. One is growing in glucose, and another is growing on ethanol. And now the comparison we want to do. So now the comparison is that the acetyl coenzyme is consumed at two different phases, in the glycolytic phase and the respiratory phase. And the comparison says that the malate synthase, which is present in the TCA cycle, is responsible for consumption of a huge amount of acetyl coenzyme. The flux from malate synthase is more when it goes on ethanol. So we want to engineer the malate synthase based on the flux inference or 13C NFA data. So we have actually downregulated the malate synthase, which is shown in green color, and then we have added ACL, ATP citrate ligase, which is basically transferring the citrate, metabolic citrate, from TCA cycle to acetyl coenzyme. So we get more amount of acetyl coenzyme in the cell by inserting this gene in the chromosome. And also we actually remove the competing pathway, which is actually taking away the carbon from the central metabolism. So using these two this metabolic engineering strategy, we almost got double production of free fatty acid in Saccharomyces cerevisiae. And this is completely done on flux analysis data. The flux analysis data which help us to actually target this gene. Then we worked on the fatty alcohol production in Saccharomyces cerevisiae, where we improved the production of fatty alcohol to 1.2 gram per liter in flux and then 6 gram per liter in fed batch. So for fatty alcohol, you see that we need one gene. So all our netting gene, only gene you insert is basically FAR gene, that is fatty alcohol reductase. And if you have this gene expressed inside the cell, you will be able to get the fatty alcohol. Again, we have done the metabolic flux analysis using 30CFA at the genome scale level. Since we have done the flux analysis at the genome scale level, we are able to see which are the reaction involved in consumption of NADPH, because we know that fatty acid production depend on NADPH molecule, the level, the pool of NADPH molecule inside the cell. If the pool of NADPH inside the cell decreases, then you will not be able to actually produce enough amount of free fatty acid. That has already been explained by Professor Tang, where the energy molecule ATP he actually improved by adding one more gene. So here we have not added any new gene. What we have done, we have removed some of the genes which are consuming a lot of NADPH based on flux profile. So here you can see that the pathway which is involved in free fatty acid shown over here, the free fatty acid and fatty aldehyde, that is HFD1 and ADACs has been removed because it is it's a redundant pathway or the competing pathway. The competing pathway which is carrying carbon from the desired product, we have removed that, and also we have removed the glutamate GDH1, that is glutamate dehydrogenase, because it is consuming NADPH. So this is the third gene we have removed to improve the production of fatty alcohol, and also we have removed other competing pathway like Tag biosynthesis pathway. And for FAR gene, which we have taken for mass muscular organism, where we synthesize the FAR gene and inserted in the chromosome. Now it is here we have not expressed any any plasmid inside the cell. This is completely at the genome level and using CRISPR-Cas9. So, since the plasmid expressions are better in terms of metabolic pathway formation, if you want to produce some compound, then is better you can express plasmid, because plasmid has a high copy plasmid and you have more enzyme inside the cell. But when you integrate in the chromosome, the expression of that particular enzyme may not be that equivalent to plasmid. So to overcome that plasmid express pathway, what we did is actually integrating the FAR gene at a different locus. Here you can see that FAR gene is actually, we have inserted first FAR, and then there is a production of very less. Then we have inserted the second FAR. Then you can see that they are improved by almost 20-fold. Then we have inserted the third FAR, the gene. Then we see that there is not much improvement. So then that is why we stop. So it may not, it is not true that if you keep on integrating the same copy of the gene, you may not get increase in product directly. The increase in is not directly proportional. So here you can see the third FAR. When you inserted the third FAR, the production has actually diminished. So when you have the second FAR, we have much more production compared to the third FAR. And then we remove the competing pathway and also remove the other gene, and also we have kept the other one more copy of SEC1. So as many number of copy of SEC1, the metabolic pathway which we are constantly can be inserted in the chromosome, and this is purely at the genome level. So to make it equivalent to the plasmid level pathway, you insert many copies of the plasmid of the gene, so that you get better production. In this way, you actually improve the production of fatty alcohol to a greater extent. And this is so far the highest producer of fatty alcohol so far. So finally, in summary, what you have seen that the CRISPR-Cas9 technology, how you can use CRISPR-Cas9 technology for genome engineering, how you can engineer the cell easily without a marker. It's a marker-free genome engineering. So, so you can integrate as many number of genes as you want. For example, fatty alcohol production, we have integrated so many number of genes. Because we have applied CRISPR-Cas9 technology. Then we discuss about the genome scale metabolic network of L. Klubery, how we can construct the genome scale metabolic network And then also we discuss about the free fatty acid production, where we integrated the ATP citrate lyase, and then we downregulated the malate synthase for the production of free fatty acid production. And finally, we also use 13C genome scale 13C metabolic flux analysis for the production of fatty alcohol and also for production of free fatty acid. So this is my group. When we started the lab, we have only 5 people working, 6 people working. Now we have more people working in metabolic engineering. Hope will make much more progress in metabolic engineering. Thank you all. Thank you for listening. Thank you for your patience. And this is our School of Energy Science building. You are most welcome to visit our school.
Thank you very much, sir, for your very informative talk. The session for question and answer is open, and we have a question from Mr. Shah Bhavik. Is it possible to use CRISPR-Cas9 for Pseudomonas? What factors are to be considered for the same?
So you have to check. I'm not working on Pseudomonas, but you can check online whether the CRISPR plasmid is available or not. It is very easily done. If it is available, you can just buy it. It is whenever a plasmid is published, it is kept in Arzul. You can order from Arzul and get the plasmid at your lab. And if the plasmid is not available, then you have to make the plasmid. Then what you do, you look for the closest organism. So which is the closest organism to Pseudomonas? Then you select that organism and then get that plasmid available in your lab. And then you replace the promoter, or replace the promoter of the other host and replace it with Pseudomonas promoter. And you would be able to start work on this new CRISPR system. But ideally, if it is available, it is better, because from scratch, if you start constructing the CRISPR system for Saccharomyces cerevisiae, we actually started building the CRISPR system for Saccharomyces cerevisiae. It almost took 1.5 years. So it's better you use a already available CRISPR system for this organism. I hope most of the organisms right now it is available. So you just check in Arzul and try to order it. I hope it will go.
Thank you, sir. So there is another question from Mr. Sharath S. Considering redox potential important for the product formation, will MFA consider the redox reaction also in determining the crucial genes with respect to our objective function?
Redox reaction is, you are doing S dot V, right? S dot V is taking care of that. So all other things, the mass balance, charge balance, everything is maintained in the network. So we apply all theory of optimization, energy conservation, and everything is applied. So there is no issue on that.
Are there any more questions? I think then we can conclude your session, sir. Thank you very much again for your very nice talk, and we'll move on to the next speaker. So our final speaker for the morning session is Mr. Piyush Nanda, who is a PhD scholar at Harvard University, USA. Mr. Nanda recently earned his BTech MTech dual degree in biotechnology from IIT Kharagpur. He has been a recipient of the Honda Young Engineers and Scientists Award in 2018 and the Khorana Scholarship in 2019. In addition, he has 2 patents on carbon capture and fermentation technology and Kalium non-polluting engine. His talk is on taming the microbes for bioproduction, a systems-scale synthetic biology approach. Mr. Nanda, you can start, please.
Am I audible?
Yes.
All right. So my topic today, as already Manali mentioned, is Taming the Microbes for Bioproduction, a system-scale synthetic biology approach. I am Piyush Nanda. I'm a graduate student doing my PhD in the Biological and Biomedical Sciences program at the Harvard University. So just to give a brief intro of who am I and what am I doing now. So I completed my dual degree, BTech and MTech from IIT Kharagpur in biotechnology and biochemical engineering amidst the global pandemic. During my time in IIT Kharagpur, I did my internships at the Great Lakes Bioenergy Research Center, the University of Tokyo, and Harvard University. Currently, I'm a G1 student. That's like the first year of the graduate studies in Harvard University in Biological Biomedical Sciences program. I am primarily interested in metabolic systems biology and synthetic biology for production of fuels and chemicals. Some of my hobbies are reading, guitar, cooking, and now I'm interested in something that's like tracking social behavior ants. I would be happy to talk about this later sometime. So this is an image of the apparatus I'm using, and I'm just doing it in my home. So to just begin about the topic that I have proposed, let us first start with how did metabolism originate? In this way, we'll be able to better capture as to what are the limitations currently in producing biofuels at a large scale and what are the current challenges with us. So these are all the hypotheses. We don't know. We were not there. It happened like billions of years ago. So these are all hypotheses. It's also very difficult to experimentally validate it because we don't want to start the evolution once again. So one of them is primordial soup theory that was proposed by Miller and Urey. It's a very elegant, simple experiment, although a lot of assumptions are put into this. So you just take four gases, that is methane, like water vapor, ammonia, and hydrogen. These are the four gases which were prospectively present in the prebiotic atmosphere. You circulate it through a system in which you are sparking them in between. So you can also heat them instead of sparking. Then you are taking it through water. So the water here is telling you about the ocean that were present at that time, and the gas is talking about the atmosphere that was present that time, and the spark is talking about the lightning and other things that were present that time. And when Miller Urey did this experiment and they repeated it for several cycles, they saw that they were getting almost 20 amino acids which are required for the survival of life. And that was quite interesting because we created 20 organic molecules out of 4 inorganic molecules. And it was pretty exciting. And this was like very widespread. People appreciated it a lot. Like, but there were a lot of assumptions. Like we don't know whether spark or lightning was there in the prebiotic atmosphere, right? And like that's why like it's like whenever I'm telling this, take this with a grain of salt. We are not very much sure about how this happened. But in 2007, when so Miller Urey did this experiment somewhere around mid 1950s. But then later on in 2007, the vial in which they stored their final samples were reopened and they were subjected to GC mass and LCMS. They found the mixture actually contained more than 20 amino acids. So like this might be one of the possible ways how life evolved. And then there are like alternative hypotheses they call as like RNA world, because you know like RNA also has catalytic activity apart from storing information. So it can both store information, which is crucial for the propagation of light and for life, and also do enzymatic reactions. So that's why RNA might be one of the starting material for the synthesis of life. And then there is a lipid hypothesis, or what we call as the lipid world. So what basically happened, you had like reactions happening in, let's say, oceans, you had reactions happening all over. But you know, like people who know about chemical engineering, they know if you are doing reactions in a very dilute atmosphere, it's simple law of mass action, which says that the reaction rates will drop because your like interacting molecules are dilute. But what happens if there's a lipid missile or there's a lipid droplet inside which your enzymes or your RNA is localized and they start doing the reaction? The good thing about it is you get an isolated atmosphere and you increase the concentration several folds. That's why the reaction rates increased. Now what happened through the time of evolution, there were several RNAs, several proteins which were formed. They got inside these lipid vesicles and they started doing some very simple reaction. Let's say they just took carbon dioxide and converted that to formaldehyde or converted that to methanol. And slowly evolution took place and we got like even more complex set of reactions. And at some point we started making lipids. So this is a very simple concept of surface tension physics. If you have like a lipid droplet, and you have like enzymes immobilized inside it, then what you are basically trying to do is to put enzymes inside the lipid vesicle. And if the enzymes themselves start producing these lipid molecules, then the lipid molecules are going to get stuck in the lipid membrane, right? And the lipid membrane is going to grow as the enzymes does the synthesis from, let's say, carbon dioxide or glucose. Then you reach a point in which your surface tension energy is going out of control. So now, like to maintain the surface energy, to maintain the entropy and the enthalpy here, you need to break apart the bubble. So here I have a diagram in which you have like a bubble and it splits into several bubbles when the surface tension exceeds. And this is how like the origin of cell division started. So it's all based on like how surface tension. These are hypotheses. We don't know if this was the best way, but this is one of the most potential alternatives. So then what happened? We started getting all this complex reaction. Then Darwinian evolution played its role. We had like survival of the fittest coming into picture. And then what we have today is a super complex network of so many metabolites and so many reactions that manifest the whole metabolism currently. So given that the metabolism is so complex and it's such an important part of life, because whatever the cells are, they are all due to the metabolism, right? Because metabolism just takes simple sources. If you have ever grown E. coli or yeast, you just give like a clear liquid, you put a single or a couple of cells or like some amount of cells, and they completely take over that media, converting them into super intelligent molecular machines. So that's why they require severe amount of regulation. So that's why we come to the next part, that is the metabolic regulation. Yeah. So to just begin this topic, I'd just like to mention about this simple pathway known as galactose induction pathway, which many people use for inducing gene expression in yeast cells. And the beauty about this pathway, it's like a simple pathway where the cell has to decide whether it has to take galactose or glucose. If you have both galactose and glucose, it needs to take a decision, right? What would be the best given, like the best benefit that it can get by expressing the enzymes and all. You can see in this pathway as simple as this, there are so many regulatory bottlenecks here. Like you have like MIG1P, which represses GAL4P, URS, and this is part of my rotation project now. I would like to speak a lot about it, but like I don't have like much data to talk. That's why it's just like a very beautiful paper in PNAS from the lab that I'm working on in Michael Springer's lab. And they were able to show the yeast galactose gene expression, like the GAL1, which is the first enzyme that catalyzes the conversion of galactose to galactose 1 phosphate, actually responds to the ratio of galactose and glucose. I mean, it's quite interesting, right? How does a small cell, which does not even have like a proper neuron in it, like it does not have any nervous system, how is it responding to a ratio instead of, I understand that it can respond to the amount of glucose, but it's very difficult to digest that it responds to a ratio, but that happens. And that's like the beauty of these systems. And then so now if you look at the gene expression in eukaryotes, it's not as simple as gene expression in E. coli or in bacteria. So you have this complex chromatin structure, right? And there are a lot of enzymes which are involved in chromatin regulation. So you have enzymes which goes and binds to chromatin, like you have like a complex way in which your DNA is wound around histones. The histone needs to be acetylated, and there are a lot of chromatin modification which takes place, which opens up the DNA sequence so that it is accessible by RNA polymerase and other things. So that is also another level of regulation. And in this beautiful paper, another beautiful paper in PNAS in the same year, Burt and Burton, they showed that many of the TCA cycle metabolites like acetyl-CoA actually regulates chromatin like accessibility. And this becomes particularly interesting because you see there's a reciprocal regulation of metabolism. You have like enzymes which are making the chromatin more accessible so that the gene expression can take place, the metabolism can kickstart, but again, the metabolism itself is giving a feedback to the chromatin. So this is like what I'm trying to explain is like the level of complexity that is there in cells currently. So we definitely have a lot of questions in our mind how to explore these systems, but the two questions that I'm particularly more interested about is how do we determine this set of all interactions? I mean, if you just take into account, there are like 6,000 genes, there are like thousands of proteins, there are like many reactions, many metabolites. If you just take like the combinatorial space of the whole system, it's huge, it's astronomical, it may be beyond the number of stars in our universe. So that's why, how do we really find out like what are the set of interactions? So this was one of the questions I was very much intrigued about during my time at IIT KGP. And the second question was how much synthetic should be the cell for the optimal function? I mean, there are concepts like in synthetic biology known as orthogonal circuit, in which you just make a circuit inside a cell which is completely independent of cell. So it does not interfere with the cellular growth. But again, we don't want to miss out the beautiful things that the cell does, right? We definitely want to get rid of those circuits in the cell which actually impede the production of our chemical of interest. But we also want to keep those part of the circuits preserved, which are actually playing a good role, right? So that's why how much synthetic should be a cell? I mean, of course, we can make a completely synthetic cell out of scratch, but that is not possible as of now because we do not understand biological systems that much. So that is another important question. How much synthetic should be the cell for its optimal function? And then there are other interesting things that are related to metabolism. That is, what is the behavior of microbes in bulk? I mean, we are all very much familiar about the fact that when we are alone and when we are, let's say, in a crowd or with your friends, you definitely behave differently, right? So microbes also do the same. Like, and there are three most interesting phenomena which I would like to discuss. The first is genetic noise. I mean, all these cells you see in this picture are having the same genome, they are having the same plasmid if you have put one, and everything is same. It's just that you see a variability in the gene expression. And this is what we call as genetic noise. It has been studied a lot. It has been implicated in the development of antibiotic resistance and like millions of other things. So this is genetic noise. When you take like all cells together, even they have the same chromosome, they have the same gene, but they have like different level of expression. Then there is very interesting paper in eLife, which was published, that is known as the spatial organization of metabolism. I mean, all of us who have worked with yeast, we know like when we grow yeast cells, like when we plate yeast on a plate, you see colonies. But now you can ask a very interesting question as to, is that all the cells in that colony uniform? I mean, like, are all the cells behaving the same? No, this was like proved now. Actually, this paper was published in 2020. Sorry for putting it 2008. It was recently published in 2nd of September, and it's a lab from Sunil Lakshman lab from NCBS. They are doing like wonderful work on spatial organization of metabolism. And what they showed is, you know, like there's aspartate, which can be used as a nitrogen source. We already know like aspartate is a very important amino acid and can be used as a nitrogen source. But these people, they showed that it can also be used as a carbon source. And what happens when you are trying to grow a colony? They go under a process known as division of labor, which we know very famously like economics. Like what happens, half of the cell decide they are going to use aspartate as a carbon source, grow and forage the media around it. And some cells, they decide, no, we are going to use aspartate as a nitrogen source and we are going to do something different. That's why you see in this colony, even it's a single colony, you see like there are two different type of cells. There are dark cells and light cells. So this happens really, like we do not notice these things a lot, but these things are very much relevant in biological system. And the last and the like my personal favorite is cheating and cooperation. I mean, we know like in human societal setup, we have a lot of cooperation, we have a lot of cheating, but that goes on also in the case of yeast. So how does it happen? So it's a very famous concept from game theory. I mean, I mentioned game theory in my abstract so as to just touch on this topic. So what is game theory? It tells us about how do individuals interact with each other. So if you are growing yeast cells in sucrose, let's say you have a medium having sucrose. So sucrose needs to be break down into fructose and galactose, right? So that the glucose and fructose can be used by the cell. So the sucrose needs to be break down into glucose and fructose. So the cell express an enzyme, which is your SOC2, and SOC2 breaks down your sucrose to these two monomers. But just imagine this situation, just try to get the intuition. You have cells in your system. Some of them are expressing the enzyme SOC2 so that it can break the sucrose. But the glucose and fructose is for everyone, right? It's not just for that enzyme because this enzyme is actually expressed extracellularly. So the glucose and fructose is diffused into the extra, like the media. And there can be some population of yeast which can spontaneously arise, who will say, we are not going to produce any enzyme. We are just going to take away the glucose and fructose that the other yeast produce. And this is what we call as a cheater strain. They are not doing any effort, but still they're consuming glucose and fructose and still growing. And you have this very like cooperator strain, which is trying to be very good, but again, it has a load on its metabolic machinery to produce this enzyme. That's why it is most supposedly going to have like a retarded growth rate. So these are the phenomena that happens in the population level, which have become like really interesting in the coming days. So let's come to the topic and ask the question about like how are all these things connected to biofuels, and why do I personally think these things might be like really an important clue to making organisms that are efficient producers of biofuel. I mean, of course, like we know a lot about global warming, about all the things that are happening around, but there are certain things that we do not discuss very much. So we know like the global warming is causing the increase in global temperatures. There are a lot of things that happen, But these are two things which I personally think are like really important. So some papers have shown that long dormant viruses and bacteria which were trapped in ice are coming out now. So what used to happen when we used to have like normal temperatures around us, normal carbon dioxide. So your ice sheets in some of the places used to melt, let's say 50 centimeter, and when there was again the freezing of ice, you used to like anneal back to like normal height. But now what's happening due to the increased temperature, it's melting beyond 50 centimeter, and it's going, let's say 125 centimeter. And there are many pathogenic bacteria and viruses which are trapped inside the ice sheets, which might be coming up now. And we don't know. And this is one of the beautiful thing that was published like maybe a couple of years ago. And this is a serious concern. The second thing is the decrease in nutrient content in fruits and vegetables. It was published by the Harvard THN School of Public Health. And what they showed is, okay, this is a very simple concept of mass transfer. So if you have carbon dioxide in your environment and the carbon dioxide is being taken by plant, if you have more carbon dioxide, plants can take more amount of carbon dioxide because their uptake rates will increase, and slowly they will start getting bulkier. But only thing that they can make out of carbon dioxide is carbohydrates, right? So like, or let's say lipids, like all simple molecules, but it cannot make nutrients, right? Because nutrients are taken from the soil. So you have this same amount of nutrient in the plants, but your carbohydrates have increased or your other components have increased. So basically what has happened, the per gram nutrient content of fruits and vegetables has decreased. And this is like a very big concern because we don't want the nutrient content in the fruits and vegetables to disappear. So that's why it's really important that we come up with strategies to like propose alternatives for petroleum and like the existing fossil fuels. And some of the very important challenges in biofuels are getting a very good titer. So what is titer? Titer is the grams per liter of the biofuel that we get, and it's really important because it has a role to play in the separation energy. Just imagine you have like 1 liter of your broth which produces, let's say 300 microliters of, let's say, butanol or isobutanol. You're not going to separate it that easily, right? You will put a tremendous amount of energy. And if you look at the calculation, it looks something like this. If your ethanol concentration increases, your separation energy decreases. But if you are having ethanol concentration, let's say 2%, it's going to, your separation energy is going to be around 30 megajoules per kg. And unfortunately, the calorific value of ethanol is also around 32 megajoules per kg. So that's why basically you will not get anything out of it. You are basically putting the same amount of energy which you are getting back. And titer is also very much important for tolerance of the microbes. If the titer exceeds like astronomically, then the organisms are going to die. The second important thing is the rate. Rate is really important because we want the organisms to produce all these chemicals as fast as possible. So that was again like another of my interest, how to get organisms which are super fast growing and which can produce metabolites at an extremely good rate. And then we have the yield. Yield is about how much of the biofuel you are getting out of, let's say, per gram biomass like that you are putting into the system. So it's again very much important because we have limited amount of sunlight that's falling on earth, and then we have limited amount of biomass, and photosynthetic machinery itself is a very inefficient process of generating biomass. We know like it's hardly efficiency is around 1%. So that's why it's really important for us to increase the yield so we can get the best out of the biomass we have, right? Then we come into this very beautiful topic, what we call as metabolic engineering. And metabolic engineering has been hugely successful. I'm not very much sure about biofuels. Biofuels are still, like we are still trying to do a lot of interventions so as to overproduce biofuels. But for other compounds like fatty acids and other like molecules from which we can generate an myriad of products, they have been very much successful. So we do metabolic engineering in like different ways. I think other experts today have already mentioned about like what is metabolic engineering. So I'd just like to show you like few images because like we believe in what we see. So this, you can see in the first image, you are having like some sort of globules formed here in your test tube. Those are actually fatty acids. This is from Jan McCord's lab, and they were able to show that like yeast produces a lot of these things when they are genetically engineered. And here you can see you have like control stain. So these have been most probably dyed with Nile red or body pie. And if you have like an engineered strain, it releases the fatty acid because the cell has to maintain its own osmotic environment, right? So it propels out all the fatty acids out of it. So here's another image which is showing like what a large fraction of the cell is taken by the lipid. I mean, like this is really interesting, but to convert that lipid into something we can directly put it into our vehicles is still a challenge. So that's why, what are the existing challenges of metabolic engineering? One is going for high throughput combinatorial screens. I mean, just take the example of a pathway having 10 genes, and in each gene, you can have like different promoters and ribosome binding sites. The ribosome binding site is important for recruiting the mRNA into ribosome, and promoter is important for with the transcription. And you can have like a lot of combination, right? Even if you have like 10 promoters, 10 RBS, and you have 10 genes, you can just calculate. It's simple. It's like a tremendous amount of experiments that we have to do. And the problem is in detecting amount of biofuel that is produced is itself a challenge. I mean, you can definitely stain the cells in this case, but let's say you want to produce isobutanol, let's say you want to produce butanol. It cannot be stained that easily, right, as lipid. So that's why, how are you going to see like which combination of all these genetic circuits are going to produce the maximum biofuel? So that's why that is one of the challenges. The second challenge is to make metabolic engineering very much quantitative. So currently what we are doing, the engineering that we are doing is a bit qualitative, because we say that we are either having a overexpressed version of the gene, or we are having an underexpressed version of the gene, or we are increasing the gene expression twofold, threefold. But we need to be very precise as to how much the gene expression from each of the genes should be in such a way that your net product is maximized. So in this case, we need to know like what combinations of promoter and RBS are going to give us the best gene expression that we need. And the third is to do a community scale metabolic engineering. So currently metabolic engineering is restricted to single organisms, although there are some groups which are trying to do it at a community scale. But again, the beauty of the community scale metabolic engineering is that you can go for division of labor, you can go for all different sort of tragedy of commons, and many things of main theories of economics can put into community scale metabolic engineering, and you can actually engineer maybe multiple cells at one time and put them together and let them do a task of, let's say, converting carbon dioxide into lipids, right? So this is like what metabolic engineering is. So I started my journey in this field by doing my first internship in the Great Lakes Bioenergy Research Center in the Michigan State University. It was a US department funded program in which I was selected. And what I worked was on Zymomonas mobilis. It's a microbe which produces good amount of ethanol, and it produces a very good rate of ethanol. So the rate of production is almost 2.5 times that of Saccharomyces cerevisiae. I mean, it's like one of the organisms that NREL, National Renewable Energy Lab, is trying to develop for ethanol production. And I already visited some of the fermentation industries there, which are already using Zymomonas mobilis for production of ethanol. And there's this very nice paper published recently in September 2020 in Metabolic Engineering from Robert Landick's lab from UW Medicine, in which they were able to show by changing your pyruvate decarboxylase, which actually tunnels your glycolytic flux into ethanol, you can actually produce your bioproducts. It's a very simple concept. What they proposed, you just downregulate the PDC, overexpress other parts of the pathway which can produce, let's say, isobutanol, butanol, or any other pharmaceutical compounds. So Zymomonas mobilis itself does not have the classical EMP pathway, which is present in, let's say, E. coli. That's the Embden-Meyerhof pathway. It actually has a different pathway known as ED pathway, which I think Pinaki sir already talked about. That's like Entner-Doudorf pathway. It's a different pathway. It's a bit less efficient. It produces less amount of ATP compared to EMP. So what was my task in this internship was to see what are the gap filling we can do in Zymomonas mobilis, taking genes from E. coli so that it starts growing fast. So the main problem with Zymomonas mobilis, although it produces a lot of ethanol, the growth rate and the biomass formation is a bit slow. That's why what we are trying to do is to increase the biomass. So we need to have like a genetic control in the biomass so that we can say, okay, let's switch on the biomass so the cell grows and populates the whole reactor. Then we switch off the biomass and then we turn on the ethanol production gene, and then it starts producing ethanol. So we wanted to do something similar to that. So it started with a prediction of metabolic flux analysis, and one of the collaborators of my lab was Jennifer Reed. She passed away recently. That's very unfortunate for us, but we used like metabolic flux analysis to predict what are the interventions we can do in the metabolic pathway so as to increase biomass formation. And what I try to do is to try to express these 4 genes. This is a Western blot of all these genes expressed in different conditions. And the most interesting part of this whole analysis was that of 4E. So 4E is a phosphate transporter inside the cell. And what I was able to see when you are trying to overexpress 4E, you see like a decrease in the biomass, meaning that this enzyme has got to do something. And because it's a transporter of phosphate, it directly interferes with the ATP metabolism. You know, like in the case of Zymomonas mobilis, it's a prokaryotic cell. It does not have like a separate mitochondria, which is doing the ATP oxidative phosphorylation. So this was quite interesting. Then I went ahead and tried to see what is the membrane potential of this cell. And you can see these are the two plasmids which are expressing the membrane, which are expressing the 4E in here. And these are the control strains. You can see there's a significant change in the membrane potential whenever I'm expressing 4E. So that's why, like, this is where I ended the project, and I was able to show that 4E may be one of the genes. I explored like a lot of genes, as I have mentioned here. There's TAL-A, which is involved in the pentose phosphate pathway, and some other genes. And 4E was the one which they were very excited about, and they are currently working on how to make a 4E tunable circuit so as you can control the biomass genetically. And then my second internship was at the University of Tokyo. I was awardee of Honda ES Foundation, and they gave me a scholarship to go to the University of Tokyo. And I already mentioned previously about how important it is to find like all the interactions in a cell. I mean, in the case of E. coli, it's a bit difficult, but it is not that difficult in the case of yeast. In yeast, you can do mating. So you can take like two different sexes of yeast and you can mate them to form a diploid cell. And using this process, what we name as a barcode fusion genetics protein fragment complementation assay, I developed a protocol along with a master student there. By which we can map the protein interaction networks in the scale at a genome scale. So this is like a small pilot scale demonstration. You can see like these 4 genes which are having growth here are the ones which are reported to be interacting physically. And by this method, we were able to very elegantly quantify all these interactions inside the cell. And the whole process works like this. You basically have a DHFR protein, which is like a dihydrofolate reductase, and we have split it into 2 different domains, and these 2 domains are having like different proteins with them. They are having like protein A and protein B with them. And then if these 2 proteins are interacting, then 2 fragments of DHFR are going to come close together, and they are going to reconstitute to form the working DHFR. So individual domain do not have any function because you have just taken away their counterparts. Now once they are close together, so they can very easily start doing their reaction. And if the DHFR is reconstituted, you get a growth on methotrexate. So methotrexate is a drug on which the organism can only grow if it has like the DHFR reconstituted. So then what we do, we have like something that I cannot reveal because this paper is still under review, and that's why there are a lot of things here. So basically we use next generation sequencing, correct, like in this process, to map all the barcodes. So to each of the plasmid that is containing a protein of interest, you are having like a barcode, and there is a Krelox based barcode exchange that takes place between 2 plasmids. And then we go for next generation sequencing. We sequence it very deep using NextSeq, which goes around 400 million like depth of read. And then we try to find out what are the interacting proteins. And this is like another small demonstration here. The right corner, you can see. So all the dots you see here are proteins that interact. So here you have like 96 proteins, and here you have 105 proteins. And we are trying to see like what are the proteins are interacting, and we were able to find all the possible proteins which we previously knew interacted. Apart from that, I again went to University of Tokyo in the winters. This was in the summer, and I again went back there like in the winters, and I tried to see how under different conditions. So what I was personally interested about is to use this technology in NAPIC. Let's say you have yeast growing in ethanol, you have yeast growing in normal media. What are the protein network differences that take place between 2 conditions? And I did some experiment, and I am hopeful that this paper will be out soon, and I'll be happy to share it with all of you. So this is like the one which I did in University of Tokyo. And then when I was back to Kharagpur, I started working with Professor Amit Ghosh sir, and the project was on building a genome scale metabolic model. I think Amit Ghosh sir already discussed about like how did we build the model. So I will just go briefly over it. So you can see like we saw a very good agreement between the growth rate that was predicted in silico and the growth rate that was predicted experimentally in earlier reported papers. So the major challenge with Lagrange Cluveri metabolic reconstruction was it's a fungus. In case of E. coli, or let's say in case of like other bacteria, it's a bit simple because you have like powerful platform like model seed. But for fungus, we do not have things very much developed. So that's why many of the analysis that we have to do manually, and we have to correct pathways manually. So that's why it took a long time to correct this whole metabolic pathway. But finally, we got something that was able to predict the experimental growth rates just using like a simple like 1 MB file. And that's like a very beautiful thing about genome scale metabolic reconstruction. You do not have to do any experiment. You just have to do some preliminary experiments on measuring what are the growth rates and what are the other like important uptake rates. And apart from that, it's able to predict the growth rate with unprecedented accuracy. So that's one of the great things about genome scale metabolic model. Apart from that, I also did like the sensitivity analysis in which we were able to rank the reaction based on what biomass precursors will be affecting the cells the most. So this was another interesting analysis. And apart from that, we also did dynamic flux balance analysis. And then Amit Ghosh already explained about this slide. So this was basically trying to validate whether our metabolic model is right or not. So how do we do that? We just try to see like the lethality of the genes in the metabolic model. So we try to remove one gene at a time, and we try to see like is the organism growing or not in silico. And then we try to take the data from experimental measurements and see when that gene was knocked out in some of the large scale gene deletion studies, was it able to grow? And this showed like a very good accuracy. It was around 80%, which is very good because we did the comparison with Saccharomyces cerevisiae, not Lacanchicloviri, because it's a non-conventional yeast and we do not have like much genetic tools to explore that. And then like during one of the reviews, we tried to do the flux variability analysis. And what I am currently working on is to somehow develop technologies to do differential flux analysis. I mean, we know like differential gene expression analysis or differential protein abundance analysis, these things have already been done a lot, but the main problem is flux is the ultimate predictor of phenotypes, right? Because gene expression can happen, the mRNA can be produced, then it has to go through multiple steps of post-translational and post-transcriptional modification in order to function. But what is the ultimate phenotype in a cell is the fluxes, right? So that's why what we were interested in is in doing a differential flux analysis. So we use flux variability analysis to map what are the minimum and lower and the maximum bounds of the flux. And then we use Jacquard index, which basically tells you how much overlap is there between two different conditions. And then we were able to highlight all the reactions between two conditions. So we took the gene expression data for the growth on ammonium. We took the gene expression data for the growth on uracil of Lacanchicloviri. And then we generated the context-specific metabolic model. So it's like a metabolic model in which you have pruned the reaction, you have cleaned the reactions which are not required for the organism in that particular condition. And then we were able to highlight different pathways. So definitely purine metabolism is one of the most affected pathways because you see like uracil is there and ammonium is there. So that's why the difference between them will definitely be in nucleotide biosynthesis. There were differences in histidine metabolism. And like we reported this, and just a few days ago, we got it published in Nature Scientific Reports. Like in September, it's available online and it's open access, so you can just try to read this paper. And then another part of my KGP was spent in Professor Ramakrishna Sen's lab, Sir's lab in the biotechnology department. And what I like in the beginning, I talked a lot about like the separation energy of biofuels, right? How like we are facing problems in separating very low titers of biofuel. So I was interested in developing a technology which can help us in separating biofuels from fermentation mixture without using a process of distillation. I mean, I know like most of the plants which produces ethanol are known as distillers. It's because distillation forms a very, very important part of the whole process. That's not just in terms of importance, that is also in the terms of energy. So distillation consumes a tremendous amount of energy in the plant. And that's why it's like a process which actually impedes the production of biofuel because no one is going to spend a tremendous amount of energy in just separating the fuel from the mixture, right? So that's why what I try to do is to use a technology known as salting out. So salting out is basically you add some amount of salt to your like fermentation broth, which is having your biofuel. And then you see like a phase separation of the fuel whenever you add like the salt. And we were able to show that it's very good at separating ethanol. So this was like just an in vitro reaction. So here we are just taking, let's say, ethanol mixed water, and then we are adding the salt. And you can see when the salt concentration goes around 40 to 50% weight per volume, you see like a separation of ethanol here. And then what I was interested in is just trying to do this in the fermentation scale. We already know about salting out, separating ethanol in aqueous phase, but what we don't know is like will that work in the case of fermentation broth. So that's why I just try to grow yeast in our bioreactor. It's a 2.5 liter benchtop bioreactor, and we try to measure, we try to extract the broth, and then we try to add the salt and see if we are seeing some separation. You can see like there's a separation of organic layers, and and this top layer is actually ethanol. It was a very, very euphoriatic moment for me. I mean, I was very excited because this is one of the first time we are able to separate ethanol without using any tremendous amount of energy. And like many things here are under patent. So we filed an Indian patent in October 2019, and it's filed, although it's like not international. That's why like there are many things I cannot disclose here. But again, so like this is a very important thing that I learned through my journey in IIT KGP. So one of the things was like whatever experiment we did here was mostly in lab. So we use pure YPD. We use like YPD is the growth media for yeast. We use like very well grown yeast cells which have been adapted to laboratory condition. And I mean, like it's fine. We can definitely show some result and definitely publish some papers. But the important thing is whether this is going to work when we scale it up into industry. So that's why I went to IFB Agro. It's a fermentation industry nearby. From there, directly from their fermenters, which were like 200 meter cube fermenter, I took out the fermentation broth with the help of their engineers, and I brought them back to IIT KGP lab. And by that, I was able to show we still get the same separation. So this was again a very important moment in this whole research, because we were able to show this technology also works when we take the noisy and like heavily contaminated broth from industrial systems, because there we do not actually care a lot about sterility and all those things. Their most important thing is how much we are able to get with the minimum cost, right? And I was able to show this ethanol separates very elegantly in this fermentation broth when we add like the potassium carbonate solution, which I used for the salting out. And what we were again able to show, so there's another challenge here. Okay, now you mix potassium carbonate with the fermentation broth, you separated out the ethanol. Now you can use it in your like fuel application. You can decolorize it and do all those things. But now how do we get back those potassium bicarbonate? Because that is very much important from a green chemistry perspective, right? And it's not like waste resources. So that's why I realized there's a competing group of researchers who are trying to see whether you can capture carbon dioxide. And one of the best candidate somehow like coincidentally is potassium carbonate. So what you can basically do, you can take potassium carbonate solution, you can pass carbon dioxide with at some elevated temperature, and that's going to precipitate out the potassium bicarbonate solution. And that's one of the most that's one of the famous technique people are using for doing carbon capture. And I realized that, okay, my fermentation is also producing a good amount of carbon dioxide. Why cannot we integrate both these processes? And that's what we did. And this is the technology for which we filed a patent. So basically what is happening, you do fermentation, you get the broth, and then you add the salt, you separate out the ethanol, and whatever is remaining back is having rest of the salt, but you are generating a lot of carbon dioxide from your CO2, from your fermentation plant. You pass back the carbon dioxide through the potassium carbonate, and then you precipitate out all the potassium bicarbonate. In an alternative way, this would have been really difficult, because separating salt out of a mixed solution is either people go for thermal evaporation, which consumes a tremendous amount of not just energy, but also time. So here it's a rapid separation. You can see this image here. Within like 10 to 15 minutes, we get a tremendous precipitation of potassium bicarbonate. So you can take potassium bicarbonate, you can heat it back, and then you can get back the potassium carbonate from there. So now I'm currently doing like the life cycle assessment analysis, because whatever I'm saying is like, it might be sounding a bit interesting, but again, like until and unless this has like a net energy positive outcome, then like I don't think there's any meaning of taking it to industry. So that's why that is one of the analysis I'm doing, and I think it's very important. And apart from that, what I'm trying to do is to map locations where we can actually store carbon dioxide. So this is another paper in PNAS, which was published maybe a year ago, like 2018, I guess. So where they were able to show if you want to actually capture carbon dioxide, the best place to start is fermentation industry. Why? Because you are having like a competing group of researchers who are actually trying to capture carbon dioxide from air, what we know as direct air capture. But you know, like carbon dioxide might like 413 ppm might be sounding a lot for us who believe a lot in climate change. But for if you ask any chemical engineering, they say like 413 ppm is nothing. It's not going to go for any mass transfer. So that's why DAC, direct air capture process, is very energy intensive. So why not use fermentation? Because what you can do with fermentation, you are almost getting 99.9% purity carbon dioxide out from here. You just take that carbon dioxide, you capture it into geographical locations where you actually store carbon dioxide, geological carbon sequestration. And if you look at the whole picture, what's happening, if you talk about photosynthesis, you have 6 CO2 getting fixed in photosynthesis, and you are forming glucose. But when that glucose is fermented, you form like 2 ethanol and you form 2 carbon dioxide. The 2 carbon dioxide can actually be stored in geological reservoirs. Ethanol you can definitely sell, and people are going to use it, they're going to burn it, and they're going to release carbon dioxide, and that we cannot avoid. But you can take 6 CO2 in and you can capture 2 CO2 out. So in net, you are producing CO2, but you are also consuming it, right? So if you repeat this cycle several times, you will start capturing carbon dioxide. And that's how I think like we can really go for carbon negative emissions. that I think is like really important at this point of time. You can also have like power plants nearby so that you can take their carbon dioxide and also capturing them. So that's why I just did a geospatial geoinformatic analysis in which these red dots are all the carbon dioxide reservoirs in the Indian subcontinent towards like the western coast, and the blue dots are all the fermentation industry. And then try to draw a distribution of the distance between them, because the distance is a very important factor. We need to transport carbon dioxide through pipeline through thousands of miles. So that's why like I tried to see what are the prospects, and it looks like we will be able to implement such a technology if it looks feasible through our other analysis like life cycle assessment. Then this was what I did in my fourth year. Then in my like the end of my fourth year in my summer internship, I went to Harvard Medical School through Corona program, and there I worked on something very interesting that's again related to biofuel. I mean, that was on an organism known as Vibrio nitrogens. If you just Google this name, you will get to know that this is the fastest growing microbe ever reported. So this grows as a like super fast rate. That's around like your doubling time is around 10 minutes. It can also go up to 8 minutes if you have the right conditions. And you can just see this simple graph here that's from your Crick Synthetic Biology Institute. And what you can see, your red line is the Vibrio nitrogens and blue is your E. coli. Just a simple thing to note here is you see where the E. coli completes its lag phase and starts getting into the exponential phase. The Vibrio nitrogen completes its log phase and starts getting into the stationary phase. I mean, it's super fast growing. Even with like I have personally worked with this organism, and if you're just inoculating it in a test tube with this media, if you come after a couple of hours, you can see like a good amount of turbidity. So that's why like I was very much fascinated, because why not use this organism for biofuel production? Because this is something that would really alleviate the problem of lower rates, right? So this is a microfluidic experiment from George Church's lab, where I'm going to do my next rotation. So in George Church's lab, they were able to capture, this is Vibrio nitrogens and this is E. coli, and the time frame is same. You can see in the same time frame, you have like Vibrio nitrogens populated in that microfluidic channel, like in good quantity, and you see like E. coli just growing into like few cells. So that's why from here, the message is Vibrio nitrogens is super fast growing, and then it can be used for like metabolic engineering purposes, and some people have already started it, like particularly in Bastian Bombach's lab, University of Munich. And what my project here was to do a single cell analysis. I mean, I'll talk about single cell a bit more when I come to my master's thesis project, but single cell analysis is completely revolutionizing the way we used to look at science, right? So previously I showed you how organisms behave in bulk when they are in in populations. So it's really important to know like how individual cells are behaving in a given population. So for that, we use something known as microfluidics. Microfluidics is a very good contribution from mechanical engineering and thermal science department like place. And what we can do in microfluidic channel is you can trap each and every cell and look at each and every cell individually. So you see here under the microscope, this is an inverted fluorescence microscope. This is actually a live imaging microscope with an incubator. So you can see an incubator, you can control the temperatures. And here you have like a microfluidic chip in which you trap the cells, and then you try to do microscopy, and you try to measure the fluorescence or whatever property, phenotypic property you want to measure at a single cell resolution. And you can also measure growth rate. So that's all that comes under image processing. Like I did a lot of image processing in my master's thesis project. I'm going to discuss about that. And these are the setups that I used there to do all this analysis. And so what was my project about? Where did I start? So the first was definitely cloning. That's like one of the prelude to whatever we do in synthetic and systems biology. So what I wanted to do is to take like different strength of promoters and different strength of ribosome binding site and do all possible combinatorial pool and try to see what is the gene expression noise in each combination. So here you have like 4 promoters. So these are in your decreasing order of strength. And here you have 4 ribosome binding site. So these come from a collection known as CIDAR Moklo collection. If you ever want to just like do like high-end synthetic biology in which you have like, or you already know what are the strength of the promoters, you already know what are the strength of RBS, and you can definitely go for CIDAR Moklo library. It's CIDAR-MOCLO. It's like not very costly. You can get it in AdGene, and you can use that library to generate your combinatorial pool. So we use a technology known as Golden Gate assembly using BSA1 site. And by using this method, it's such an elegant method. What you can basically do is you have plasmids having promoters individually. So you have one plasmid A having promoter in it. You have plasmid B having ribosome binding site in it. You have plasmid C having a YFP in it. You You have plasmid D having terminator and plasmid E having the backbone, like the plasmid. You can basically pull all this plasmid together and take like a 10 femtomole concentration of all the plasmid. Then you can just add this golden gate mix, and all these circuits are going to join and they will form the whole genetic construct. This is basically like Subway. You go to Subway and you say like, I want this, I want that, and you just put all of them together and you get like a sandwich, right? So that's how it happens here. So you basically say like, okay, I want promoter of this strength, I want RBS of this strength, and I want this fluorescent protein, I want this terminator, and you can get like a genetic construct out of it. So I tried to create a vector which they named, so my name is like PPN710 because I created it on 10th July, I guess. And this vector has like very good properties. We can transform it into cells, and it can have like different combinations of promoter and RBS. Apart from that, it also has like a constitutively expressed PFP protein. So what it is basically accounting for is the levels in the plasmid fluctuations. So it's very easy to imagine, okay, if like someone says like the copy number of plasmid is 20, it's 20, but it's not. Like in the case of systems biology, we know like there are variations within cells about the plasmid copy number. And that's why we need to normalize for that, because if you have 10 plasmids in one cell and 20 plasmid in the other cell, then whatever fluorescent readout we are getting is not actually due to the promoter and RBS. It is actually due to the number of copies of the plasmid, right? And this last image you see here is one of my personal favorite, because here you can see I have taken different combinations of promoter and RBS. It was a lot of cloning. It was a lot of high throughput cloning actually. And you have like decreasing strength of RBS. Here you have decreasing strength of promoter. And you can see the one having the strongest promoter and strongest RBS is giving us the maximum fluorescence. And as you go down to less amount of RBS and less amount of promoter, you get like the fluorescence fades away. So it showed us like how we definitely knew like whenever we take from different like high strength of promoter and high strength of RBS, of course we get like the highest expression in that case. But what we were personally interested about was about finding out what is the noise in each of them. So here it's just a streak that I've made on the agar plate. So here you won't be able to know about the noise, right? So for that, we need to go for some other high-end technologies. So for that, what we used is single cell microfluidics. So basically we use, so whatever images you see here are microfluidic channel. These are like very, very small channel around like 1.7 micron, let's say. And you have like E. coli cells trapped inside that channel. And if I don't have a video, but I would have shown when the cells grow. So one of the end is closed and one of the ends is open. So if you have the cells growing, it pushes away the other cell, like in the open end, and you can get like the whole time lapse of the cells growing. And you can see like each of the channel contains one unique combination of promoter and RBS. And you can see there's variability within them. Within a single channel, you can see like there is differences between them. So that was the interesting part of this analysis. And I also developed, there's something known as dye barcoding. So now what you want to do, you want to pool all these different combinations of promoter RBS and try to see them under the same field of view, because you want to remove all the batch effects from this analysis, right? So that's why we use dye barcoding. So basically you can use dye, which are known as HADA and TADA. Those are actually FDAS. So those are fluorescent D amino acids dyes. So what they can basically do is they can dye your membranes differentially. So you can have one E. coli like Vibrio nitrogen cells having, let's say, promoter A and RBS A. You can color it blue. You can take another cell having promoter A and ribosome binding siting B, and you can color it red. So that's what I'm doing here. You can see all the test tubes have different colors. So I have dyed all the cells differently. Then we just pool all of them together, inject into the microfluidic chip, and then you get like all different cells here, and you will be able to study the noise within them and a lot of things. But here's something that I would like to connect it to metabolic engineering. So there's another paper published in Nature Methods from the same lab, in the Johan Paulsen's lab, where I worked through the whole summer in Harvard Medical School. And they published a method known as M-SIFT, where what you can actually do, you can use optical tweezers to separate out of your cells of interest. So whatever I did with promoter RBS, and I was expressing, let's say, GFP, you can take that promoter RBS and you can put it along with some biofuel producing enzymes, and you can take like different combinations of promoter and RBS. You can put some enzyme that's important for biofuel production. You can have some systems so as to detect the fluorescence from the biofuel. And if you see one combination working very well, you can just take 100 promoters, you can take 100 RBS, you can combinatorially clone them together using golden gate assembly. Of course, using robots. These are not like to be done by human beings. You have like robots like Tican, who can do all these tasks very well. And then you get like a huge combinatorial pool having like 10,000 possible combinations, right? 100 into 100. And you don't know which of them will be producing the best biofuel. Like normally you have to grow them in separate conical flask, and maybe someone will tell you like do it in triplicates. Then you cannot do like 30,000 flask cultures, right? That's not possible. So you don't even want to do, right? Right. So that's why you can just pool all of them together. You can dye barcode them. You can put it into microfluidic chip, and then the cell lines that you find interesting, which are producing very good amount of biofuel, you can basically use optical tweezer. In 2018, I think they were awarded the Nobel Prize for physics for discovering the optical tweezers, for inventing optical tweezers. You can use optical tweezers to catch a hold of a single cell, and you can push it back into a single like a microfluidic boat, and it will come out into your conical flask. So basically what you can do, you can take a lot of different combinations together, you can pool all of them together, grow them in a microfluidic chip, and then whatever is of your interest, you can just pull it out and do a conical flask, and that's your cell line. Then you can just take it for your downstream application. You want to do HPLC, GC, and all those things. So this was quite interesting. So when I was coming back to India during my 5th year, I started thinking about how can we leverage this single cell analysis for biofuel production. And then these are the things that came to my mind. I mean, we know like genetic noise is very much predominant in biological system, and that is mostly at the level of gene expression. We also have noise in different other things, like we have noise in translation, we have noise in chromatin remodeling, we have noise in various other phenotypic properties and also genotypic properties, right? So that's why my question was, okay, what if we are able to measure the noise in biofuel production? I mean, if the gene expression is not same in all these cells, that's what I learned through my summer, then of course the biofuel production in all these cells won't be the same. So my proposition was, what if a cell is producing less amount of biofuel? Won't it grow fast? Because it does not need to really care about like producing biofuel a lot. But some other cell is like really serious about producing biofuel, and it's producing a lot of biofuel. Won't that impart its growth? So you will get like populations of cells inside a reactor, right? Some of them will grow fast, some of them will grow slow. And we need to make sure that all of our organisms are actually producing the biofuel in good quantity. It's not like there's a normal distribution of biofuels there. That's what we want to ensure. So currently what we have been doing a lot in metabolic engineering is to increase the mean, right? We just want to push the mean. We want to increase the average biofuel production that we get out of the reactor. But what if we can change the standard deviation itself, and then also we can increase the biofuel production? We can just make all these cells produce high amount of biofuel, and then we can definitely increase the biofuel production. But one of the first precursor to such an analysis was to find out, okay, how do I measure the amount of biofuel production at a single cell resolution? Then I came back to KGP, and this was part of my master's thesis project. I had like 3 objectives in my mind. First is that I wanted to quantify the variability in the single cell lipid content. Why lipid? Because it's easy to visualize. I mean, had it been butanol, isobutanol, it's a bit difficult. I'll discuss towards the end how can it be made easy. But like lipid was easy to visualize, right? So that's why to quantify the variability in lipid content and its trade-off with single cell biomass, my question was, if your cell is having actually a lot of biomass, is it producing less amount of lipid? Because your resources need to be diverted between growth and for like lipid accumulation, right? So that's why there must be some trade-off between them. The objective 2 was to see what is the different trade-off between growth rate and lipid content. So biomass, the amount of biomass and growth rate are 2 different things. So that's why I just also wanted to see if some cell is actually having a lot of lipid. Is it getting bulkier? Is it getting bored with metabolism so that it's not growing? So that's what one of the objective. And for that, I had to develop some microfluidic techniques. And apart from that, the objective 3 was to propose a method how to reduce this noise. I mean, it's quite interesting to study all these things, but there should be something in my mind also how to like, elevate this problem of noise if it's there. So that's what, there was one paper in Nature Communication from the Grigory Stephanopoulos lab at MIT, in which they were able to show there is actually a trade-off between biomass and productivity. So if you are having more growth, if you are having bigger cells which are having a lot of biomass, by biomass I mean lipid-free biomass, then you will be having prospectively less amount of cells. So they did like a single cell analysis using quantitative phase microscopy, and they were able to see this trade-off exists. And this is the model that I propose. If you are having like higher yields, then your like mode is greater than the median, and if you have like lower yields, definitely your mode is less than the median. It means like most of your cells are actually not producing a very good amount of lipid, whereas only a small fraction of cells are producing a very good quantity. We want this one, the first one, right? And this is a confocal image that I took of Yarovia lipolytica cells. You can see like the cells are stained with Nile red. You can see like the number of lipid molecules here are variable, even they were grown in the same condition. You can see there's variability in the lipid content. And then my next task was to see what I can do out of this. Like how can we use this information. Right. So what I try to do is to use flow cytometry. Flow cytometry is an excellent technique if you want to see the variability within like fluorescent molecules between different cells. So basically I grew the cells in a bioreactor, in a 2.5 liter bioreactor. Then I just assumed the fact that if I stain the cells with Nile red, then the fluorescence that you are getting like from fats will somehow tell me about how much lipid is there, right? So to measure lipids very accurately, you definitely need to use quantitative phase microscopy or Raman spectrometry along with it. But it's like, it was a bit difficult for me to do at this stage. That's why I just assumed like the lipid content was proportional to the fluorescence that we get from the lipid, which is not actually very much wrong. And then you also have a channel in fluorescence cytometry, which is known as FSC, that's the forward scattering, which tells you about what's the size of the cell. And I assume that will be proportional to the gram mass of the cell, which is also not quite wrong. And what I was able to show as the fermentation proceeds, you can see there's a decrease in cell size. So just one thing to note, these cells will grow in high C/N ratio. So this is the condition in which you trigger the lipid production. I wanted to see what's the noise in the lipid content whenever you have like high C/N ratio. You can see like the average size of the cells decrease whenever you go ahead in the fermentation. So it says like and here you see the lipid content, you see like the single cell lipid content. So this is not a bulk measurement. Normally whenever we do bulk measurement, we just take like the total amount of yeast, we sonicate it, then we use like enhexen and other solvents like dichloromethane to separate out the lipid. But this is not that. This is the measurement of lipids at a single cell resolution. So whatever you are saying here are single cell properties, and the violet plot is showing the distribution inside the cell. So whenever cells are growing, you can see there is actually a decrease in cell size, which was very much similar to what Grigory Stefanopoulos lab reported, and which was very much similar to what I thought of. Is like there will be a decrease in cell size whenever the cells are growing, while there is an increase in the lipid content, because you see there's a trade-off, right? This is decreasing and this is increasing. It means there's a trade-off between like the lipid content and the cell size, right? And then what I tried to calculate was the single cell lipid content. What if I divide like the lipid content, that is your this one, by the figure B, like the values. So you basically get how much lipid is there per gram weight of the cell. You can see that that is also increasing. Of course, it will increase because it's a stressed C/N condition. And then I tried to measure the noise. So for measuring noise in the case of flow cytometry, we use like a part of statistics that's known as robust statistics. So normally, if you are using coefficient of variation, it's like a bit sensitive to outliers. But if we have like robust coefficient of variation, it takes account of all the outliers. You take like robust standard deviation and robust mean. And I just try to measure what is like the like noise here. You can see like when you are growing the cells from 12 to 24 hours, whenever the cells are becoming more confluent, they are trying to grow fast. And because there you have like sufficient amount of glucose. But once they reach 24 hours, your lipogenesis is triggered when the cells start accumulating lipid. And then you can see there's an increase in the noise. So RCV is somehow telling us about the genetic noise, right? So that's what I was able to show. When the cells get into more and more stressed condition, they try to accumulate a lot of like lipid, but that lipid is variable. The noise, the distribution of the lipid is very rapid. So my next question was how to measure the single cell growth rate. So this is a very simple agar pad experiment which people do just to initially see and just to standardize things. So you can see like I grew the cells on this agar pad, and you can see there's a live cell imaging going on here, and you can see like the cells growing on. Then I used image processing to find out like what is the growth rate of each and every cell in that reactor, in that agar pad. But that was not a very efficient system. So I developed like another system for which I'm like filing a patent currently. That's known as Camber, single cell analysis using microbial. You can see at the design how simple it is. I just took like a 1 ml pipette. I just cut it half. And then I just put 2 tubes inside it. And I just covered it with microscope. And this whole setup can be placed on an inverted fluorescence microscope. and then you can just use a syringe pump to pump your media. And I have immobilized the cell on the glass surface using concanavalin. And then when you grow the cells are stuck to the surface and they start dividing there. And as the cells are dividing, I can measure the single cell growth rate. And what I can do, I can just also measure the fluorescence at the end using Nile red, and then I can just superimpose both the images using something known as template matching, so that I just get like the positions of lipid. Then what I can ask is the cells which are actually growing fast, what are the lipid content in them? And then I can just draw this table, like how many cells are having high growth and high lipid, how many cells are having low growth and high lipid. So I can get like a contingency table. From here, I can do statistical test and find out like, is the hypothesis right that the cells which are having like less amount of lipid are able to grow fast compared to the cells having more amount of lipid. And that's what happened. And I like did this experiment. You can see here the flow, like blue color light, which actually excites the Nile red. And here you have a very nice time-lapse image. You can see like the top cells, the black globule-like thing you can see inside the cell are the lipid content. You can see this is all taken in the same time frame. The cells which are actually having like lot of lipid within them are not at all growing, and the cells which are having like some amount of lipid in them or few amount of lipid in them are growing fast. And I was able to say there are like a significant enrichment of cells which are having a low amount of lipid and high amount of growth rate, and like high amount of lipid and low amount of growth rate. Actually, majority of the strains were having like less amount of growth rate and high amount of lipid. What we want to do is to also increase the growth rate, right? We want the cells to consume all the glucose possible in the media to convert them into lipid. So that's why this was one of the interesting thing I derived from this study. And then the next thing was to propose as to what I can do from this analysis. So there came about like proposing a system, and then like COVID-19 pandemic occurred in between, and I was not able to continue the experiment. So what I basically wanted to do is to see why is this noise coming? Is it because we first grow the cell in a high growth conditions like a low C/N ratio, and then we spontaneously transfer it to a high C/N ratio? Is that the reason why you are having like all these cells disturbed and they are just trying to adapt to different conditions? That's why you increase the noise. That's why what I wanted to do is to just make it a one-pot reaction. So industrially also it is very much infeasible to first grow the cells in one big fermenter in low C/N ratio, then transfer the whole thing into a new fermenter. There are like many costs incurred in this process. What I wanted to do, I wanted to take C/N ratio, like a low C/N ratio, and I wanted to spike zeolites into it. So zeolites are minerals which can actually absorb ammonium from the solution, and that's going to change your C/N ratio. So what I wanted to see is the whole process able to change the C/N ratio in a more uniform way. And then I would have done facts and seen, is that changing the genetic noise within the system? But again, like as I mentioned, the COVID-19 pandemic came and I was not able to do this, but this is an open idea. I mean, if any of you would like to try, I would be like more than happy to share with you whatever I was trying to do. And from this, maybe we can see like improvements in lipid production by minimizing the noise. If we have more continuous depletion of C/N rather than a like binary depletion of C/N, because normally we just grow it in one C/N, we just throw the cells into another C/N ratio, right? And that's why it's really important, like for us to develop technology where we can make this transition more smooth, right? And then I come to the end of the whole lecture. And what I want to say is like all the things that I explored made me realize biofuels is definitely a systems biology problem, as cancer is called as a systems biology problem. I think biofuel is also a systems biology problem, because you have so much of the molecular machines involved here in producing all that we want. You can see like here is a eukaryotic structure. You have like tremendous amount of complexity. You have transcription factor that needs to come close to the binding sites on the chromatin, and then it needs to recruit the histone acetylases, then it has to open up the chromatin, needs to make the promoter accessible, then another RNA polymerase needs to bind, it has to produce the mRNA transcript, then mRNA transcript go through several processes of like maturation, and then from there proteins are produced. But that's not the end of the story. Even proteins fold in different ways. There is even noise in protein folding, and then you have proteins getting allosterically inhibited or activated by some other proteins. You have like proteins getting phosphorylated. There are a lot of post-translational modification which triggers like inductive effects on the whole protein chain, and that changes your active site. So there's a tremendous amount of complexity. So how, according to me, metabolic engineering maybe will look after a few years is we'll be doing engineering at the scale of the whole system. Like not just we are replacing promoters, not just we are replacing proteins, we are also trying to change the chromatin structure. We are also trying to understand the molecular circuitry involved in that cell. And maybe like this will be one of the ways which we can improve biofuel production because it's very important for us to understand that whatever we see inside the cell has been optimized through millions of years of evolution, and out of nowhere we cannot just easily change this whole cell into something that is going to overproduce biofuel. We need to first understand the whole system, and then we need to strategize in such a way so that we conserve the important parts of the cell which are required. Like one of them is definitely cell membrane. Like people are also trying for cell-free biofuel synthesis, but like the importance of cell membrane has got to do with like a lot of things, like you can read about it. And then like it's very important for us to work at a system scale. And then I would also like to talk about like microfluidic interventions which have been done in this field. So you have like microfluidic setups which you can use to measure the amount of biofuel. So here's this chemical known as resorphin and also resazurin. So basically, if you want to measure the amount of alcohol in individual cell, you can use droplet microfluidics. So what you can do, you can take droplets, you can put single cells inside those droplets, having your unique metabolic combination like combinatorial pool, and then you can stain them with resazurin. And then if there's an alcohol, then it will be oxidized and you will get some fluorescent out of it. So it's a very good system if you want to optimize the production of alcohol-based biofuels like isobutanol, butanol, even ethanol inside cells. And then what you can use, like the MSEP system, which I described earlier, like just catching one single cell and isolating out of the microfluidic port. That is very much possible. I mean, there's a complete single cell core at Harvard currently, and they are working a lot on this droplet microfluidics, how to isolate individual cells of interest. So this is what I would like to say at the end. I mean, the future is definitely bright, given that there is so much of knowledge that's accumulating in this field, particularly in understanding the whole system scale of how we are having metabolic regulation, we are having transcriptional regulation, and a lot of exciting things are coming out. What I say personally is now we are looking at individual cells, but many at a time. So we are not just recording what's happening inside a single cell, but we are also recording what is the variability between different cells. And I think all this knowledge will somehow help us in engineering better cells and make this earth a better place to live, possibly in years to come. So by that, I would like to just end it. Thank you.
Thank you, Piyush, for your very charged series lecture. So we have a few questions from Mr. Abhishek Chattar. He has two questions. The first one is, how could we confirm species endemism using DNA barcoding and/or phenotypic analysis? And the second question is, what would happen if all the colonies of the DHFR PCA survival selection medium grow at the same rate?
Right. I think these are like very important question. The first is like definitely finding the endemism, which we call as like how like the ecological state is there in a defined geographical location, as we know. So we can definitely use barcode. I am not really familiar with any of the papers which have been published recently, but the beauty of barcode is you can do pooled analysis, right? So normally when we do next generation sequencing, we just take normal cells and we just lyse it, take out the DNA, do the next generation sequencing. But when there is barcode, you can actually take multiple cells. If you're talking about community, you can use some transposon based system to like enter barcodes into your cells in a community level. And then you can just take the whole community together and do next generation sequencing, because you have the barcodes in your adapters. By that, maybe you can confirm as to like what is the composition of the community, and you can definitely do metagenomics. And I think barcode technologies are being used a lot in metagenomics and all, because they help us to pooled analysis. The second question was regarding the DHFR. Of course, there are like many trade-offs with DHFR, and like whatever you mentioned, those we see those type of things inside like the cellular processes. And that's why what we try to do, I will not say like our technology is completely elegant. We have like complementary methods. So there's another old method known as BFG-Y2H, which is these two hybrid. So basically whenever we do the analysis, what we do a like barcode fusion genetic Y2H assay. We also do an barcode fusion genetic PC assay. And we often see like they do not have like a good overlap. I mean, the number of protein interaction which is detected by PECA is very different from the one which detect from Y2H. So that's why it's very important for us to do complementary analysis and to confirm whether these proteins, I mean, for protein interaction network, there are several methods. Like you have like co-immunoprecipitation, which is a bit low high throughput, but there are people who are trying to make it high throughput. And you can definitely confirm it by other methods. I mean, this was one of the best we could come up with. because Y2H has a very important limitation. So Y2H cannot take place in the cytoplasm. So if there is any protein interaction taking place specific to cytoplasm, we cannot capture it using Y2H. So that's why we developed BCA, because it is very good for doing protein interactome analysis in the cytosol and also in other compartments. So that was the purpose. I mean, definitely you are right. There can be several trade-offs, but I think like technology will come slow as to overcome those challenges.
Thank you. And there is another question by Ms. Shikha Thakur. Did you consider using algae as the biofuel production platform? The green algae,
Right.
which accumulates biofuel in it naturally, can it be exploited in this regard and be engineered for better yields?
I mean, absolutely. I think like microalgae, there's a tremendous amount of research which is going into microalgae research. I mean, there's Center for Algal Biotechnology in like California. There are a lot of people who are into metabolic engineering of cyanobacteria. I think the future is prospect very high, like very high prospect. The only important thing at this point of time is we need to try different combinations, because we do not know what's actually going to work, right? Because it can also happen that sometime like some other or like a group will come and they say that there is this one fungus which just produces biofuel. It produces minimum amount of biomass. So that's why it's very important for us to explore. Cyanobacteria and microalgae are definitely, I mean, I also worked on cyanobacteria and microalgae, but the presentation would have been lengthy. That's why I just avoided presenting it. So microalgae are like really powerful, even cyanobacteria. The good thing about them is they directly take in carbon dioxide. You don't need to care about sterility and all those things. Whoever have worked with microalgae, they know like we just do it on bench. We don't even like need LAF most of the time. So that is one very good thing about microalgae. The only bottleneck is you get some amount of contamination of bacteria and all those things happening in raceway ponds and all. But definitely there are a lot of people who are working on microalgal, cyanobacteria, metabolic engineering. And you can read, there are several papers already out there of people producing isobutanol, fatty acids, and other things just from carbon dioxide. So yes, definitely microalgae is one of the prospective hosts for biofuel production.
We are done with the Q&A session.
Piyush, I have one question. So, hello Piyush, can you hear me?
Yeah. I can hear you.
So I have one question. So it was a very nice talk and very informative. So you talked about the ratio thing. The microbe choose the ratio between two substrates. So I think there must be some regulation also. Usually we do experiments with 5 gram per glucose, 5 gram per liter glucose, but we do not use usually 25 gram per liter of glucose.
Right.
So definitely, what is the limitation? What is your?
I mean, like I'll just like share my screen for a second, just as to press on this one. I mean, I think I already showed like there's already events in the cell in which they are able to respond to galactose glucose concentration. And I think the ratio is a very important thing. Cells actually go for ratiometric sensing. It's a very, very emerging field currently, which is the ratiometric sensing of metabolites inside the cell. So like definitely that would be one of the things to explore. Like, of course, that's a great idea. We can definitely go for seeing if we change the C by N ratio. I hope like that's what you were talking about. Like if you change like the C by N ratio and see like how the transcriptome is changing and how the downstream regulatory mechanisms are changing, we can definitely find that. And then it would be really important because, you know, like I worked with all this fermentation, like what I showed you earlier. And the problem is, you know, like many yeasts are able to tolerate high glucose concentration, but not all of them are able to. Like normally we grow the cells in the laboratory in let's say 10, 20, 25 grams per liter of glucose. But in industry, they grow it in like 200, 300 grams per liter because they want to maximize the titer of the biofuel. So that's why we don't know like what are the differences between those conditions. There must be some ratiometric sensing which actually dictates how metabolism goes on. And if we start knowing those things, definitely we can design better bioreactors, better continuous systems, which will help us in probing all these events very precisely.
Thank you, Piyush.
Yeah.
Thank you, Piyush,
Thank
for
you,
your
Piyush,
great
for a wonderful talk, a very detailed talk.
Thank you, sir.
So, yeah, I mean, you have not spoken about SARS-CoV-2.
Yes, sir. I mean, like it would have been a bit lengthy. That's why I just kept it.
Right. You know, we have time also. No problem. Anyway, thank you for all talk. It was nice having you. So I don't know when you will give the next talk. So we'll wait for that.
Definitely, sir.
Thank you so much.
Thank you, sir. Thank you.
So thank you. We'll now conclude the first session, and there will be a break of almost 1.5 hours. So the next session starts at 3:30 p.m. Indian Standard Time, and we hope to meet you all again there. Thank you very much.
We'll go for a lunch break, have lunch, and come back. So we'll start this session sharp at 3:30. So be on time.